Hi everyone, for those who are viewing, we are running a bit late and we will be getting started soon.
Hi, can anyone hear me? Ken, can you hear me over there?
I can hear you.
Okay, awesome. Yeah, so for the viewers who are watching the live stream, just a quick reminder that we are running a few minutes late. We're going to get started in anywhere between 1 and 2 minutes. So actually, while I'm doing my talk, Yeah. Okay, everyone, we're really about to get started now. There, live stream is showing the transition. This is great. And I'm going to share my screen, and that way everything should start working out. Okay, this screen indeed. Share that, and boom. Okay. And yeah, it looks like the live stream is catching up. So far, everything is working nicely. Hello, everyone. Welcome to our first workshop of the year. We're trying something pretty new here, a hybrid workshop, meaning that most of you are online and only some of us are on location in San Francisco. And, oh, I don't know why I'm suddenly getting a lot of Slack messages here. Wish that wasn't popping up. Anyway, so there may be a few snags here and there as we try to figure out how this works best. It seemed like a really good way to make a workshop available for free to everyone, get as many people to join in as possible, not have any difficulties with speakers having to travel at last minute or anything like that. Although some speakers are nice enough to have come and joined us here in San Francisco. I'll introduce some of the speakers in just a moment. Okay, so I see that our group is slacking a bunch of stuff. I'm glad to see it hasn't popped up on the live stream so far. All right, so first of all, as we're here, I'd like to, oh, now they're popping up, okay. I guess it's always just a little delayed. I'd like to thank our sponsors, first of all. They really helped out at the last minute to make this workshop possible. I'd like to thank 3Scan very much for offering us the space here in San Francisco. Oh, it looks like I have to mute something here. Why am I getting a feedback? Oh, I see. Somebody on the Google Hangouts probably has feedback.
That was me. Hold on just a second.
Yeah, if everyone who is on the Google Hangouts and who is not speaking could mute themselves, that would help a lot. Thank you. Okay. So anyways, Thanks to 3Scan for offering to use their space and helping out with the setup and everything else. I'd also like to thank the 2045 Social Strategic Initiative or Strategic Social Initiative, which helped us with some of the funding for the event, as well as Fathom Computing, that has likewise helped us out financially. And just they wanted me to quickly say what Fathom Computing is, so I will. Fathom Computing is building an optical computer for deep learning. Their processors use the wave nature of light to do computation, communication, and architecture suited for artificial neural networks with unprecedented speed and performance. Fathom's goal is to scale AI to human levels and beyond. They're hiring talent at fathomcomputing.com/careers. So this is Fathom Computing, and thanks for their contribution. And then also Scott Jackish of oaklandfuturist.com. Thank you very much to all of our sponsors. My name is Randall Kuna. I'm the founder and chairman of the nonprofit Carboncopies. So for our remote participants, I'd just like to mention that whenever you have a few minutes, say after the workshop, during the break, or whenever, please take a moment to participate in our survey. We'd like to understand more about your interests and how we can take those into consideration in our activities. The URL for that is really simple. It's carboncopies.org/survey. And I should mention that we have, say, what is it now? 3, 4, 5, 6, 7, about 10 people locally at the moment. I know that there are more people who will be showing up here, so we'll have about 20 locally. But online, judging by RSVPs, we have something between, on the order of 150 to 200 people who'll be watching, probably not the whole time, since people can just log in and see sessions they're interested in and disappear whenever they want to. But it's a really nice turnout. Okay. So yeah, let's see. I hope this all works out. Please bear with us whenever things are a little bit weird. We're going to have to be unusually careful for a workshop about giving the floor to people rather than interrupting, because of this this issue of getting what people are saying, their questions and their answers, to live stream as well. But I'll try to make sure that that all works out okay. In any case, let's not worry about that. You know, worst case scenario, we have a good series of talks. And best case scenario, we've got some productive discussion as well. Most importantly, let's try to have fun. Okay, I'd like to mention to those of you who are from the group,
Randall, we're not hearing you over here.
Randall, can you unmute?
okay. I managed to unmute myself just now. So it seems like someone else muted me, and we don't know who it was, but I just managed to unmute. So I'll have to be careful about that one. Okay. So let me just share my screen again. Grab this one, share. Back we go. Okay, we're back. So sorry, everyone. We're still ironing out the issues here, like accidentally muting one another, which is a feature of Hangouts that is kind of strange. But let me try to just proceed as normal at this point. I'm sure that once we're an hour into this, it'll all be figured out. Okay, so most people who are joining today have probably already heard of either mind uploading or whole brain emulation in one way or another. I'm just going to say a few words about why we at Carboncopies have made it our mission to push for its development and accomplishment, and why we think it's important to humanity or human society as a whole. I'm leaving all the really cool presentation making up to the other speakers, because given all the time that went into preparing the workshop, I didn't get to prepare a really great presentation of my own. But I'm going to try to be a little bit more specific about what the idea here is, and the goal and the challenges. And in, oh gosh, so it didn't manage to turn off my Slack after all, it seems. I don't understand why not. So many issues. Okay. So in a world where physics, biology, chemistry, currency, and even intelligence are becoming code, I guess one of the statements that I'm making is that our species can flourish only if we also come to grips with ourselves as information processes of a sort. So the purpose of this introductory talk is to introduce concepts and to provide the context for the remaining sessions and speakers in the workshop. Let me say up front that it's not going to be a fantastic presentation, but I hope that you all get the gist of what I'm trying to say. And see if everything works out nicely from here on. Oop, just jumped ahead a bit. Okay. If we're going to talk about mind uploading and whole brain emulation, it's important that we understand what we all mean by that we have some common terminology. So here I've tried to show this on a slide in some sense, to use an analogy. We begin with what I would call the experiential perspective, or human experience, to that which a person or a human being experiences. We could say that it's an abstraction level of sorts. What it does include, for example, are cognitive faculties such as sensation and perception, learning and memory, goals and desires, and awareness, and several others, of course. Without those, it's really hard to imagine a human mental life. But what the experiential perspective doesn't include, for example, are interactions of atoms and ions in the chemistry of cells, metabolic processes, molecular signaling processes. And I know that last one is potentially going to sound a bit controversial to the neuroscientists listening to this. But what I'm trying to say is, at this moment, I really have no idea which synapse in my brain was involved with what I was thinking, or which receptor channels were being activated, or which ones tried to activate but failed. All of these little details about the underlying biology are not part of my experiential cognitive existence. Of course, that doesn't mean that those things don't matter at all, and the cutoff is definitely far from absolute. What it does mean is that we need to recognize that depending on our goals, different abstraction levels may require particular attention. So for example, if you're trying to cure Alzheimer's disease, then it's probably really important to pay close attention to molecular processes. But if you're trying to build an intelligent machine, or a device that replaces the information processing carried out in a small part of the brain, then it's really the effect on the cognitive experience that matters most directly. And this is where I put up the analogy. Imagine, for example, that you want to run an internet browser on a computer. You don't want to have to care about the precise layout of every transistor involved in that process. Otherwise, for example, it would be impossible to design and upgrade to new CPUs. All you want to do is you want to make sure that every CPU is able to run the same program code and produce a satisfactory user experience. So there is an abstraction level that's important. In this way, we could say that the path to a successful mind upload begins with identifying your success criteria. For many of us, that means, at a minimum, retaining what our conscious day-to-day experience is presently like, and then aiming to upgrade to an ever better performance. Now, the term emulation or emulator has been used to describe this separation of scales, being able to abstract away the details below the level of interest, and still ending up with the desired result. In the example of the internet browser, as long as the program code runs properly, everything below that can be accomplished in a variety of ways. In fact, in that example, it might be sufficient just to ensure that the browser interprets HTML-coded websites correctly, allowing even the underlying software to be rewritten from version to version. Similarly, in a computer system, if a computer system can run an emulator to permit using programs that were designed to be run on a different or older computer system. Now, our cognitive processes are normally run by the human brain. Our default hypothesis is that everything about our mind that is relevant to the human experience is an emergent result of the processes taking place in the brain. And to date, that's a hypothesis that has passed every test with flying colors. We see that lesions in the brain directly correspond to changes in cognitive behavior, and we haven't yet found anything that that would indicate something lying beyond this brain processing for human cognition. So if we can run cognitive processes by emulation on another platform, then that ultimately results in what we're calling whole brain emulation. We sometimes then call that substrate independence because a brain emulator could be implemented in many different ways, no longer necessarily depending on biology. Now, in neural engineering today, the effort to build neural prosthesis is the earliest attempt to achieve something that is closely related to brain emulation. A neural prosthesis is meant to replace or support the cognitive function that's carried out by some set of neural circuits in specific regions of the brain. The prosthesis is built by creating computational models of the processing carried out by those neural circuits, and those models are then run on a computer or embedded in some device that interfaces and communicates with healthy neural tissue in a patient. The more accurate the model that was built using brain data, the better the implementation can be. The principal operators in the brain are called neurons, tiny simple processors that only experience an increase or decrease in their membrane potential, and at some threshold they respond with an electric discharge. And ultimately, it's the orchestration of billions of neurons that is the information processor that plays the symphony that's our experience of being. You can imagine that progressive neural prosthesis for every piece of the brain is ultimately identical to whole brain emulation. Now, it's pretty easy to see why individual neural prostheses are useful and desirable. At a minimum, they help with a brain dysfunction, for example. And beyond that, they hold the promise of augmented capability. It's also pretty easy to see why mapping and modeling brain functions is important to science, medicine, and the development of analogous functions in AI. But why does Carboncopies specifically promote attention to and advances towards whole brain emulation? The biological brain isn't optimized for access or monitoring, and it's not optimized for modification or augmentation. Once neural prosthesis reaches the point where you can emulate the whole brain, you reach a very special milestone. As long as part or all of cognition rely on the biological brain, there are hard limits to the sort of cognitive function that's possible. I'm trying to show that here in this simple graph where, say, the range of things that people can do and experience is a fixed amount of stuff. There's I don't know, is there a way to actually get a pointer on the screen here? I think there is. Okay. Oh yeah, here. So this, it's going to show up a little late on the live stream, but I'm pointing at that part where it says now. There's a limited range of what we can do and what we can experience. Then if you have neural prostheses, then you can do a bit more because you could tie in augmentation that is carried out on a device and tie it into the rest of the brain. But of course, as long as there is still a part of that cognition that relies on the biological brain, well, you're still going to have to deal with those limits. So for example, the limitations of neural responses mean that it's impossible to cognitively be aware of or respond to events at a microsecond scale, something that machines are capable of doing. So this is the human thriving argument for whole brain emulation and the understanding that comes with it. To expand the range of possible human experiences and capabilities, instead of submitting to the narrowly constrained way in which we can experience the universe today, and ceding the bigger picture to machines. There's also the important survival argument, though. Without being able to actually modify our mental abilities, we're constrained to what I might call an evolutionary niche of sorts. And the history of evolutionary change demonstrates that it's very likely that this niche may disappear, and that humans may soon play an increasingly minor role in the future society of intelligence. Adapting to such change may very well be a survival requirement. So the main reasons we have are both thriving and survival, this ability to move beyond the limits of what the biology gives us. Now, since Carboncopies was founded back in 2010, and even a couple of years before that, we've been keeping track of the biggest challenges and possible routes to solutions. It began with the 2008 roadmap that came out of the whole brain emulation workshop at Oxford. The purpose of this road mapping is to determine how to map the structure of the brain, how to record the response characteristics of its neurons and synapses, how to build a working model with parameters defined by those data maps, and then how to bring to life all of that in a real implementation of the whole brain emulation. So what are the biggest challenges today that need to be overcome for neural prosthesis and ultimately whole brain emulation to be possible? The greatest challenge is undoubtedly still the problem of acquiring sufficient data from an animal or human brain. To build a prosthesis, you need to be able to adequately approximate how a specific set of neural circuitry transforms incoming signals into outgoing signals. That process of approximation is called system identification. It's a systematic process of developing a computational model of the transformation from brain data. The more we know about the structure of the circuits in the neural tissue, the better we can constrain the possible models, reducing the search space and easing the computational burden for system identification. Today, developments in the area of connectomics seek to give us access to both the general and patient-specific neural circuit structure. 4 of our speakers are going to be addressing particular approaches to that problem, or to, especially the connectomics problem. Ken Hayworth, Sean Mikula, Tony Zador, and Adam Marblestone. Knowing structure alone, though, is not enough to build a working emulation. After all, the 3D connectome image is certainly not a working emulation. More inference is needed to convert a structural diagram into a working model in which the specific response functions of the components were correctly selected from a range of possibilities. We do not have these libraries of possible response functions and ways to select them and their parameter values. Today, functional recording with implanted electrodes is the most feasible way to collect data about the actual response of the system. And statistical regression or machine learning can then be used to build models and fit parameters, constrained by what we know about the circuit structure. It's this method of functional recording, followed by models building upon the known circuit structure of the CA3-CA1 transition in hippocampus, that the Berger Lab at USC and its associates and collaborators use in their neuroprosthetic efforts. Professor Theodor Berger will present state-of-the-art in this effort in the final talk of the roadmap session. Okay. We're almost through my presentation here now. In addition to identifying, tracking, mapping, and seeking solutions for scientific and technical challenges, Carboncopies also tries to draw attention to opportunity challenges. What I mean is, there are problems around creating projects, collaborations, or organizations in which researchers can take on the challenges. Most of these projects and collaborations still depend entirely on academic groups, but there is no limit to the variety of organized approaches that we consider, explore, and support. In the past few years, there was opportunity to explore how private startup enterprise could enter the mix. Incentives and obligations in the for-profit structure are different than those in academic settings, leading to a different set of advantages and disadvantages. It makes sense that there are types of projects on the roadmap to whole brain emulation that are well suited to development in a startup, and other types better suited to development in academia. A couple of high-profile efforts along these lines have had a little over a year to gain traction at this point. Kernel and Neuralink are the ones we mentioned in our schedule. Their founders declare that their companies exist to create bridges between human and machine, even to achieve neuroprosthesis to lift up humanity. What do these companies contribute that is difficult or impossible in academia? And what may still be beyond their reach? On a panel in topic B of the workshop, I will discuss with Dr. Diana Decca some of the nuances of this new development and generally how startup efforts fit into the big picture. Okay, so that's for that bit. And sorry, when I just paused for a brief moment, my computer was telling me that I would have to restart Windows soon for an update. I don't think that's something I want to do right now. It's lovely to see these things happen all the time. Okay, so this year, our team of Carboncopies researchers is launching a technological review project. This is because a living and useful roadmap needs ongoing renewal, and the tech review is going to be a major input to that. 5 members of the team will be introducing the project at this workshop by discussing their problem statements for the first subset of domains that the tech review will cover. Now, so what they're going to be talking about is what are the problems in each of these review topics that they'll focus on. How do they select the science and technology to study based on the potential to solve a particular problem? The project is going to invite peer review and will publish its results as part of the foundational literature on the science and technology of whole brain emulation. Abul Fazl Alipur, one of our researchers, will introduce the review for the question, what are the hardware requirements for useful performance of neuroprosthesis or brain emulation? Ilya Sapranidi will introduce the review for the question, what are the different pieces of neurotechnology that are involved in the process of neuroprosthesis and brain emulation? Although, as it turns out, Ilya is actually very ill, and so I'll be presenting on his behalf. So the question there is, for example, how can you best collect functional brain data at a high spatial and temporal resolution in a living animal or patient? Then we have a researcher of ours who only wants to be identified as John Doe, he prefers anonymity, who's going to introduce the review for the question, what types of brain mapping are needed for neuroprosthesis or brain emulation? So for instance, what's the right resolution for a useful connectome? And does that require electron microscopy or optical microscopy, possibly enhanced by expansion microscopy? Oh, I'm wondering why the live stream just popped away. Javon, I don't think it's focusing on me anymore, on the thing that, on my, my screen share. There we go. Yeah, there we go. Yep, back up to that. And then Bill Crowan, I don't exactly know how to pronounce your name. Is that right? Krogan? How do you say it?
Bill Krogan.
Krogan. Yeah. Bill Krogan is going to introduce the review for the question, what sort of computation brain models are relevant for neuroprosthesis and brain emulation? For example, what describes the dynamic characteristics of components in a connectome? And Alicia Smallwood, our research director, is going to introduce the review for the question, what are the fundamental theoretical requirements upon which the possibility of whole brain emulation actually rests? Okay, we're seriously almost done with my presentation, I promise. We want to use this workshop as an opportunity to highlight and empathize a challenge that's received surprisingly little attention so far. When applications of neurotechnology are discussed in general, when we talk about neural interfaces and prosthesis, and even brain emulation, access to brain data is such an urgent problem at the front of the queue that questions about how to interpret and use that data are often set aside for later. And yet, getting from a pile of data to a working prosthesis or brain emulation is non-trivial. Indeed, it's an entire research domain in its own right. So how do you approximate brain function from brain data? How do you do it so well that the result satisfies the criteria for a successful emulation? This is why we picked the special topic, Discovering Neural Circuits in Brain Data. And Diana Deka and Mikhail Lebedev will give talks in this particular part of the workshop, and each of their talks takes on an approach or a perspective on the problem. There are many more perspectives on that particular problem, and it'll take a lot more than a small part of a workshop to deal with this. We're just introducing it at this point. But this year, Carboncopies intends to shine a light on that particular challenge through a succession of efforts. Possibly the most ambitious of those efforts is to prepare a 24-month research project where we hope to use both functional recording and simulation to describe what we predict and what we cannot predict about data acquisition and data processing requirements. Yeah, this is a very complicated slide that I just added in for this, and I was hoping to get into details of that, but I'll do that at a different opportunity, I think, and just leave it for now. So I'm going to just switch to the last slide here. Let's wait for it to pop up on the live stream. Just to give a quick overview of something that I think is a, come on, there we go, is a reasonable, say, temporal ordering of what may happen in the years to come. Because, you know, whole brain emulation depends on a lot of things. Even neuroprosthesis depends on many, many things. And so these dependencies and the opportunities, they lead to a kind of temporal order that makes sense. And what I'm trying to show here is that we might, for instance, imagine that we're going to get some kind of application that is either for patients or even for normal users that involves non-invasive recording as a neural interface, and that can give you brain diagnostics or something else. And stimulation interfaces, of course, are going to be used in some form or another. Perhaps we'll start wearing sensors and various interfaces a lot of the time. Then, at some point, neuroprosthetic medical devices are going to start popping up in patients. And you could say, if you look at the lower end there, where it says 2036 to 2042, that patients who receive these implants, these devices, may end up being the first people with some kind of superpower. Let's say, for example, that you have a retinal implant, and the retinal implant is tuned to allow you to see X-rays. Well, this is a kind of superpower. Or let's say that you have an implant that replaces a piece of the hippocampus, which is what Ted is hopefully going to talk about. Then perhaps you can in some way categorize what is happening over time in that implant, and you can choose which memories you want to strengthen and which ones you don't want to strengthen, or which ones you want to help recall more easily. And that way you have a way of selecting among your new memories that you're forming in a way that we currently don't have. And that itself would be a kind of superpower, even if perhaps the quality of those first prostheses in patients won't be perfect. You know, even if perhaps they don't work quite as well as a biological hippocampus does, they may still have the ability to do certain things that we currently can't. Then eventually, hopefully, you'll see neural prostheses in many regions of the brain. And at some point, you could say that leads to whole brain emulation, either directly through this multiple neural prostheses approach or a parallel approach where we learn how to replicate brains by knowing more about how the structure can be, how the function can be inferred from the structure. So this is sort of an outline of a possible future. Okay, now I have to try the next part of this, which is I have to pop up the questions here to see if any questions have come in. Oh, this is the wrong one. Let's see, where is the real link? It's going to take me a second just to find. Here we go. All right, yeah, so first questions are all about the delay. Then we've got, let's see, how much time do we actually have for this? Okay, we have 1 minute, perfect. Yeah, we'll get going with the next speakers first. I mean, a lot of these general questions that are related to the talk I just gave, we can address those anywhere in the workshop, really. So if those of you who shared questions with us don't mind, we're going to move right on. And Javon's going to take that, putting us into the transition here. Sorry, what? Oh, okay. Right, there we go. All right, I hope people can still hear me, because I'm going to be introducing this first session as it starts. Our first session, topic A, is also called the 2018 Roadmap. This is about state-of-the-art and promising developments in the main challenge areas for brain emulation. So things like, how do we map the structure of a brain? How do we record the response characteristics of its neurons and synapses? How is a working model generated with parameters defined by those data maps? And how is that brought to life in a real implementation of the whole brain emulation, or in a similar, smaller subset, such as a neural prosthesis? Now I'm going to be introducing our distinguished speakers one at a time. Each speaker is then going to give a presentation. And if necessary, I'll give a brief signal to the speaker when they have about 5 minutes left. And then I'll moderate the Q&A discussion for that talk. The Q&A is going to be an interesting challenge, because we'll have live questions from speakers who have access to the Hangouts that this is being generated from. And we'll also have live questions from members of the audience here in San Francisco. But then again, we also have the questions that are coming in through the Q&A link on the site. And I'll be trying to make all of that work. So we have 5 speakers in this session in the following order. First, we'll have Ken Hayworth of the HHMI Genelia Research Campus. Then Tony Zador of Cold Spring Harbor Labs. followed by Adam Marblestone of MIT and Kernel, then Ted Berger of USC, and finally Sean Mikula of the National Institute for Physiological Sciences in Japan. Okay, so I see that Ted Berger is probably getting set up over there. So we'll take about a minute or two right now, just a pause to make sure that Ted Berger is all set up. And then, oh, actually, no, it's not Ted Berger's turn yet, so no worries there. Sorry, that was my mistake. Yeah, Ken's going next. That was my mistake. I was seeing Ted getting over there, so I was assuming for some reason that would change the schedule, but we didn't. Okay, so now I'm going to introduce Ken. Our very first speaker, Dr. Ken Hayworth. Ken is a senior scientist at the Howard Hughes Medical Institute Genelia Research Campus. He's also the president and co-founder of the Brain Preservation Foundation, and he's probably one of the scientists with the longest history of carrying out explicit research with the aim of making whole brain emulation possible, and a longtime friend of mine. Ken, please feel free to begin sharing your screen to present.
Thanks, Randall. Okay, I hope people can hear me and can see my screen. Randall, can you give me a little feedback and let me know that it's working?
I can certainly hear you. Your screen hasn't popped up on the live stream yet. Probably a slight delay. Actually, I'm not seeing your screen on the Hangouts either. Are you sure you're sharing your screen?
All right, let me try that again. It says that I am presenting to everyone.
Oh, okay. That's odd because I still see your face.
Okay.
Did you select to share your whole screen, or did you select a particular window?
I selected to share the whole screen. All right. Again.
Okay, something's happening. This is good. No, it didn't.
Okay, am I back in?
I now see, let's see, I still see your face.
Share.
Now I see something different. This looks like it's your screen. Yes. Okay.
You see my slides?
Yes, I see them, and I'm going to mute myself now so that you can speak.
Great, thank you very much. Okay, so this workshop is about human mind uploading, a great topic. I probably don't have to say this, but the goal of human mind uploading is overwhelmingly difficult. In fact, it is so overwhelmingly difficult that its achievement is at least many, many decades away, as Randall's last slide was showing. And therefore, it doesn't make too much sense to talk about it, except insofar as it can motivate today's research milestones. So with that in mind, I want to start off with a speculative timeline that I think can help organize some of these research milestones. So this is going to be quite a bit different from the timeline that Randall just put up, but we can discuss that later. So here we are in 2018. If I had to speculate, I would say that the first successful human upload would not occur before the middle of this century. And it will require tens of billions of dollars of research project on the order of an Apollo moonshot. And it will probably take till late this century until the cost comes down sufficiently for quote unquote normal people to have a chance at experiencing uploading firsthand. But there are many milestones along the way to this first human upload, and these are what we should mainly be concentrating on now. That is, making sure that they occur and trying to accelerate their progress. Proof of memory decoding from a small connectome is probably the the top item that that should be concentrated on, and people are working on this very seriously today. And I expect that a convincing proof of this will be demonstrated early in the next decade. Following that, imaging a whole mouse connectome. Again, people are working seriously on this, but it is really beyond the bounds of today's connectomics imaging techniques. So there's some serious developmental work that needs to go on to address this, and that's what I'll be talking about in my talk. The successful uploading of a mouse, and I'm putting that at about 20 years from now, because this is a lot of basic neuroscience knowledge will have to be gleaned in the intervening decades to make this possible. So this is really quite out there. And then imaging the first human connectome, I would put that out at about 30 years if I had to speculate, because it is just such a monumental task. Now, I see these as the major technical milestones leading to the first successful human upload. But there are a ton of neuroscience ones that I would love to go into detail, but I can't really in this talk. A question that should be asked of any tens of billions of dollar project should be, what good is this to humanity? And Randall gave some ideas of that, but I think one that I would like to put forward is that I think the first successful human upload will usher in an era of what I might call experimental philosophy. A revolution in our understanding of what consciousness is, what the self is, what states of mind are possible. This is what really gets me excited about mind uploading. When we have complete access to an uploaded volunteer's brain, the Neil Armstrong, if you will, of mind uploading, they will be able to explore the depths of consciousness in ways that we can't even imagine. These explorations of the uploaded mind will give us, all of humanity, insight to the next stage of human evolution. So I think crystallizing the impact of that first human upload, even if it is a tens of billions of dollar project, is probably a good milestone to motivate the earlier ones that we really should be focusing on today. Now, given the current pace of neuroscience and technology, I don't think this timeline is completely unreasonable. It may be overly optimistic by a factor of 2 or 3. So why are so many neuroscientists reluctant to embrace that mind uploading is clearly the end goal of a successful neuroscience? Why are so few pursuing these early milestones? Well, here's my speculative take on this question. Joe, neuroscientist here, we'll assume he's a middle-aged or elderly distinguished neuroscientist, understands enough neuroscience to understand how monumental the task is. Mind uploading is just incredibly monumental task. And he understands that he will be long dead before mind uploading is achieved. And we all have a tendency to say things that things are impossible if they are only going to happen after we are dead. But by its very nature, mind uploading has the potential to change this. If one could come up with a method of preserving the brain of terminally ill patients today for long-term storage, then anyone alive today could be expected to bridge that gap, a gap that might be 100 years long or more. Thankfully, the technology to bridge this gap is available today in the form of brain preservation by aldehyde-stabilized cryopreservation. The ASC technique was invented by Robert McIntyre and Greg Fay and published in this 2015 article. The ASC procedure is quite straightforward. The animal's vasculature is first perfused with the deadly chemical fixative glutaraldehyde for about 30 minutes. Glutaraldehyde almost instantly stops the metabolic activity and begins cross-linking all of the cellular proteins into a sturdy mesh. After 30 minutes, they start to slowly introduce cryoprotectant into the fixative perfusate and ramp up its concentration to about 65% over the course of the next 4 hours. All of this is done at room temperature, allowing for uniform cryoprotectant concentration in all cells. This uniform high concentration of cryoprotectant allows the brain to be stored solid at extremely low temperatures, minus 135 degrees Celsius, without ice crystal formation. Now, fixation by glutaraldehyde is known to preserve the patterns of synaptic connections among neurons and to preserve the ultrastructural details of synapses. In fact, it has been the gold standard for electron microscopy for decades. And it is important to understand that glutaraldehyde is also known to preserve the primary structure and relative locations of most proteins, as papers like this and this one describe. The key point is that glutaraldehyde fixation is simply the best method known to science to preserve both the structural and molecular information across an entire brain. As a result of this cross-linking, a glutaraldehyde-fixed brain is immune to biological decay processes and will remain stable for months. But eventually, diffusion would result in the slow dislocation of biomolecules, for example, membrane lipids, that were not cross-linked. But even this slow diffusion is halted in an ASC-preserved brain stored solid at minus 135 degrees Celsius. Time has essentially stopped for such a brain. Here is a video of a rabbit brain preserved by ASC being lowered into a minus 135 degree Celsius cold storage chamber. And here is that same brain removed from storage the next day. Notice that it is completely solid. When this brain was rewarmed, we sliced it up and prepared the tissue for 3D electron microscopy. The ultrastructural details of the brain were as well preserved as those in control brains that had undergone only the normal glutaraldehyde perfusion fixation. Here is a 3D electron microscopy video of a small piece of that ASC preserved rabbit brain. This dataset was acquired by me using focused ion beam scanning electron microscopy. There is no ice damage seen, no cracks. The ultrastructural details of synapses are clearly visible, and the connectome is clearly traceable. Now, the key points to note: ASC preserved the structural connectome across the entire rabbit brain, including ultrastructural details of synapses that should make the determination of their functional strength possible. ASC preservation is compatible with the later tissue processing steps needed to map the structural connectome. And this, to me, means that ASC should be compatible with future mind uploading. This same ASC technique has now been applied to whole pig brains. These are pictures of an ASC preserved pig brain that was rewarmed after being stored at minus 135 degrees Celsius. Again, there was no macroscopic damage seen, no ice crystal damage, and no cracking. And the ultrastructure across the entire brain looks well preserved. This technique could just as easily have been performed on a human patient, and the result would have been a preservation of the structural and molecular information contained in that person's brain and compatible with storage that can get that information to the distant future. So the implication of this that I want to stress is that ASC preservation of human patients will almost certainly support the possibility of future mind uploading. ASC is simply glutaraldehyde perfusion of the brain combined with indefinitely long-term cryostorage. ASC has been proven to preserve the structural connectome, ASC should preserve the locations and primary structures of key neuronal proteins, like ion channels. ASC storage is resilient to short-term rewarming because glutaraldehyde fixed brains are stable for months at room temperature. And when rewarmed, ASC preserved brains are in an optimal state to undergo the types of tissue preparation steps likely necessary for mind uploading. They can be reperfused with lipid fixatives, heavy metal stains, and potentially more targeted stains designed to highlight particular classes of proteins. And they can be dehydrated and plastic embedded, allowing for precision sectioning and volume electron microscopy. The implications, as I see it for today's discussion, is that mind uploading is very difficult, and may not be available till next century. Because of this lack of near-term benefits, it is difficult to attract the interest and funding necessary to reach even the near-term milestones. In contrast, brain preservation is easy and could be made available in every hospital today, giving hopes to millions of terminal patients. I believe that once people begin to realize this, it will create a virtuous cycle. More people interested in personally preserving their brains will lead to more funding of research to determine what would be required for mind uploading. This will in turn lead to better evidence that mind uploading will work, which will close the circle, creating more people interested in preserving their brains. So with that preamble, let me spend the rest of my talk focusing on these two milestones: imaging mouse and human connectomes. This is a figure from Sean McCullough's recent paper, which highlights just how daunting a task we are facing. The field of connectomics is currently struggling to image a single cubic millimeter of cortex as part of the well-funded IARPA Microns project. It's a $100 million project, I believe, funding 3 different groups, and they are all struggling to image the entire connectome of 1 cubic millimeter. To put this in perspective, a whole mouse brain is 450 cubic millimeters. And a human brain is over 1 million cubic millimeters in volume. So it's a daunting task. Let's take a look at today's main connectomics imaging technologies and see if any of them are up to that task. They aren't, by the way, but I will go through them anyway. Each of these technologies has proven its ability to map the connectomes of small pieces of brain tissue. Only 2 of these, SEM and Atom SEM, are currently in the running for the IARPA 1 cubic millimeter challenge because they are the only ones currently compatible with high current electron imaging. Serial section transmission electron microscopy allows fast imaging because the electron beam is spread across a tens of microns wide field of view, allowing an entire image to be taken simultaneously. This has the advantage of having orders of magnitude more electron current available for imaging than, for example, a single beam scanning electron microscope. But of course, the disadvantage is that the sections must be incredibly thin, and such sections are inherently fragile and difficult to collect, even when using automated tape collection, for example. Atom SEM collects sections on a solid tape. It allows fast imaging because it is compatible with the Zeiss Multisim microscope, which is a scanning electron microscope which has up to 91 electron beams imaging the sample simultaneously. And really, this multi-beam scanning electron microscope, which I think Sean McCullough will talk about much more later, is the thing that I think will break open the mapping of connectomes. The current electron microscopes, the current Zeiss Multisims, have 91 beams, so that's about 91 times faster than a single electron scanning electron microscope. But Zeiss is working on hundreds of beams in a single microscope. So I think this is a rapidly advancing imaging technique. So Atom-SEM, it's important to understand, is compatible with the Zeiss Multisim because it collects many sections, and these are spread across a wide wafer. And so you're able to image all of those very efficiently. Again, though, it has to collect those incredibly thin sections, and they're inherently fragile and difficult to collect. Now, the Z resolution of both of these techniques is determined by the section thickness. And this represents a fundamental Z resolution versus reliability trade-off. At the resolutions needed for tracing neural circuits, which is less than 40 nanometers, probably considerably less than 40 nanometers, this means that these techniques must collect hundreds of thousands, and in a human case, millions of fragile ultra-thin sections. For this reason, I don't believe either of these techniques can credibly be scaled up to whole mouse brain level, let alone human brain level. This brings us to the two block face imaging techniques. S-beam, S-B-E-M, uses a diamond knife to scrape an ultra-thin layer off the surface of a sample block prior to each scanning electron microscope image being taken. That's how it gets its 3D volumes. And FibSim essentially works the same way, except the diamond knife is replaced with a focused ion beam of gallium ions. Both of these techniques avoid the Z resolution versus reliability trade-off because they do not require handling fragile sections. In principle, either might be able to be scaled up all the way, but in practice, it is still difficult to see how either could be scaled up to whole mouse brain level, let alone whole human brain level. Personally, I think the focused ion beam approach has the most potential to be modified into something that could be scaled up all the way, which is why I will concentrate on it for the rest of the talk. Now, because it is based on ion milling, which means it has essentially no moving parts, FibSim, focused ion beam scanning electron microscopy, can easily achieve isotropic resolutions below 10 nanometers, as this volume through an optic lobe of the fruit fly shows. We have even, at Genelli Research Campus, we have even acquired FibSim datasets with 4 by 4 by 4 nanometer resolution. So no matter what resolution is eventually required for mined uploading, it seems that ion milling approaches will be able to meet it. A significant limitation, however, is that the width of the sample in the direction of the focused ion beam is generally limited to less than 50 microns. This is because the Fib beam is hitting the sample surface at a glancing angle, which is necessary for the smooth polishing to occur. We got around this limitation by cutting larger samples into thick slabs using a heated and lubricated diamond knife. This is a video of this hot knife cutting procedure. The hot knife sections are typically 20 microns thick, which means they are incredibly reliable to cut, and there are comparatively few sections to handle compared with ultra-thin sectioning. And this is an overview of the procedure. A large block of tissue is cut into 20 micron thick sections. These are individually mounted on tabs so that they can be focused ion beam imaged with their thinnest dimension oriented in the direction of the ion beam. And this is the what an image of one of these tabs looks like looking down on the surface of it. And you can see that the hot knife cut sections, edges, are extremely smooth. I've been cutting whole fly brains using this technique. And the individual sections are then imaged across multiple FibSim machines in parallel. And the individual imaged sections are then computationally flattened and stitched back together to create a full volume. This is a zoom-in movie on one such stitch between two separately imaged hot knife thick sections. You can see that there is a boundary right down the middle where there's a stitch line. you can see that that the neuronal processes are easily traced across this cut. And here is a side view of 7 such sections spanning the entire central complex of a fly brain, all stitched together. And here's just an example of tracings performed by Varen Jain's group at Google, automated tracing of this data set. Now, but the Zeiss multisim has a field of view of over 200 microns in diameter. So FibSim cannot even clear off a single multisim field of view. And FibSim is also many orders of magnitude slower than the multisim imaging. Thus, FibSim seems fundamentally incompatible with multisim imaging. So that limits us for the 4 existing techniques. So I believe that we need a new technique for connectomics imaging. What I believe what is needed is a top-down broad ion milling approach that can mill a few nanometers thick layer off of a hot knife cut thick section. So, for example, you would have you would cut the whole, in this case, human brain coronally at 20 micron thick slices. Those 20 micron thick slices would each be put onto a silicon wafer. A broad ion milling beam would be used to mill just 10 nanometers off of its surface. Then it would be transferred into a Zeiss multisim. Surface would be imaged in its entirety. And then it would be transferred back to perform more broad ion milling. And this cycle would be repeated up to 2,000 times across this 20 microns of depth in order to create a 3D image of this 20 micron thick hot knife section at 10 nanometer isotropic resolution. To image a whole human brain this way, it would first be hot knife sectioned at 20 microns thickness into about 5,000 slabs, each mounted on its own wafer. 5,000 is a large number, but not incredibly large. And these wafers would be shuttled between multisim machines that are and ion milling machines in industrial scale robotic imaging machines. It would require several thousand multisims operating for several years straight to image an entire human brain this way. It only require one operating for about a year to image an entire mouse brain. So the entire operation in this case for a human brain, a modern semiconductor fabrication plant complete with automatic shuttling of wafers. This is, by the way, where I get the $10 billion figure estimate for the first human upload. Now, I've been nurturing this idea of broad ion milling for several years, but I could not figure out a way to get the broad ion milling to work. But over the past year, I've finally built a small test rig that I think is finally getting some promising results, and I'm going to show those first here. This is the first proof of principle test of this broad area ion milling SEM technique. So on a single beam system, and it's early days on this, but I think this is a good proof of principle. This is a volume acquired at less than 10 nanometer isotropic resolution, spanning a series of 3 1 micron thick sections. So in conclusion, I think there is a path to whole mouse brain and whole human brain connectome mapping that involves hot knife thick sectioning with ion milling and with the Zeiss multisim. I would like to think that such a technology might actually be the one that enables the first human upload, but we are talking quite a few decades before that would occur. And I want to stress again here that every step along this path to the first human upload is a step closer to proving that persons preserved by aldehyde stabilized cryopreservation can be revived in the future? Thank you.
Thank you, Ken. Just 1 second until the live stream gets there. Okay. We've reached the end. Thanks. And I'm going to give you some applause right now.
Okay. Thank you.
It's hard for you to hear the applause from here, perhaps, but there we go. Now, this is going to be a little weird locally, because to hear Ken's answer, we have to have that on. Let me just turn it off for a moment.
This is going to be a little weird.
Okay, so because I'm going to moderate the questions, and online, all of that seems totally normal. Here, the live stream comes in 10 seconds later. So after I ask a question or present a question to Ken, then I'll turn on the volume again and we'll listen to his answer. I wanted to start right away with a question that came in from the online viewers. In this case, it's Michael Andrag, actually. He's asking the question, with the FibSem approach, how much damage to the sample is there from ion scattering?
Yeah, so in terms of damage, I'm not exactly sure what you mean by damage. It destroys the entire sample while it's being imaged. So there is no going back and imaging that.
I'm not exactly sure what you mean by damage. It destroys the entire sample.
I'll get you the next question in a sec.
So there is no going back and I'm not exactly sure what you mean by.
So, is there a question from this audience, by the way? Very good question. What's your name, by the way? Daniel. Daniel asks, would it be possible to collect molecular information from that which is being scattered during the process so that you could use that to further identify what you're scanning at that moment?
Yeah, that's a great question. So there is a technique called secondary ion mass spectroscopy that does exactly that. It collects the
Great question. So there is a technique called secondary ion mass spectroscopy that does exactly that. It collects the.
So I think the answer is in principle yes, but how I like to think of this is that the structural connectome is probably sufficient for mind uploading, and that could lead into a very, very long discussion. But in essence, the idea is that the structural connectome has so many is so high resolution, has so many correlated features that you should be able to infer molecular information. Now, let's assume that that's not the case, though. Let's assume that we go through and do the initial experiments and show that we really do need additional molecular information. That additional molecular information is probably going to be of very specific types. It's going to be of we need to know the density of ion channels on these particular neurons. That is almost certainly what it is going to come down to. Give me the density of ion channels on particular classes of neurons. And that's the additional information. If that is the case, then I think the more proper way of doing this would be to design some type of tags, molecular tags, using immunostaining or something to that effect, that would stain the original brain. And this is why I think aldehyde stabilized cryopreservation is probably a very good technique to allow this, because you preserve the brain with glutaraldehyde, but then you stop it at that stage before going through any of the other staining steps. And so in the future, 100 years from now, they can rewarm that brain and put through whatever staining they need to highlight those particular proteins as well as the membranes, etc. And so I think that is really the way that molecular information will be brought out. It will be highlighted with particular tags that are visible under the electron microscope, and during the scanning for the structural connectome, those additional tags will be able to be read. That gives these very specific ion channels that are necessary the ability to be to see what their densities are in particular places in the brain.
Necessary the ability to be to see what their densities are in particular places in the brain.
Thank you. I've got another question. This one is from the online audience. I've got to turn this off for a second here. Okay. This is a question by Odiseo, and he's asking, are you joining efforts with Max Moore from Alcor?
So I don't know what's happening with Alcor. I haven't talked to them in a while. Here is my take. I'm not joining efforts with anybody. Actually, I'm trying to get the best preservation possible in hospitals. Whoever does that is fantastic. What I do require for my stamp of approval, if you will, is that whatever technique is used needs to be demonstrated and published in the literature to preserve the connectome of the brain. And whatever technique is applied to human beings needs to have a whole list of quality control checks built in. And I don't see that happening at Alcor. Maybe they could make that happen, but I don't see that happening at Alcor, and I don't see that happening anywhere else. And so what I'm really looking for is I'm looking for the larger mainstream scientific and medical community to buy into this, to say, yes, this makes sense. We need to do it right, and we need to integrate it into mainstream medicine. That's what I'm really. That's the kind of introduction that I am looking for before this is ever applied to human beings. So aldehyde stabilized cryopreservation has not been applied to any human beings. I wouldn't recommend it being applied to any human beings before those basic medical procedures have been published and the medical community has had a chance to review them for the quality checks that are necessary.
Procedures have been published and the medical community has had a chance to review them for the quality checks that are necessary.
Thank you, Ken. We have time for one more question for you. And before I check for another one from the online community, I'd like to, Ted Berger has a question.
So I'm not sure if I missed something, but what I saw was evidence for structure, but not connectivity. That's very different.
Okay, Ted Berger says he wasn't sure if he missed something in your presentation, but what he saw was evidence for structure, but not connectivity.
So I've looked at these 3D electron microscopy volumes, and I've shown them to many of my colleagues that trace neural circuits in normally prepared tissue. And all of them have said that the connectivity is being preserved in this aldehyde stabilized cryopreservation, these samples.
Thank you, Ken. I think that Ted Berger probably still had follow-up questions, but because we have to move on to the next speaker, we can take that to the lunch break or whatever.
Okay.
I thank you very much for your contribution once again. Thanks also for stopping the screen share at this time. so that we can switch to our next speaker, whom I will introduce right now. Our second speaker today is Professor Tony Zador. Tony is the Alla Davis Harris Professor of Neuroscience at Cold Spring Harbor Laboratory, unless that's changed recently. He's also a co-founder of the prominent Cosine Conference. In the context of today's workshop, Tony's extremely interesting contribution is an altogether different approach to connectomics using molecular biology to map the brain. Tony, please feel free to begin sharing your screen to present.
Okay, let's see how this works.
Biology to map the brain. Tony, please feel free to begin sharing your screen to present.
Okay, let's see how this works. Am I sharing? Is it working? Can I get some feedback?
Looks good.
Oh, good. All right, well, let's start. All right,
yes,
so
it seems to work. Good.
So thanks for the introduction. Thanks for that first talk. It was fascinating. So I'm going to be talking indeed about a different approach to uploading the mind eventually to connectomics based on molecular approaches, and it's summarized here in the first slide. Basically, our idea is that we can use molecular biology to uniquely label all the cells in the brain, then grind up the brain in one way or another, and use high-throughput DNA sequencing to read out the projections and connections of the brain. I should say as a caveat right now that we haven't achieved that goal yet. It has the potential plus side of much higher throughput, but there's a lot of technology development that needs to happen before that works. We're getting very close to making it work in the mouse, and there are some pretty more fundamental, but perhaps not insurmountable challenges to getting it to work in humans. All right. So I added one slide sort of in the context of this talk, and just wanted to give some thoughts about the problem of uploading a brain. I think the bad news is that it's going to be very, very hard to copy every neuron, every synapse, potentially every protein. And even if we did that, even if we were able to measure all of this, it would be very challenging to know what to do with that. Would we make a huge simulation? If we made a huge simulation, we would probably have to have very strong opinions about which of those components was important in order to sort of make up for the properties we didn't measure. I think the good news is that maybe we don't need to do it exactly that way. So I'll point out that the brain that each of us walks around with today is not the same as the one we were born with. It's not the one we had 25 or even 5 or even 1 year ago. And yet we have this illusion of identity continuity. We feel like we're at some level a continuous extension of the person we were when we were born and 5 years ago and certainly yesterday. And so I think that suggests that there's a path to uploading a brain in which we introduce some kind of a system that interacts with and mirrors our brains during life. And such a system would have the potential to sort of represent the substrate for a permanent uploading of our minds into potentially a very different kind of a system. But I'll say that I think we have little hope of uploading a brain if we don't understand many of the fundamental principles of brain function that we absolutely do not yet understand. I would say that we know a great deal about individual neurons and synapses, and we have very little understanding of how they work together to produce thoughts and feelings and identity and memories and consciousness. All right. So what we need to do is we need to understand simple circuits, and I like to sort of put a historical perspective on this and just remind people that the first person, I think, to really study circuits in the way that many of us are trying to do these days and tease apart the circuit was Descartes, who proposed a circuit by which a person withdraws his foot from a fire, a reflex, if you like. And the mechanism that he proposed was that the flames of the fire displace the skin, which pulls on a tiny thread. The thread opens a pore in the ventricle, and then the animal spirits flow from the ventricle, which is labeled F on the diagram there, through a hollow tube, and the animal spirits inflate the muscle of the leg, causing withdrawal. Now, many of you may recognize that our modern interpretation of what goes on differs in some ways. We no longer believe in the precise location of the source of the animal spirits F. But nevertheless, I think Descartes did make an important move forward here by actually writing down a potential circuit that we could, in principle, copy and use to build a brain. And I think that's the goal of many of us working on this. So why is circuitry important? Again, I don't need to belabor that point for this group. I'll just say that we want to be able to reverse engineer the brain. And to do that, I think we need to not only make a lot of measurements, because I do think those measurements are important, and many of the and develop new technologies to make those measurements, but we have to really think hard about how the circuits that we get out by making those measurements give rise to the kinds of things that would ultimately be needed to build a brain. And then the other really immediate need for understanding circuits is that most neuropsychiatric disorders arise from disruptions of neural circuitry. Autism, schizophrenia, depression, addiction, bipolar disorder, they're all probably, or many of them are probably in part, in large part indeed, caused by disruptions of either structural or functional circuitry. And the incredibly limited set of tools that until very recently we've been forced to work with have really limited progress, to the point that, as many of you may know, major pharmaceutical companies have largely given up the search in their own labs for discovering new drugs. And it's because what big pharma is good at doing is making a drug, not finding targets, and we don't even understand what those targets are. And that's really emphasized by the fact that really in the last decade or two, really the last decade, the major frontier in treatments of neuropsychiatric disorders have been in deep brain stimulation, circuit-based treatments that exploit the little that we do know about the organization of circuits. And so if we could know more about those circuits in mammals, in either humans or in models of brain function, that would be incredibly useful. All right, so why is circuitry hard? And that's shown here. The problem is simply that the axons are all tangled together. At least that's not simply, but that's really one of the big challenges. You see a bunch of neurons here, a bunch of wires. I'm not sure if you can see my cursor, but all those wires are somehow related to neurons depicted or shown here, and also neurons not on the screen. And we really need to be able to track them. Of course, with serial electron microscopy, that's increasingly possible, and yet that remains very slow. And the challenges are not just in the data collection, but in the data analysis. And although that technology is improving rapidly, I'm going to propose that there's a shortcut that complements the EM approach. Okay, so our core idea is to recast connectomics as a problem of high-throughput DNA sequencing by labeling each neuron with a unique sequence, a DNA barcode. And the idea is that way we don't have to trace neurons, merely read the identity of a neuron at its soma and at any place we wish to look, and we don't need to look at all the intervening sections. And that greatly simplifies the problem, and it allows us to exploit the tremendous improvements in DNA sequencing technology and more generally molecular biology. So why sequencing? Because sequencing is fast and cheap, and many of you may be familiar with this, with the fact that maybe aware of the fact that sequencing has gotten cheap, and yet it's still striking when you look at this graph to see just how quickly DNA sequencing technology has improved. So this is on the x-axis is time since 2001, which is when the human genome was sequenced, and on the y-axis is cost. And for this is cost on a log scale. So Moore's law on this is a straight line. Moore's law is a pretty healthy clip of improvement, and yet you see that the cost per genome has blown away Moore's law since about 2008. And so basically, any approach that you can sort of hitch on to the incredible improvements of DNA sequencing seems to me to be a win. All right, so right now what I'm going to focus on is the form of this technology that actually works really well right now in my lab today. We've set up a core facility. We offer this as a service now to collaborators, and that's projectome mapping. That's the mapping of long-range projections at high throughput at single neuron resolution. And to put this into perspective and motivate it, I'm showing here a slide from the Allen Brain Atlas. So what the Allen Institute did was a huge service to the entire mouse neuroscience community, which is they systematically went through and made injections throughout the mouse brain of a virus encoding GFP. So if they make the injection there on the right, they wait about 3 weeks or 4 weeks for the neurons that take up the virus to express it all through their axonal terminals. And then you can look throughout the brain, and anywhere you see green, you know that the neurons that you've injected send a projection. And they did this systematically, one injection per mouse, localized as far as they could to a single brain area. And then they, for each of those 1,000+ mice that they injected, they sliced the brain, took pictures, and put the pictures up online. And it's been an incredibly useful resource to all of us who work on this system. But that said, it has limitations. The challenge is that bulk injections don't distinguish single neuron projection patterns, and that's illustrated here. So on the left, you see an injection site. Those 3 circles are 3 neurons, and it could be that if you see green in the 3 target areas shown here, it would reflect the fact that neuron 1 projects exclusively to area 1, neuron 2 to area 2, and neuron 3 to area 3. Could also be, instead of a one-to-one mapping, an all-to-all projection mapping, or the brain being the brain. Most likely it's complicated, and that complexity actually is important to understanding what's going on. And that's the complexity that is missing when we use these kinds of bulk injections. And so the alternative to these bulk injections is to do single neuron fills, but that's incredibly low throughput. So here's a wonderful paper studying the projections from the cortex to the striatum in rats. For the purposes of this, what's important is that it's 1 neuron per rat. And this very nice paper showed the reconstructions of 25 neurons from 28 rats. This is a group that, at least at the time, it's a very skilled group. Since then, throughput has increased a little bit, but it's still not going to allow us to map all the neurons in a single brain at any throughput. So the inspiration for our approach actually came from a technique developed by Jeff Lichtman, Josh Sains, and published in 2007 called Brainbow. And there the idea is that to make the tracing easier, it would be convenient if the axons had different colors, if you could color code them. And there's this brilliant idea of coming up with a very clever genetic trick for endowing each neuron with a randomly selected collection of 3 colors drawn from a palette of about a dozen. And if you work out the combinatorics given by the genetics they did, that led to a theoretical diversity of about 200 colors in principle. And that would actually be pretty nice. But unfortunately, color space is limited and hard to read. So although the theoretical diversity was 200, the number of resolvable colors that you could actually resolve with real microscopes, actually it's not even an issue of the microscopes, it's the overlap of fluorophores in general really is closer to, it's less than 10 generously, it's probably closer to 5. And that's shown here, where those 3 purple neurons are probably different purples, but they can't be resolved. So basically the core idea here is to replace colors with DNA sequences. And the reason is simply that there are a lot of possible DNA sequences. So we can kind of think of MapSeq, which is the name of the technique we developed, as a kind of digital rainbow where you replace colors with numbers and have each neuron express a unique sequence of DNA, a genetic barcode with a theoretical diversity that is essentially unbounded. And just to drive that home, there are 4 nucleotides, A, T, G, C. If you have a barcode, a random barcode of length 1, you have a potential diversity of 4. Of length 2, it's 16. And by the time you get up to 30, you get a potential diversity of 4 to the n. I'm sorry, of 10 to the 18, which is vastly more than the number of neurons in a mouse brain, a human brain, or for that matter, even in the human body. So a random 30mer is sufficient to uniquely label every cell in your body with very, very low chance of double labeling. More concretely, 10 to the 9 neurons are sufficient to label all 10 to the 7 10 to the 9 barcodes are sufficient to label all 10 to the 7 neurons in a mouse cortex with pretty high, with sufficient diversity so that the chances that 2 neurons will be labeled the same by chance are pretty small. And in practice, how do we do that? What we do is we've generated a virus. Right now we're working with a particular virus called Synbis. And viruses are generated in the lab from a plasmid. And so to generate a viral library, all we do is we take the plasmid that generates the virus, and we introduce these random sequences directly into the plasma, into the plasmid backbone. So we get a plasmid library, and then those plasmids give rise to a viral library of sufficient diversity. There's this remaining challenge of getting the barcodes to the synapse, because typically most RNAs don't travel to distant axons. And here in one slide, I'm summarizing the PhD thesis of a very talented graduate student in my lab, Ian Pycon, who figured out a modification of a synaptic protein that is shown on the far right that allows that protein, which normally traffics to axon terminals, to grab onto the RNA barcode through a so-called N-lambda domain. And it grabs onto the box B component of the RNA barcode that we put in and drags it out to the synapse. So our first proof of principle of this was to the locus coeruleus. This was published, I guess, a year and a half ago now. And what we did was we studied the projections of the locus coeruleus throughout the brain. It was known already that the locus coeruleus projects throughout the cortex relatively uniformly, but it wasn't known whether it was suspected and believed, but not really known, that the neurons in the locus coeruleus just kind of bathe the cortex in noradrenaline. So the locus coeruleus is the sole source of noradrenaline to the cortex. Noradrenaline is a kind of wake-up signal. It tells the brain to go on alert. And the usual view is that there's a cortex-wide alert signal that goes off when the locus coeruleus is activated. But alternatively, one can imagine that the locus coeruleus actually can specifically activate subparts of the cortex. So for example, if you need to specifically listen for sound, you might imagine that the auditory cortex is selectively turned on, and maybe if you need to selectively be on the alert for light, it might be the visual cortex. So I'm going to skip over some of these details and just say that for the most part in these experiments, it turns out that the RNA acts like GFP. It acts like a bulk signal that bathes the brain. And the way the experiment goes is we inject in the locus coeruleus, that cross in the back there, and then cut the brain into slices. And then we extract the RNA from each of the slices and figure out which barcodes appear in each of those slices. And then we can demultiplex that, and we can get a picture that looks like this, for example. So here is the profile, the spatial profile of projections for barcode 28, which corresponds to neuron 28. And the projection strength is measured in barcode molecules, which we quantify carefully through sequencing. And the x-axis shown here is the rostrocaudal position from those slices. So it's important to recognize that with this technique, the spatial resolution is determined by the at the time of dissection. So we collapse all the barcodes that are found in whatever chunk of brain that we collect and put them and can't resolve them further. So this particular neuron you see has a very specific spatial pattern of projections. Another neuron, and that's at odds with the traditional model, another neuron has a much broader one, barcode 79, and there are even more complicated ones like bimodal projections. And in all, we were able to get 1,000 single neurons and their projections from just a handful of animals in just a couple weeks. So this is orders of magnitude more throughput for this system than has ever been. Well, since there were I think there were no locus coeruleus single neuron reconstructions, so more than orders of magnitude is infinitely more. But in general, 1,000 neurons in a handful of experiments is high throughput. We've since then, just a technical note, many of you are probably worrying about double infections. What if you get two barcodes into the same neuron? And in the interest of time, I'm not going to go over why that's not bad, but it isn't bad, and you can ask me during the questions. What would be bad would be degenerate barcodes, insufficient barcode diversity that would end up with the same barcode ending up in 3 different neurons, and that would be disastrous. So we go out of our way to avoid that, and then we confirm that we avoided it. So if people have these technical questions, I invite you to ask me during the question period. I'll also say that more recently, we now have, in collaboration with Tom Mersick-Flogel's lab, we did who did a bunch of single neuron reconstructions in visual cortex. We compared their reconstructions, 35 neurons in a couple of years, to some MapSeq studies of those same projections, 533 neurons in a handful of experiments. And for the purposes of this talk, what's important is that they agree to essentially the resolution that we can determine in this experiment. And that there's a whole scientific story that I don't have time to go into, but that should be coming out in Nature sometime in the next few weeks, and it's on bioRxives right now. All right, so we have a core facility. This is up and running. We're now studying the projections in different animal models, different brain areas. And we're scaling up to do the whole brain in a single experiment, and I'm just going to go through that very quickly. We tile the entire brain with, or the entire cortex with barcode. Then we take much finer slices than in the previous work, align it to the Allen Brain Reference Atlas, and then we can project these back to Allen reference coordinates. And in a single experiment, in about a week for about $10,000, we can get the projection patterns of about 100,000 individual neurons. And for each neuron, we can see the areas to which it projects. So here are the strongest projections of 2 of these 100,000 neurons, just randomly selected neurons, to give you a feel for what kind of data that we have. So this is so much data, and of a different form, that although we've had these data now for almost a year and a half, we're still figuring out how to make sense of them and what to say about them. But we're writing this up right now. Nevertheless, none of this gets us to the connectome. So far, I've only told you about the projectome. And so I want to, in the last couple minutes, talk about that. So the limitations of MapSeq are that first, it has crude spatial resolution, as I mentioned. It's limited by dissection. There's no notion of morphology or cell type. And I should say that I'm not getting any feedback on the screen right now. So I think I'm 25 minutes in, but if the MC wants to communicate with me, I'm looking at my cell phone. You have my cell phone number, and you can let me know how I'm doing on time or anything else. And you want to communicate with me? I'm looking at my cell phone. You have my cell phone
Tony?
number. Yeah.
Yeah, you're fine. You're fine, actually.
Okay. If you want a side channel, I'm staring at my phone, so you can text me or whatever you want. Okay. In fact, that's reassuring to hear that you hear me, because for all I know, I've spent the last 25 minutes talking to my screen. I got to say, this is a strange forum where I don't see the faces of the people in the audience. You know. Anyway, so the second issue is that there's no notion of morphology or cell type gene expression. And my own background is in computation and in physiology. So the fact that anatomy is traditionally very hard to correlate or relate to neural activity, to knowing what the neurons did when the neurons were still inside the animal's head and the animal was doing interesting things, is distressing. And of course, as I mentioned, I haven't told you how we can get from the projectome to the actual synaptic connections that were needed to figure out the connectome. And the solution to all of those is a technique that we call BAR-seq, that's modified from in situ sequencing, which is a technique that was developed in George Church's lab, and which we learned from them through collaboration, and then modified and adapted to the particular goals here. Okay, so to tell you how that technology works, I have to actually take you through just a little bit of the nuts and bolts of conventional DNA sequencing. Many people who do sequencing only have a vague idea of how the sequencing actually works. This is, when I say conventional sequencing, I mean conventional short read, high throughput sequencing as done by Illumina. And so this is how it works. On the left, you have a tube full of DNA, little bits of DNA that you want sequenced. You wash it onto a so-called flow cell. Those little black bars sticking up are sequences that grab onto the purple DNA that you want sequenced. And then the next step, you amplify each one of those single pieces of DNA into a little cluster on the slide of identical bits of DNA. So that's done by bridge amplification. And then the key thing is that then the sequencing actually becomes an imaging problem. So you take a snapshot of those clusters, but before taking the snapshot, you label each cluster with the next nucleotide in its sequence. So again, I'm not sure if you can see my pointer, but for example, this one in the left, the first nucleotide in that sequence might be a C, and so it's labeled with a fluorophore that's white. This one on the top is an A, so it might be pink. Same with this one, and this one here on the right is a T. Then you take a picture of this using originally they used a spinning disk confocal. They've actually changed that technology, but originally it was with essentially a confocal microscope. Then cleave those fluorophores off and add the next nucleotide. And so to build up the sequence of DNA associated with each one of those single molecules really is a matter of taking a bunch of pictures and aligning those pictures properly. And so the number of dots per picture represents the number of molecules that you're sequencing, and the number of cycles tells you the length of the DNA that you're able to get. So in this case, C, T, T, those are the first 3 bases, and then some dots. And typically these technologies can nowadays go up to several hundred nucleotides. So the key point here is that conventional high throughput next generation sequencing involves taking a picture. So for in situ sequencing, we can do essentially the same thing, except that instead of first extracting the DNA, we first immobilize the RNA in the cell, and then using a different set of molecular biology tricks, we convert each one of those individual molecules of RNA into a ball of cDNA, complementary DNA, that corresponds to the sequence we're interested in, the RNA. And from there on in, it's essentially the same problem of imaging. So here's an example that I hope will clarify what I'm talking about. This is a picture of a bunch of neurons in culture. The bright circles are cell bodies. I'm showing you these pictures from cell culture because the pictures are prettier, but we also do this routinely now in slices. And so what you see is, here's the key for reading it out. Each neuron is full of one barcode derived, or in a couple cases, 2 barcodes, but typically 1 barcode derived from infection by our virus. And just to show you how this plays out, here are 2 neurons that I'll show you the sequence of. The first base is an A, then we cleave that off, the next base is a G, and so on. And so by taking a series of pictures, we can read out the sequence of barcode in each neuron, and with different optics and modifying and optimizing a couple of things, we can also do it in axons and dendrites. So we're now applying this to study the projections in auditory cortex, and in the interest of time, I'm going to skip over most of that and just say that here's a picture of about 1,500 neurons whose sequences we've determined in a brain slice. Well, we took these neurons, we took a brain, we injected the virus in auditory cortex, then we waited 2 days, then we did in situ sequencing, bar-seq, at the injection site and looked at the long-range projections using conventional map-seq. And in that way, we were able to get the projections of cell body positions of about 1,500 neurons. And so for each one of those little dots, we can click on it, and I could tell you the strength of projections to the dozen or so areas that we've studied. And again, there's a whole story that we're working out here. We can combine this with in vivo activity. We first use 2-photon imaging to figure out the activity of a bunch of cells. And then ex vivo, we can find those same cells and then figure out their projections. We can combine this with gene expression. Let's skip over that. And then here's what I think which is what's most relevant to the focus of this meeting, which is how do we achieve synaptic resolution? And essentially, I've already told you the answer, which is that we combine the axonal and dendritic labeling with barcodes in this tangled mess that I showed you. And we can figure out what the 2 barcodes are, what the 2 barcodes are of the potential processes that are, or the synapse, of the processes that are forming a potential synapse. We have to actually go one step further and figure out whether there is an actual synapse there. Currently, we're doing that by using a synaptic marker. Going forward, we have some molecular tricks for actually entangling the barcodes associated with the 2 processes so that a single ROLINE will form that includes the sequences of the 2 barcodes, but only when the 2 barcodes are touching at a synapse. So in summary, map-seq and bar-seq open up the possibility of combining connectivity, gene expression, in vivo function at high throughput in a low-cost way. And I'll just summarize by saying that if the brain were so simple we could understand it, we would be so simple we couldn't. And I can't end without first thanking my collaboratome, especially Eustace, Xiaoyan, Lee, and former lab members, including Ian Pycon, and also my fundosome, especially IARPA, NIH, and the Paul Allen Family Foundation for some very early funding for this. And with that, I will close. Paul Allen Family Foundation for some very early funding for this. And with that, I will close.
Thank you, Tony. Let's give you an applause. I have to say that was really, really fascinating, and that looks like it's something that is going to help us go leaps and bounds forward in terms of understanding the connectome, in terms at least of the projections of circuits, maybe even more depending on how refined this technique gets. I'm pretty sure that there are quite a few questions, and I'd like to start by asking the people here in the audience for a question, if there are any.
Can I just ask?
Just 1 second.
I think I'm in this weird mode right now of infinite regression.
Maybe you could just stop sharing your screen. That might be the solution for that.
So I should go to just chat. No, if I just toggle. Oh, okay, there we go. Thank you.
Okay, good. Yeah, sorry, Daniel, you had a question. Oh wow, I'm not sure if I even understood the question correctly. Could you say that? Maybe explain a little bit more. What
I can't hear it. Or
so, yeah, I know. I'm going to have to repeat the question for you. This is how it's going to work from the audience here. Yeah. Mm-hmm. Ah, okay. Right. Okay. So Daniel is asking, now that you're doing transfection, now that you're putting things in the cell, what about putting in proteins to do something like a kind of computation, to record something else, to identify other things that are going on? And this
you mean like
this sounds like
DNA ticker tape?
Like the DNA ticker tape, sure. Yeah, basically.
I mean,
okay, let me just turn you on. There we go.
I mean, we're you know, the simplest version of that in principle is to combine it with calcium imaging. Right now, in the current form, there's a technical limitation in that the virus we use to do the MAP-seq is actually fairly toxic to the cells, and so this is a terminal experiment. We're in the process of switching over to a different virus, AAV, that should be compatible with calcium imaging, chronic calcium imaging. We have basically right now the way we do it is we do calcium imaging. Then when we're ready to end the experiment in the mouse, we inject with the toxic Synbis MAP-seq virus, get the wait 2 days, and then sacrifice the animal. A somewhat more convenient way to do it would be to combine the AAV calcium imaging with the barcode. And that's certainly on the to-do list, and we're working on it. But in general, anything that you can, I mean, it really, the compatibility right now is limited only by our choice of virus. In general, anything that you can, I mean, it really, the compatibility right now is limited only by our choice of virus.
Hi, Tony. Yeah, thanks for that answer. Just to clarify, if you hear yourself afterwards, it's because the live stream is a few seconds late.
Sure.
And so then I'm opening up the microphone for you to hear that, just so that you know that, you know, we're still listening to your answer.
Got it.
Yes, we have another question from the audience here. 1 second.
George Church is working on some like molecular recorders using CRISPR. Do you know much about that technology and whether it can be integrated?
Okay.
So the idea is to look at temporal synapse firing.
Yeah. Okay, the question is, George Church is working on some molecular recorders using CRISPR, et cetera. So this is very closely related to the previous question. And what do you know about that and about the what was it? How it would complement your method?
So as far as I know, George's lab is working on, is or has worked on 3 different things. So the very first thing that they were working on, which Adam Marblestone, as far as I know, George's lab is working on, can you turn off my mic because I'm getting 3 different things. So the very first thing they were working on, which Adam Marblestone, confusing to me. Anyway, so the first thing that they're working on, that Adam Marblestone worked on, was the DNA ticker tape. And I'll let Adam discuss what the current and future status of that is. The second is that George and I are part of an IARPA consortium to apply this MapSeq and BarSeq technology to achieve single synapse resolution in the same IARPA project that Ken was talking about earlier for figuring out the connectivity of the visual cortex. So on that, we're full collaborators. My lab is applying this technology in all sorts of other ways, and I think, as far as I know, George's is in the context of neurons only focused on the ARPA, although a lot of things go on in George's lab. And then there's the third component, which is that I believe that they, as well as several other labs, have come up with CRISPR approaches to barcoding neurons in, well, barcoding cells in the germline. They published one version of that. There are several other groups that have published different versions of that. Doing it in the germline has advantages and disadvantages compared with doing it with viruses. The advantage, of course, is that you can be confident that every cell has one and only one barcode. The disadvantage is that you limit yourself to working with a transgenic line. And so because we're particularly interested in applying this to other model systems, mice, all sorts of rodents, marmosets, and potentially eventually humans, although that's a different discussion how that would work. We're actually pretty excited about the viral approaches, which turn out to be also more convenient. We're actually pretty excited about the viral approaches, which turn out to be also more convenient.
Thank you. We still have time for more questions, so now I'm going to grab a question that came in through the question and answer form online. Rose, or Rose, is asking, can DNA barcoding keep track of all the multiple connections of a neuron's dendrites and their strengths?
So we hope that, so there are 2 questions, I guess, embedded in there. First, can it track all of the connections? And using this spatial readout that I was describing based on BarSeq, the answer to all is yes, subject to technical limitations. The second question is, can we read out synaptic strength? That remains an open question in the same way that it remains an open question through all non-physiological approaches. I'm a physiologist by training, so for me, synaptic strength is defined by a slice experiment in which you record the synaptic connections between a pair of neurons, and you stimulate one neuron and record in the other neuron. There's a lot of data from EM, actually some of it, I think some of the first work from my own lab, the lab that I was a postdoc in, Chuck Stevens' lab, showing that there's a pretty good correlation between synaptic strength determined in that way and EM, the size and other structural components of a synapse determined by EM. And there's been, I think, a lot of follow-up experiments showing that there's a good correlation there. I think going forward, we can exploit some of that and also exploit the fact that there are molecular determinants of synaptic strength that we can access using in situ sequencing. So, for example, right now in a separate project that I didn't describe here, we are actually using a related set of approaches to figure out which synapses have recently undergone plasticity, where we use as a marker of plasticity the recent insertion of a GLUR1, a type 1 glutamate receptor. So we're doing this in collaboration with Robert Malinow, whose lab worked out the basic mechanisms by which synapses, by which LTP works, long-term potentiation. Turns out that the insertion of a GLUR1 receptor is a very good marker for synaptic plasticity. So right now we're using essentially the same set of, or we're trying to use the same set of approaches to figure out, if you like, the LTPome, the set of, the subset of connections that have recently undergone plasticity in the context of an animal learning something. So I think there are a couple routes to figuring out synaptic strength, either sort of directly structural or combining structural with molecular. So I think there are a couple routes to figuring out synaptic strength, either sort of directly structural or combining structural with molecular.
Thank you. I have another question from the online forum. This is by Keith Wiley. And he asks, can DNA barcoding be combined with a sort of slice and imaging that Ken described in a single subject, a single brain, or must we choose one or the other toward the connectome reconstruction goal?
So the question is, can we combine this technology with some kind of serial EM type approach? We've talked to various groups about that. Certainly, it would be awesome if we could figure out a way to do it. I don't think it's impossible. So I'm not an expert in those kinds of approaches. So I don't think it's impossible. It would clearly be just spectacular if we could do that. But so far, we didn't find a straightforward enough approach that would sort of, where we could crank out a proof of principle with, you know, half a postdoc's time in 6 months, which is sort of at this point the level that we'd need to. I think that from what I understand, there are some routes that we could pursue, but we'd have to really double down and commit to pushing it. And so far, we haven't found the resources or the collaborator to do that. So I hope that the long-term answer is yes, and the short-term answer is not yet. So I hope that the long-term answer is yes, and the short-term answer is not yet.
Thank you. I think we have time for maybe one more question. So let me see, first of all, if there is one in the audience here. Okay, Daniel has one more question.
I was going to ask, is there any implications of this Neuralink versus infecting humans starter?
Okay. Oh, this is a tough one. Okay, so Daniel asks, are there any implications to development of this technique that are relevant to, say, startups like Kernel and Neuralink that are hoping to bring things to humans fairly quickly, or is this just a non-starter for use in humans?
So that's an interesting question. So, you know, obviously the first answer is that anything we learn about the fundamental principles of brain wiring and connecting that with function is relevant to all these groups. So one route for getting there is that this is, we think this is, and I think the NIH also agrees, that this is probably the most efficient way to combine, to sort of achieve the Rosetta Stone goals that Adam Marblestone described in a bioRxiv paper a couple of years ago. Basically, to combine in a single specimen at single neuron, single synapse resolution information about gene expression, long-range connectivity, and function. And a lot of those components are probably preserved. So if we want to understand, for example, the circuitry, the subset of neurons, for example, that project from one brain area to another that might be particularly important, let's say, in alleviating depression or even in driving motor learning, this is potentially the fastest route to get there, certainly in a mouse. And once we have a pretty good idea of what the answer is in a mouse, we might know, we might have a better idea of what it is in a human. So that's one answer. I think the other, so I used to say it's a non-starter to ever apply this in humans because of the ethical issues. And I would point out that this is a destructive technology, and that trying to label up a bunch of human neurons with viruses might be challenging to get through an ethical review board. And at one of the talks that I presented this idea, a neuroethicist came up to me and said, well, actually, it's not inconceivable that you could get approval to do this in a terminally ill patient who's basically going to be unplugged in the next couple days, if, you know, under certain circumstances. And similarly, one could imagine doing it in brain slices ex vivo. So we're not actively pursuing that right now, but I think if this technology matures a little bit, it may turn out that it's not absolutely inconceivable that it could be adapted. And there are some really crazy ideas, just finally, that we've toyed with that might allow the barcodes to propagate even under certain ex vivo conditions other than slices for a whole brain. So I guess that those are the routes that I see right now. So I guess that those are the routes.
Thank you again very much, Tony. This was
Thanks a lot.
Fascinating talk. And okay, so if you've stopped screen sharing, so that's all fine. Then I'm going to go on and introduce our next speaker. Our next speaker is Dr. Adam Marblestone. Adam spent a good number of years driving research at the MIT Media Lab of Ed Boyden, with work on optical connectomics and expansion microscopy and many other things. He's also known as the main author of a number of strategic papers, such as the one that Tony already mentioned, exploring ways to accomplish high-resolution brain mapping. And Adam is the Chief Strategy Officer of the neurotechnology startup Kernel. I think that Adam's talk today is going to be an excellent complement to the one that Tony just gave. So Adam, please feel free to begin sharing your screen. With the neurotechnology startup Kernel.
Okay, can you guys see my screen here? Let's see. Can you hear me and can you see my screen?
Yes, we see you, we hear you.
Okay, great. Let me know if that changes. Yeah, so it's great to go right after Tony, because it's going to make it a lot easier to explain what I'll be talking about, which is mostly kind of additional layers of chemistry and some extra explanation that will hopefully clarify some of the things that he said as well, very much in the same spirit. And yeah, the purpose of what I'll be talking about is this notion of static. Is this notion of a Rosetta brain, very comprehensive mapping of as large a volume of a brain as possible, ideally an entire brain. And this is describing work that's done in the Boyden Church and also Tony Zador's lab over the past few years. So as I think some other speakers talked about, kind of the gold standard way to look in fine detail at neural circuitry today is the electron microscope, which involves doing very, very fine slicing and imaging, essentially, of the morphology of neurons. So this is the cell membrane is typically the main thing that's labeled. You see a little bit of extra, electron density for certain molecular complexes, like the postsynaptic density that tells you something about synapses or synaptic vesicles in some cases. But mostly you're looking at the cell membrane, and that's about it. Or at least the membranes of the cell and some organelles. And in terms of doing a Rosetta brain type concept of being truly comprehensive, this is sort of going to go over similar ideas as Tony presented, but just in a slightly different angle on them. There's a few other things that we would like to be able to see, that it is still an outstanding challenge for electron microscopy to be able to give us this additional information. So one of them is basically molecular. So on the left here, this is a slide from Yves Martyr, who studies the crab stomatogastric ganglion, which is a region in the gut of a crab that basically has about 30 or so neurons, and it actually has more neuromodulatory molecules that affect it, influencing how the synaptic transmission works, or influencing how the ion channels in the cells respond in time, or things like that. It has more modulators than it has neurons in the circuit, and it's interesting to think about whether that will also be true for other circuits, or at least the complexity might not have gone down much as you go up into something like cortex of a mammal. The other is sort of very fine spatial resolution, which electron microscopy has, but we would also like to combine with these barcode aspects, and also to combine with the molecules. So on the right is showing a diagram from a region of the thalamus called the lateral genicular nucleus that relays information from the retina into the visual cortex. And what you're seeing is basically the incoming synapses onto one of these thalamic relay cell dendrites. And what you can see is that not only are these synapses arranged in a complex pattern in space with different branching axon terminals and so on, but also each type of synapse is using slightly different combinations of transmitter and receptor molecules that are going to influence how what the actual meaning of these signals and how they affect this thalamic relay. And likewise, it's quite plausible that this kind of complexity also extends into other circuits throughout the mammalian brain. So these are things that we would like to have. Electron microscopy has a bit of that in terms of the shape and the spatial layout, but doesn't have the molecules right now. And of course, we want to have what Tony talked about, I think, which is basically, in my mind, what makes it dramatically easier to scale this to large volumes and long distances for any kind of structural mapping. Because oftentimes what we would want to look at in neuroscience is not just a local circuit, a few hundred microns or cubic millimeter size circuit, but you might want to look at something like the connections between the cortex, the thalamus, and the basal ganglia, and how those are all mutually interacting to have a kind of complex architecture. You really want to know the inputs and outputs to an area, in addition to just what that area is doing internally. And so having these unique identifiers for cells, these barcodes that Tony talked about, is also something that I view as essential for doing comprehensive mapping at a kind of whole brain level. So as Tony mentioned, a way that we can try to put all these things together is by doing the sequencing of barcodes, and as I'll mention, also potentially of other molecules that are present in the sample in addition to the barcodes, doing this sequencing of those as an imaging approach. And as Tony talked about, basically this mirrors the way in which, if you get your genome sequenced today, what you're essentially doing is taking your genome, get chopped into lots of small fragments of DNA, which land at different positions on a glass microscope slide. And then essentially the sequencing machine is actually a fluorescent microscope that takes a series of images of each of those fragments, reading out as a sequence of colors, one letter at a time, the identity of each strand of DNA. And then computationally, what people do is they take all those short fragments and they try to re-stitch that back into your genome. Here, and this is a technique that was developed in the Church Lab, and now as part of this collaboration with Ed and Tony, further developed, is this notion of fluorescent in situ sequencing, and is being applied to these cellular barcodes. So I think the question that we get to if we want to combine all these elements, though, is how do we get a sufficient spatial resolution of doing this in situ sequencing to read these barcode molecules or other endogenous molecules, and we can illustrate this question of spatial resolution by considering just how much spatial resolution do we actually need to see every individual synapse, because the synapses are packed closely in space with about one or a few per cubic micron of the brain tissue, at least in the cortex. And so one of the issues that you run into if you're trying to use an imaging-based approach to read whether it be barcodes or morphology or other things, other molecules, is that the synapses are packed densely in space, and you don't want them to overlap, or else you won't be able to resolve what you're looking at. And so what various people have concluded, starting with Yuri Mishenko back in 2011, who studied a related problem of optical detection of synapses, is that in order to actually uniquely see each synapse, you don't necessarily need quite the level of resolution of electron microscopy, the kind of resolution that Ken Hayworth is talking about, where you would maybe have 5 or 10 nanometers XYZ resolution, but you do need probably resolution more on the order of 100 nanometers, whereas a conventional optical microscope is more like 300 or 400 nanometers in the XY direction, and slightly worse than that for most microscopes in the Z direction that the light is actually propagating along. So we felt that we needed to introduce some additional elements into this picture to try to boost the spatial resolution, initially to try to see resolve synapses better, and as I'll talk about, to also to potentially resolve other aspects of the cell better. So back in 2014 or so, when we were starting to think about this, this seemed like a rather difficult problem. There were certain existing super resolution microscopy approaches that could be applied. Some of them involve in looking at the stochastic blinking of single molecules, kind of sparsifying the sample so you have most of the molecules are off, and you can turn a subset of them on and try to find the centers, and then stitch the image back together by localizing the centroids of certain dots. Another idea that came up early in the work around in situ sequencing was maybe there are other ways to sparsify the image, so you wouldn't be looking at everything at once. Everything would not have to overlap. You could look at it in a series of stages where only some of the molecules were being looked at. There were other groups that were doing ultra thin sectioning, kind of mirroring some of the work that's done in electron microscopy, but here being used with an optical microscope. And then there's some other ideas around computational approaches to resolve overlapping objects. But basically, I was fortunate to be in the loop at that time with Ed Boyden's lab as well, which had started developing a totally different approach to increasing the spatial resolution of any kind of optical observation of hunks of intact 3D brain tissue. And the new approach is called expansion microscopy, and is basically just that. It basically means physically making the brain itself larger rather than trying to essentially make the light smaller. You take the same microscope, but you physically swell the brain. And the way they do that is by infusing the brain tissue with this swellable polymer, sodium polyacrylate, which can, when it is exposed to water with a low salt content, will start to create chains that repel each other, and will swell and hydrate as an expanding hydrogel, and will actually blow up physically the sample initially in what they demonstrated by about 4 or 5 times along an axis. And so that gets us kind of exactly into the range of resolution, starting with a regular optical microscope of sort of several hundred nanometers to a micron resolution, down into the sort of sub-100 nanometer resolution range that seems to me to be kind of the right range for doing a Rosetta brain mapping effort. And in order to actually allow the brain tissue to swell, one of the things that the folks in the Boyden lab had to do was come up with a trick, which is how do you allow the tissue to swell uniformly, right? Because if you just infuse this polymer, different parts of the tissue are going to have different mechanical stiffness and so on. And what you expect to happen naively is basically that the tissue will tear rather than swelling uniformly, or it will swell to a certain point and then not be able to swell any further without tearing. So what they did is they played a trick, basically, where what they do is they first deliver to the molecules of interest that they want to look at, which in this case was proteins that are tagged with a chemical tool called an antibody, which binds specifically to certain proteins and could deliver other molecules to them. So they tag the proteins that they want to look at with an antibody, and attached to the antibody is a little strand of DNA. And the DNA has on one end a molecule which will link chemically into the expanding gel network, and on the other end it has a fluorescent dye. And so what they do is they deliver these tags, anchor the fluorescent molecules into the expanding gel, and then they digest away with an enzyme a lot of the endogenous structure. They get rid of a lot of the protein, a lot of the lipids, and they're only leaving behind a kind of a shadow mask of what they originally wanted to see. But then that shadow mask is this very dilute solution that is basically able to be expanded uniformly. And so this works very nicely, as they showed with a rainbow labeling of neuron membranes here. This is this exact same exact same image of the exact same location in a chunk of rainbow labeled tissue, on the left without any expansion, and on the right with expansion. And you can see that you're resolving finer details after expansion. Again, this is the same microscope. And so what they've done here is they've anchored fluorescent tags indicating the different colored rainbow proteins, and allowed those to expand uniformly. So we have been trying to combine this method of expansion microscopy with some of the things that Tony has been talking about, and also with a whole suite of other molecular tagging and indexing and readout chemistries to try to read out as much as we can from the same expanded sample. And again, for each thing we want to see, we have to find some way of tagging it into the expanding gel and allowing that to expand along with the gel while everything else gets digested away. And what we're showing here, this is actually a simulation based on electron microscopy data of what we would see, so that we can understand the parameters in kind of an ideal scenario. We have a simulation of what we will see looking at labeled synapses, which could, for example, be barcode tagged synapses at different levels of expansion. You can see that kind of at 1x or 2x, it's sort of blurry and hard to resolve individual objects. But once you get up to about 4x or 3x or 4x expansion, you start to see the individual synapses as very well resolved objects. And this is similarly borne out in some of the papers on expansion microscopy where they've been labeling synapses. Essentially, this approach for Rosetta brain mapping that we're trying to do is to combine these three technologies. So expansion microscopy, barcodes of the type that Tony described, and in situ sequencing into a single approach. And there has been some progress on this from Richie Komen in collaboration with Tony's lab and the rest of this group on doing that combination of all three technologies that we're continuing to work on. At the same time, we are asking the question of whether we can push this to even higher resolution. So the original expansion microscopy papers did about 4-fold expansion, and again, this is getting us down into sort of 100 nanometer or so resolution range, which is quite a great boost for in situ sequencing. technologies, but is not at the level of electron microscopy. What we're showing here is an iteration on the approach, which is literally done by iteration. Actually, what they do is they take the expanded hydrogel, and then they do a second round of the same expansion microscopy process on top of that initially expanded one. So they're doing it twice, from Jaebun Cheng et al. In Ed's lab, where now with the 20-fold expansion, you are starting to get down into the kind of 20 to 30 nanometer resolution range, which is still not quite at the level of electron microscopy, but is starting to get close. And here what you can see in these images is actually that we're starting to become limited actually by the chemical tagging process, where we're starting to see the discreteness of the chemical tags down at the molecular level as a limiting factor in our resolution. But there's ongoing work to try to push beyond that. And so here also you can do this with synapses, showing now optical imaging of several different molecular tags on the synapses, starting to get at the idea that when you're looking at the connectome, it would be great to also characterize something about the molecular makeup of each synapse. So here on the upper left there, they're showing three different antibody tags against different components of the synapse. Now, how do we fundamentally put all these things together? So we're also thinking about computational strategies that you could use where the morphology information that you would get from a high resolution expansion microscopy picture of the cell membranes, kind of like an electron microscope image, except probably at still somewhat worse spatial resolution than the best that one can do with electron microscopy, a la FIBSEM that was talked about. If you imagine you have a kind of crude optical approximation of the morphology, but still at relatively comparable spatial resolution, and you also had barcodes indicating the cell identity in a unique way. So from a single point, wherever you see a barcode, you could uniquely determine the identity of that point, but perhaps of nowhere else. Can you use that to synergistically reconstruct the full geometry of the circuit over longer distances than you can with electron microscopy, let's say, or with purely with the morphological imaging, and also, kind of take a barcode that you find and fill out from there, and then go and identify the nearby synapses and identify the detailed shape of the cells that are surrounding that barcode that you've identified, maybe comes from some other part of the brain. So this is also something we've been trying to evaluate in computer simulations, where we're starting with data sets that are collected, in this case from Jeff Lichtman's lab, in a published paper, with showing the reconstructed geometry of a small volume of neuropil using the electron microscope, and then simulating what that would look like if we took that same volume and imaged it with expansion microscopy with this 20x form as it currently stands, roughly, in terms of the performance of labeling and resolution that it can get. And we simulate that we are distributing in these neurons a kind of smattering of Tony's barcodes in space, and that we can do the in situ sequencing process at the same resolution in the same sample. And so the approach that basically we're taking is basically shown in this series of panels here on the slide, where there is a relatively conventional approach that's used to reconstruct neural morphology if in the absence of any barcodes, which is to say that what you do is you first identify the cell membrane. So you have a first stage, and this is often done with a convolutional neural network, where what you do is you just label each pixel. Is it membrane or is it not membrane? Or essentially, what's the probability that any given pixel is on the membrane? And that gives you a probability map showing how likely each pixel is to be a boundary. Once you've identified those putative boundaries, you can do a segmentation operation called a watershed to basically try to find what are the contiguous regions that are bounded by that boundary map. But what that tends to do is to over-segment and create lots of little mini pockets that really should be identified as part of the same neuron, but instead is identified as its own little contained boundary. And so the question is there anything that we can do at that stage to assist the process of merging those pockets back together to identify which cell, which actually go together and are part of the same cell? And that's in this algorithm scheme. That's exactly where the barcodes come in, because if you attempt to merge together two local units that have different barcodes in them, then you know that you've made a mistake. And on the other hand, if you have two barcodes that are from the have the same identity, the same DNA sequence,
I don't know if you can hear me, Adam, but we just lost your sound. I'm not sure if the problem's on your end or ours. I don't know if you can hear me, Adam, but we just lost your sound. I'm not sure if the problem's on your end or ours. Yeah.
can you guys hear me again?
You're back, Adam, I think? Yes, we can. Back, Adam, I think?
Okay, great. I think I just had an internet connection issue on my side, so I'm just going to start back where I left off, and then I'm almost done. Does that work?
Yes, we can. Okay, great. I think I just had an internet connection issue on my side, so I'm just going to start back where I left.
Okay, so
Perfect.
anyway, closing up here, but we would like to also use the DNA that naturally happens in the expansion microscopy process, and actually use that as a barcode to tag different proteins that are present in the sample as well. And so basically using this and building on what Tony said, we really do think that we could eventually achieve a kind of Rosetta brain type comprehensive mapping where we'd be looking at connectivity with barcodes as well as with morphology, gene expression by looking at endogenous RNA using the same sequencing process, looking at many proteins also using DNA barcoding, and potentially other things like the cell lineage tree, which can be encoded in DNA, or ticker tapes, as some folks were asking about in the questions. So with that, I will close and acknowledge a large collaboration that has mostly done all of this, including Tony's lab, Ed Boyden's lab, and George Church's lab, and funding from IARPA. Thanks a lot.
Thank you, Adam. Thanks very much for this fascinating talk. Let's give you an applause here. Okay, so the questions about this, I mean, this is kind of questions for both you and Tony in a way, because it's sort of one big thing. There are lots of different kinds of questions we can ask. Here's one that happens to reach all the way back to, you know, the origins of the whole workshop as being about mind uploading. This comes from Alexander McLean, and he's asking, if brain preservation becomes common, is there a possibility that the preservation protocol could make it difficult to use expansion microscopy techniques if it becomes an important tool for mind uploading?
If brain preservation becomes...
Yeah, that's an interesting question. Yeah, so I think one thing that I should mention is that the expansion microscopy protocol actually that I described there, a formaldehyde-based chemical fixative in its current embodiment. So it actually can be done on heavily fixed and preserved tissue. Not even yeah. So for example, this aldehyde stabilized cryopreservation work from Robert McIntyre and colleagues, something that has a fixative, is something that can work with expansion microscopy. I think that it relates to a question that was asked to Tony, which is, how would you do the more genetic and barcode based approaches in a human to begin with? And that's obviously open as to whether that's possible still. I think that would be sort of the more difficult part, is that these barcoding technologies, they rely on viruses to actually replicate inside the neurons and make copies of these barcodes. And so it's something where the procedure essentially starts while the animal is alive. But the preservation as such, I don't think that necessarily has to inhibit expansion microscopy. And we're working anyway on trying to preserve tissue as well as possible to get the best just morphological information out of it with expansion.
Get the best just morphological information out of it with expansion.
Thanks, Adam. We're going to go on with a whole variety of different questions, some maybe far beyond what you just presented. But before I go into a question that I thought was a nice tangent, let me just check the audience right here in the room. Daniel? Yeah?
What does your research reveal about how expansion
Okay. So Daniel is asking, what does your research reveal about how expansion microscopy changes protein folding, etc.?
Yeah, it's an interesting question. Yeah, so it hasn't yet been done at a level that tries to fully preserve protein folding. So in the original version that I described, it has proteins in a state where they are chemically fixed, and then they are addressed with antibodies to tag them, but then the proteins themselves are digested away. So we know that it preserves, because the fixation preserves enough of the protein conformations, they're called epitopes, that the antibodies can still bind, that's pretty good. But and then there's been various follow-on works that is trying to preserve more protein information and be less disruptive to the proteins, in the sense of not actually digesting them away. But this has not been done yet down at the kind of sub 5 nanometer length scale, where we're still kind of one level of size above where we're looking at detailed protein conformations. Not to say it's impossible to look at detailed protein conformations, but that might be another couple expansions on top of the 2 iterations there.
Not to say it's impossible to look at detailed protein conformations, but that might be another couple expansions on top of the 2 iterations there.
Thank you. Okay, so a question that maybe wasn't directly related to the technique you're showing, but is kind of it's sort of a question about all of this connectomics related stuff that we've been discussing right now. Online, one of our viewers has asked the question, how can neuroanatomy be correlated with activity or thought?
Right. Well, I mean, one of the ways, and Tony started to describe this as well, is that one can try to actually measure both in the same brain. And regardless of any analytical predictive method for correlating that, you can actually just try to take try to just measure them both in the same organism, in the same circuit. And this is something that one can do, and it's actually something that this IARPA project is actually requiring people to do for a certain volume, around the size of a cubic millimeter, to try to get what's called calcium imaging. So basically you're seeing the dynamic movie of the cells having influxes of calcium as the neurons are firing. And the calcium basically triggers greater fluorescence of a fluorescent dye. And so you're seeing a movie of the cells turning on and off. And then we can go take that and then go and try to do structural connectome type mapping on it. From a kind of analytical or predictive perspective, I think it's still an open question. Essentially, what exactly do you need to do to predict, based on anatomy, some aspect of the dynamics? And you may not be able to predict everything about it, but if there are things like stable attractor states or so on in the circuit, you might be able to at least get enough to know something about how that circuit is going to function, or what information is going to encode, even if you won't in a single trial kind of predict, every ion channel opening or so on.
Information is going to encode, even if you won't in a single trial kind of predict, every ion channel opening or so on.
Thank you. So I wasn't sure if Ken is still actively listening to us at this moment, but I couldn't help being curious.
I'm still here.
Okay, great. I couldn't help being curious, given these 2 last talks showing the promise of barcoding and expansion microscopy and using optical microscopy. What would you see as sort of the limitations that still make EM an interesting approach, or where do you see sort of the transition from one technology to another? And maybe Adam can then chime into that. But first, I'd like to hear from you, Ken, if that's possible.
Well, thanks. Yeah, I'm just here waiting to have them put me out of a job. That's all. No, I think that these techniques are fantastic. I think in terms of understanding the nervous system, there is probably places for all of these techniques to attack the exact same systems, so that they can be cross-checking each other. For example, I think what Adam said to one of these questions, though, is really my main take on it. Some of these techniques require that the animal is injected with viruses while it is alive, so that these barcoding techniques, etc., can have time to express, and that is very powerful in a research context. I think it would be very difficult to imagine a way for it, although they've done a great job explaining one way, for this to potentially work its way into the brain preservation protocol, if you will. So the way I look at it is that the brain preservation protocol tries to do the least amount of change, because we don't know what the future technology is going to be and what is going to be necessary. So preserve as much information as possible. And if we were to say go down the route of developing a set of barcoding virus tags that are injected just prior to the brain preservation procedure, which I think is what they were saying, that would mean that we would have to figure out what tags are necessary, what molecular tags, etc., I believe, would be necessary today. And that might be too much to ask for. So in any case, in answer to your question, I think that these are fantastic techniques, and they could vastly accelerate the imaging of the connectome for at least research study beyond the type of very slow electron microscopy that I was describing.
Yeah, so I guess I agree with what Ken said, and I would further add, that these are the optical and molecular techniques are still very much works in progress, right? So we don't have the connectome of anything using them yet, and as with any other biological technique, there's always unexpected hiccups and challenges. So I think that it's kind of a proof is in the pudding situation in general in terms of the ability to do connectomics with this still, although I think it's making progress. The other aspect is that we really don't know that we can get the same spatial resolution and the same preservation of the super detailed morphology. And in the absence of that, exactly what you can do is still open. So for example, if we're trying to traffic the barcodes to synapses, are they going to traffic to every synapse? If we're not trafficking the barcodes to synapses and we're relying on some kind of morphological filling out, starting with a barcode and going out into the morphology down deep where the synapses are, then we have to make sure that the morphological quality of that is good enough. And none of those things are yet established. So I think that both because of intrinsic differences and because of where the technologies stand right now, it makes sense that electron microscopy is still sort of the de facto way to do connectomics, and that it should be pushed, even further, like in the ways that Ken is doing to try to get, FibSem and super precise mapping that can allow you to do larger scale reconstructions with that.
Mapping that can allow you to do larger scale reconstructions with that.
Yeah, thank you both. Okay, so taking a quick look at the time, I think this is a great moment to switch topics somewhat. Let's see, I've got some feedback here. Okay. Thank you both. Thank you, all of you. Now, finally, we're welcoming Professor Ted Berger, who is here with us today at our San Francisco location. He's momentarily moved into the room next door so that he won't be bothered by the live stream audio in here while he's talking to all of you. Ted holds the David Packard Chair in Engineering and is the director of the Center for Neural Engineering at the USC Viterbi School of Engineering. He's professor of biomedical engineering, and while there are many fascinating projects in his lab and collaborations, for our workshop today, we are of course particularly interested in the work that Ted and his associates have done towards building functional neural prosthesis. So we may give Ted whatever minutes are necessary for the final wiring up here, and so perhaps we'll have just a little bit of silence if necessary, or Ted can start anytime he wants to. We are now open for screen sharing at any moment.
That's great. You start at the beginning. Yeah, good. Let's see that. Okay. Let me see if you can. Hold on a second. Okay. Okay. Great. I think you can. Okay, great. Thank you, Randall. I appreciate the opportunity to be here, and it's already been incredibly fascinating. So this has really been a wonderful workshop. I'm going to tell you about the development of a prosthesis, a neural prosthesis that can facilitate human memory. And this is work that's been done by a large group, and so I have to make sure I recognize my colleagues. There are quite a few that are involved in mathematical modeling and biomedical engineering and hardware development, and of course the clinical aspects of introducing this into humans. So our group keeps growing and growing, and I represent only a part of all of the hard work that's done by this team. This prosthesis is for, as I mentioned, for the hippocampus, and the hippocampus is responsible for creating new long-term memories. And these are memories particularly for what are called declarative memories, or memories for labels and facts, names and facts. So if you lose both of the hippocampi on both sides of your brain, previously created long-term memories are still intact, and you still have a fully functioning short-term memory, but the ability to create new long-term memories is what's lost. And these long-term memories, in fact, by virtue of those clinical data, it's obvious that long-term memories are not stored in the hippocampus. The codes, the neural codes for new long-term memories are stored, so the long-term memory codes are created in the hippocampus, but then they're sent elsewhere in the brain for storage. And there are a variety of conditions that can lead to loss of the hippocampus, and as a consequence, this loss of an ability to create new declarative memories. And we all know most of these. Alzheimer's disease and various forms of dementia, epilepsy, certain types of stroke, and blunt head trauma. All of these disorders result in damage to the hippocampus, and as a consequence, they result in loss of the ability to create new long-term memories. And so what we have been trying to do is to develop a procedure that will allow us to circumvent the effects of this damage by first learning enough about how the inputs to the hippocampus are gradually transformed into new memory codes, new long-term memory codes. We need to know enough about that we can reproduce it in the form of a mathematical model. And then, and that's shown in this slide. It's labeled MIMO model. I'm sorry, I probably should have labeled it something else, but the MIMO model stands for multiple input, multiple output. And so this is a MIMO model that instantiates this transformation of a neural code from short-term memory to long-term memory. That's obviously an essential piece of the prosthesis. And then there are two multiple electrode arrays. One records from multiple neurons that are upstream from the damage. So we're recording from the inputs to the hippocampus. And these then are transmitted through these electrodes to the biomimetic model, which then, as I mentioned already, transforms this input into a different output, into a long-term memory code. And then there's a second multi-electrode array that transmits this new long-term memory code back into the brain, downstream from the damage, and we electrically stimulate the output of the hippocampus with this new code. So whatever the part of the brain that's the part of the hippocampus that's been subjected to damage and can no longer convert the input to the output effectively, we bypass that, and by using this approach. And this is the model. This is the concept. This is the plan. And what we've been doing over the course of the last 15 to 20 years now is studying this evolution of short-term memory codes to long-term memory codes. We've been recording from animal and human brains so that we can understand more and more about this transformation from short-term memory codes to long-term memory codes. And we're able to mathematically model this fairly effectively, so that now, given a short-term memory code, given the input to the hippocampus, we can very accurately produce what the output hippocampal code should be, and I'll show you examples of that. But we started first in using hippocampal slices to remove a certain part of the hippocampal slice and show that with an input-output mathematical model, we could recreate the transformation that normally occurs as activity propagates through the hippocampus. And then we worked in behaving rats, showing that we could apply this prosthesis to memory in the behaving rat, and that we could, by inactivating the hippocampus, that our device could substitute for hippocampal function. And then we moved to the behaving monkey, and now we're working in humans. So it's been a gradual process where we've progressively applied this concept to the hippocampus and through these different species. And forgive me if I'm stammering now and then, because I'm hearing I'm hearing what's coming in over the internet, and I'm trying to speak at the same time. So I'll do the best I can. So what are we doing now? And again, this is the work that I'm going to show you today is primarily the product of work with our colleagues at Wake Forest. We now are getting epilepsy patients. We're using epilepsy patients for most of these studies. And we now have three sources of patients, one from Wake Forest, second from USC Medical School, and then from Rancho Los Amigos Rehabilitation Center, which is also in Los Angeles. So anyway, this slide shows you the approach that we use for implanting electrodes into the hippocampus, and we use this lateral approach. And the type of electrode that we use is shown in the bottom right here. And there are these very wide bands that you see there. Those are for recording EEG-like activity. But the small holes that you see, there are electrodes that protrude from those holes, and these electrodes record single unit activity. So they record the activity of individual hippocampal cells. And we use two parts of this, two sets of holes, so that we record from two locations. And this has been a common strategy that we've used through all of the work that we've done. We record from two locations in the hippocampus, one in an area called CA3 and in another area called CA1. And I'll show you a bit more about this. But the point is that we can look at activity in two locations, two parts of the circuit, from the input to the output in the hippocampus, so that we have a chance to examine this transformation. The codes, the memory codes that we record from these two locations are not the same. They shouldn't be. As activity propagates through any brain structure, spiking activity is transformed quite radically. The synaptic transmission is a nonlinear process in terms of signal analysis or signal transmission. So the codes are very different, and so we examine the codes in these two locations so that we can develop an understanding of how input codes become output codes. And this slide shows you, this is a cross-section through the hippocampus. It happens to be a rabbit brain rather than a human brain, but it's okay. But I'm showing you different parts of this circuit, and its activity begins in this region called the entorhinal cortex. And I don't want to go through labels, but the point is, here you can see that area CA3 then projects to area CA1, and then from CA1 it goes to an area called the subiculum, and then back out through the entorhinal cortex. So there's this loop. Activity propagates through this loop, and the CA3 zone and the CA1 zone are a major part of this internal hippocampal circuitry. So we record from those two areas, and we get a chance, as I mentioned already, we get a chance to see what the memory codes look like in these two locations. And so the point is that we're going to use activity recorded from CA3 to predict the activity that's shown that occurs in CA1. And it's on this basis that we will reintroduce long-term memory codes in the hippocampus. So again, this slide basically says the same thing, that we're recording from these two locations. But the point here is that when we implant the patient, and then the next day we start to record activity from the hippocampus, and usually by day 1 or day 2, everything is stable enough that we show sample objects to the patient. We ask the patient to try to remember what these objects are. And while the patient is doing that, we record from these two cell fields, CA3 and then CA1. And CA3 provides input to CA1. And then on subsequent days, we present the same set of objects, but we record the memory codes from CA3 as we did on the previous day. But now we let the mathematical model, I'm sorry, I didn't really explain this well. So on day 1 or day 2, we show these sample objects to the patient, and we record from these two cell zones. And then we take a couple of days to analyze that data and develop an input-output model that allows us to predict CA1 memory codes based on what we record from CA3 with those memory codes. So on subsequent days, we record from CA3 while we show sample objects to the patient. The mathematical model predicts what the output should be from CA1, and then we electrically stimulate the output of CA1 with the model predicted code. So the patient is endogenously creating their own code. They're trying to remember. They're trying to formulate a memory for this object, whatever the object is, and we electrically stimulate with our exogenous code. And so the two codes together, we make a stronger code, and in this way we facilitate the memory because it's a stronger code. And I can tell you more about that if you're interested, but that's what we're that's how we're. It's one way in which we're facilitating the memory for the patient. So what kind of codes are these? You probably all know that neurons communicate with each other using all or none signals, just like the all or none events that occur in microelectronic circuits. So neurons transmit information using temporal patterns. That's a key part of neuronal function. And so if there are different objects, objects and events are represented by populations of neurons, not just by single cells, but it's necessary to look at population activity. So populations of neurons, if there are different events, then there are different spatiotemporal codes. And like I've shown here in this slide, pattern A is a different spatiotemporal code than pattern B. So they would represent different events, different memories. And cognitive operations are neural functions that transform spatiotemporal patterns. So the cognitive operation of forming a new memory would be converting pattern A to pattern B. So what we're looking for are these spatiotemporal codes, and the mathematical model predicts what kind of transformations in spatiotemporal codes occur in the hippocampus as a result of propagations of input activity into output activity. And this is an example of how well the model predicts the activity in the hippocampus. These are a set of recordings from 50 cells in both in CA3 and in CA1. We recorded 50 neurons in each structure. And what we're doing, what's represented so time is along the x-axis here, and cell number is along the y-axis. So the y-axis is just location, basically, or cell number. You can just you can see the space-time pattern that's exhibited here, and the space-time pattern that we recorded is shown in the center panel, or the first color pattern panel. The space-time pattern that was predicted by our model is shown below. And these are patterns that were generated as a result of, presenting objects to a patient and asking them to remember what the object is. So each one of these space-time patterns represents a particular object. So and this is just another example of how well we can predict the input-output transformations that occur in humans. We need neurons. We need enough neurons to record with this kind of to predict with this kind of resolution. As the number of neurons gets smaller, the predictions are not as accurate. Anyway, so what we ask the patients to do, it's called a delayed match to sample task. It's very commonly used for hippocampal memories. So we show there's a phase called the sample phase, which is when we present a figure to the patient. It's called a can-tab image, so it's a square. It has 4 inner squares or subsquares, different colors, and within each subsquare there are different numbers of white squares or lines that are that occur. So it's a complicated pattern, and the patient is given 3 or 4 seconds to look at this and try to remember what it is. Then there's a delay of anywhere from 1 second to 75 seconds. We take the object away almost immediately. Then we show the patient several of these. It can be anywhere from 4 to 6, 7 patterns. And the patient has to pick the object that they saw previously. So that's very simple. So what happens. This is the result from just from one subject, just so you can see better what happens. The yellow bar here is when we're recording only on day 1. So this patient was able to accurately remember the object that they saw on the sample trial at about 36-37%. And it's an example of how epilepsy patients, because of the synchronous activity that occurs over time in the hippocampus, the cells become damaged, and so their memory capacity is lowered. On the day 2, when we presented the object, but we electrically stimulated the hippocampus with the space-time pattern for the object that the patient was looking at. So there are different objects, and they each have their own individual space-time pattern. As long as we use the one that's appropriate for the pattern that the patients are looking at, you can see that the level of accuracy increased to almost 60%. If on day 2 and day 3 we didn't stimulate, then it was lowered to about what we saw on the first day. And importantly, if we stimulate with a random pattern, that's this green bar, we actually suppress the ability to remember. So the timing, the temporal pattern, the temporal timing is really important. If we don't have the correct space-time pattern that matches the object being shown, then there's no enhancement of short-term memory. And this is just these are the results for the first 6 subjects that we had. We had actually several subjects where we worked out all the details, and then we started testing epilepsy patients with the standardized protocol. And these 6, 5 of them showed this enhancement. The 6th patient only had electrodes in 1 hippocampus and not 2, so there wasn't any enhancement in that patient. So in overall, the enhancement was about 40% for this kind of short-term memory. And then we tested for what we call long-term memory, where we used delays in the range of minutes rather than seconds. And I won't bother with the details of the paradigm, but it was approximately the same kind of paradigm. But the delays were between 15 and 45 minutes. And we also saw enhancement. We had 4 patients that so far that we've tested, and the facilitation was even greater. It was about 60%. Random stimulation didn't have any influence on these subjects, but there was a rather dramatic increase in long-term memory. So we're continuing with that. Again, these were patients that were only from the Wake Forest group. We've had more patients from USC, and we're standardizing the protocol right now, and soon we'll have we have a couple of patients already that are not included here from USC that are showing similar results. The sort of thing that we're doing in the future, I don't know how long I've been talking. Should I shut up pretty soon, Randall? Or I guess he can't hear me. Randall, am I okay? All right. So one of the areas, one of the directions we're going in is, okay, thank you. One of the directions we're going in is called memory decoding, and you see the previous tests. Essentially what we're doing is, there's a stream of activity that's propagating through the hippocampus from the input through several of these synapses and then to the output. And we have a situation where somewhere in that circuit there's damage, so that the progressive transformations that occur at each of these sites is disrupted. And so we need to reinstantiate the normal, I'll call it normal, spiking activity along parallel pathways that constitute the internal circuitry of the hippocampus. We have to essentially reconstruct these signal generation and signal propagating components, and get them to work together again. And that's what the multi-input, multi-output model does. But we don't actually know what the signals mean. We're recreating these parallel pathways and the functionality of these parallel pathways. And when we do that, the patients are able to create long-term memories better than without our device. But we don't really know what's propagating along each one of those pathways. We don't know what the signals mean. So at some point, we want to be able to look at the space-time patterns that are generated by hippocampal cells and be able to read it. We want to be able to look at the code and say, oh, well, this is a memory of a tree. This is a memory of my jaguar, or this is a memory of my mother. You know, we want to be able to identify what these things mean. Now, trying to develop the capability of reading memory codes for a huge variety, you know, a huge dictionary of objects is too much. So what we're starting with is trying to understand the memory code for categories of events, categories of things. So we're using CA3 and CA1 spike trains that we record from epilepsy patients. In other words, we're asking them to do the same kind of delayed match to sample test, except we're using standard pictures that we download from the internet. But then we test to see how normal subjects categorize these objects. Do they categorize them as animals or plants, or you know, colors, according to colors, or whatever? So, and then what we're developing a classification model that uses the multi-input, multi-output data, or the multiple unit data recorded from hippocampus, and we're seeing if we can identify the categories that are represented by the space-time patterns that we record. And I won't go through the details of the model, but we're using these clip art images. We're using up to 100 objects, 1,000 images, and we're categorizing them using what end up being almost 30 memory labels. And these are the kind of results that we're getting so far, that our classification model is able to gradually learn the category represented by the space-time patterning, not close to 100%. It's more like 50 or 60% accuracy right now. This example was for animal, and this is another example for plant. So we're getting there, but this is obviously a work in progress. Well, all of this is a work in progress, and I think yes. So just to clarify, support, of course, incredibly important. We are working on microchip models, implementations of these mathematical models, but I'll just mention that we are doing that and not talk too much about it. So thank you. I think I'll end there if that's all right.
So thank you. I think I'll end there if that's all right.
If he stays there, it's useful for the Q&A. Makes it a lot easier on the microphone. Hi, Ted.
Hi.
Thank you so much for that presentation. That was absolutely wonderful. And as you might expect, of course, there are a number of questions that popped up as we went along.
Sure.
And if you can stay at the microphone there, then it helps to just make this all very, fluid.
Fine. Will do.
I'm going to start off with a couple of questions that came in on the internet. I think you partially answered those already, so sorry if it seems like there's some duplication of effort. The first question by Keith Wiley is the prosthesis giving any insight into how memories are actually neurologically represented in short or long-term memory, or is it merely learning and performing an opaque translation that, while undeniably medically useful, we don't actually understand?
Yeah, well, I mean, that's really a good question. It's true that we are just at the beginning of understanding what this data are telling us about how memories are represented. Of course, I'd like to believe that we're going to learn something about memory coding and the ways in which memory codes are represented in space and time in hippocampus. But we need to learn a lot more about why certain cell representations occur here as opposed to there, and how important particular temporal components are in a given memory representation. So it's going to require a lot more experimental work, and I think and I'm very sensitive to the question that you're asking, because it's really, I mean, that really is where the gold is. We really want to be able, I mean, there really are two pieces of gold. One is in the ability to create a medically useful product, and I really like the way you put it. It's really quite good. And so that's kind of the first goal. We want to make sure that if we utilize this in the form of a prosthesis, that it can continue to be useful to patients. Then you're right on target. That then the issue is what has this told us about how memories are represented in the hippocampus, and what kind of insights can we have in terms of the representational schemes that the brain happens to use for memories, and that I believe is going to take, as I just mentioned a while ago, a lot more experimental work, meaning that we need to manipulate the objects. And so, for example, if we, one of the objects that a patient, learns to look at, what happens when we, when we change the colors, or we change some of the shapes that comprise the object. What happens to the neural representation? And so, I think that's going to take a continued work in animals. It's not the kind of work that we can continue to do too much in humans, but part of what we're doing with the memory decoding model is definitely going to tell us, is going to start to answer some of those questions. So, for example, part of what we're setting up with the memory decoding model is attempts to electrically stimulate with the human brain with one of these classification or category models, and then see if we can bias the brain toward identifying the objects that are in that category. This is something that we plan on doing, and hopefully it's going to begin to tell us something in the directions of what you're asking. That's a very good question.
the directions of what you're asking.
Thank you. I'm going to give you one more question that came in on the internet before we switch to audience questions. This one is from Alexander McLean, and he asks, is it plausible to be able to generalize the mathematical process used to model transformations to the rest of the brain, or is the brain functionally too diverse to be able to generalize effectively?
I'm sorry, I didn't quite understand the question. There was some interference.
Let me ask you again. The question was, is it plausible to be able to generalize the mathematical process used to model and predict the hippocampus input-output transformation to the rest of the brain, or is the brain functionality too diverse to be able to generalize effectively?
It's entirely possible. We plan on generalizing the mathematical model and the entire approach to other parts of the brain and for other neural functions. This is the kind of approach that we're using is one of the approaches that Randall talked about in his opening statements. It's a systems identification approach, and it has been developed within engineering as a very general strategy for characterizing the properties of a system based on how inputs are transformed into outputs. So the functionality of the system is described and understood, quote, in terms of input-output transformations. So we not only hope to come up with a very general model of the hippocampus. In other words, our intent and our belief is that once we record from enough humans, that once we encapsulate in our model enough examples of inputs and outputs, that our model will be sufficiently general that novel objects, novel inputs will be accurately predicted. I'm sorry, novel inputs. This business of hearing what I'm saying in a delay is really hard. Okay, so once we record from enough different objects from different humans, the model will be able to correctly predict output memory codes for novel objects that have never been seen by the patient. And this is there's a lot of history. There's a lot of evidence I can't go into for believing that that can happen. And we've shown in a lot of ways in other neural tissue that that's a real possibility, and it's generally what happens with this kind of systems identification work. We've already been applying this same approach to the motor system, predicting EMGs based on from various muscles, based on recordings of population single units in the primary motor cortex. So essentially creating a mathematical model of the spinal cord so that we can take primary motor cortical space-time patterns and predict how up to 12 or 14 different muscles in the arm and hand should produce the correct movement, the correct space-time movement based on the intended movement predicted by the motor cortex. So this is definitely a general model that we hope to apply to further in hippocampus and to all other parts of the brain.
Thank you, Ted. Yeah, I'm so sorry about this whole problem with the feedback of the live stream going over to you. This is one of the complications of using this method. We're still learning as we go.
Sure.
Okay, I'm going to switch to some audience questions before we go back to more online. And I see here that we've got one. Yeah, Peter?
Yeah, so with your predictive model of CA1, what would the entropy of the observed firing be, and then how much of that entropy?
I'm sorry, I can't understand the question.
I'll repeat it.
And these
Okay.
are the 50 neurons in the plane.
Okay, so I'm going to try to repeat Peter's. Oh, actually, yeah, you can just tell Ted right there. That'd be much easier. Okay, go ahead.
Great.
All right. So the question, I'm going to see if we can get in front of the video.
Sure.
The question is, for your CA1 predictive model, there's some complexity or entropy to the distribution that you observe of firing there. And then how much of that can you explain or remove with your predictive model? Or perhaps another way of, is there some other mathematical measure that you have of how good your model is thus far and how much it's not yet explaining with those 50 neurons?
Okay, I'm not really sure. Not really sure I can answer it. I guess I mean, we use a statistical evaluation of the input-output model. It's a Kolmogorov-Smirnov method for evaluating. It's a non-parametric model. And that's what we use for evaluating the accuracy of the model. And in this case, for the very good examples I showed, we examine each one of the CA1 cells individually, and typically 3/4 of those will show statistically reliable or statistical reliability in terms of their predictive value. So some of the cell models don't reach significance, but about 3/4 of them, half to 3/4 of them do. And beyond that, I can't really say much else.
I have also a quick follow-up, which is it's very surprising that you can do as well as you can with 50 cells out of 420,000.
Yeah.
Do you have an intuition about what that curve of progress looks like?
Yeah, that's a really good one. So did you hear the question? I guess I can repeat it. So the question was, how can we do so well with recordings from just 50 neurons, particularly given the fact that the CA1 cell population is about 450,000. The CA3 cell population is about 350,000. So we're getting a fairly small sample. We're getting a very small sample from the large population. And of course, we always wonder about that. But it gets turned out to be the case. And certainly for motor cortex, it's very clear now from multiple different labs that we being the field, can extract a tremendous amount of information from very small samples of neurons. So there's something very important in there, that we only need small samples of neurons to be able to do a lot of wonderful things. And so that for example, recording from 40 primary motor cortical units, you can get very accurate movements of an artificial arm for three-dimensional movement, reach and grasp kind of functions. And that's always been a surprise. Right now, it's pretty much accepted, and so people just go forward. I didn't think that we'd be able to do as well as we do with we can do fairly well with 25 or 30 cells. So I can't really explain it. I don't know why, but it seems to be true for other parts of the brain as well as for the hippocampus. If I could try to fake an answer, but that's what I'd be doing. It's a great answer. Right.
So my question is, how do you do spike sorting and neuron identification in a patient whose hippocampus is already defective? And then the follow-up question to that is, once you have that data, how do your electrodes target individual neurons for stimulation without crosstalk, if you figured that problem out yet?
Well, let me answer the second question first, which is There isn't a lot of crosstalk. I mean, you can identify the cells. Also, answer both questions. You can identify the cells individually, and identify them on the basis of their, action potential height. But the waveform is what you use, and so it's reasonably straightforward to identify one cell and discriminate it from another.
Right.
So crosstalk in that sense isn't a problem. So what was the first question?
So the first question is, does spike sorting or neuron
spike
identification become a problem when the hippocampus is defective? It makes sense that it works when you're trying to predict a healthy hippocampus.
Sure.
But once you're going to a novel hippocampus where you can't do that sort of prediction model.
That's fine. Yeah, so I didn't explain one thing that we do. Well, it's two things. So the basic question, if I can reconstruct it, is how can we improve hippocampal function when the model is based on a damaged hippocampus, right? And of course, so there are two reasons why we can make the improvement. One of the things that we noticed in intact animals, my colleagues noticed, Sam Deadweiler and Rob Hampson, they noticed that when the signal-to-noise ratio, basically, of a pattern of activity was high, if you were recording from a monkey or a rat, if the, quote, signal-to-noise ratio of the space-time pattern was high, very prominent, then the animal was much more likely to make a correct response. And when the signal-to-noise ratio was low, then the animal was much more likely to make an error. Right? So one of the things, so what we do in our attempt to improve the hippocampal signals recorded from a damaged hippocampus is that we electrically stimulating the hippocampus and making the signal-to-noise ratio of the code higher, then the patient's memory should improve. Doesn't mean it has to be true, but that was the rationale for why we electrically stimulated with the same code, and we hoped that it would improve. There's a second thing that we did, and that is that along the same lines, we used for constructing the modeled code for a given object, what we use is the data recorded from trials where the patient makes a correct response. So we use to construct the space-time pattern that we use for stimulation. We only use those data where the patient makes a correct response. So we call that a strong code. In other words, it's a long-term memory code, but we know that it's correlated with correct responding. So the codes that are correlated with incorrect responses by the patient probably have error signals in them or greater noise or something. We don't know. But what we use are the trials that are only come from ones where the patient makes a correct response. And it's because of that that we can improve data from a damaged hippocampus.
So could you build almost just a neural amplifier to boost that SNR without understanding the neural code, or do you think it is necessary to understand the neural code?
Well, I think I don't know. It's a good question. So I think yes, you can introduce improvement with just basically an amplifier, but the fact that we get better responding when we use data from correct responses means that there could be much more in there than just larger activity. You know, there could be various aspects of code schemes that we're not sensitive to, but nonetheless they're in there. And that's one of the things we're analyzing right now is what is there about the responses recorded from trials recorded from correct responses. What is there about those space-time codes? How are those different? And so hopefully we'll find out.
Okay, brilliant. Thank you.
Thanks. Okay.
It looks like we don't have another audience question from here right now. I do believe that we had another online question that was interesting. Well, it's a slightly different type of question, so I'll just pass that one along. The question is from Leslie Seymour, and it asks, have you used any learning tools for recognizing and classifying the signal patterns?
Sorry, Randall, I didn't get it.
Can you hear me now?
Yes, I can.
Yeah. Okay, the question was, have you used any commercially available deep learning tools for recognizing and classifying the signal patterns as concepts?
In part, so in part, that's what we're doing with the model classification, with the memory that's what, in part, that's what we're doing with the memory decoding model. Some of those techniques, some of the techniques that we're using are based on deep learning concepts, but we're not using explicitly. We're not using deep learning tools that are available from anybody else. So in that sense, the answer is no. But we're using some of the concepts of deep learning in the memory decoding model.
All right. Yeah, okay, there's one more from the audience.
Hi. So I guess my question is, your model predicts the activity of individual units and specific firing rates over time, but presumably the stimulation that you give out can't stimulate individual neurons that specifically. It's kind of has more broad effects.
Right.
But you still see this improvement. So I'm curious, do you think that has any implications on how the hippocampal, that area of the hippocampus actually functions in terms of memory, and what that code might require in terms of specificity?
Yeah, it's very good. This is a problem that still haunts us. So the general question is that, we're recording from single cells, and that's what goes into our model, the data from single cells, and yeah, both from the point of view of input and output. But when we're using electrical stimulation, we cannot be, almost by definition, stimulating single cells. We may be stimulating through the same electrodes that we use for recording, which we are, and we were stimulating the same region of the cells that we recorded, but we're almost certainly stimulating more cells than the units that from which we recorded. So and the general problem is that for us is that we don't really know who we're activating with our electrical stimulation. We don't know how big the population is. We don't know how, I mean, it's almost certain that we're stimulating more cells than what we recorded, but we don't know how many, we don't know where they go, etc. So what we try to do to lessen the uncertainty that's caused by that is to, we electrically stimulate with very, very low currents. And because what we want to reproduce the condition of activating the same single cells from which we record as well as we can. Now, if it turns out that we can electrically stimulate with significantly, I don't know what that means, but significantly larger currents than are generated, then the significantly larger currents to generate potentials that are larger than the ones that we record from. If that continues to be okay, then there are implications for how the code is spatially distributed in hippocampus. What it means is that if we're recording from a neuron that's part of the memory code for a given object, it means that its neighbors must also be involved in the code for that particular object. And we don't know whether that kind of organization exists in the hippocampus or not. There's some data that that speaks against that being possible. The recordings that have been done in behaving rats to identify spatial fields, it's usually the case that if you're recording from a cell that has a spatial field for the environment that the rat runs around in, if you record from a neighboring cell, whatever that means, but a cell that's probably close by, they can have that cell can exhibit a very different spatial field. But nonetheless, they're both cells are showing spatial fields for that environment. So if you ask people that record from cells involved in spatial fields, they'll say the neighboring cells show very different spatial fields, and that means that the spatial organization of information in hippocampus is what, heterogeneous, and so there shouldn't be any similarity in the firing of neighboring neurons. Well, again, they're both involved in spatial fields for that environment, so it's very difficult to understand, for very different environments, very different sets of conditions, what the topographical organization of hippocampal information even is, let alone what it means. So that's a big I don't know. We think about it a lot, but we don't have a good way of testing it right now. So that's a very good question. It's a very good question.
Okay. Thank you very much, Ted. I think we've kept you busy for long enough right now, and we could probably all use a lunch break, which is exactly what's coming up at this moment. So we here on location are going to have a little break at 1 o'clock. And in the meantime, the live stream is going to switch back to some comforting music. And of course, you can still submit questions to the question and answer form. As I mentioned briefly on that form, even if we didn't answer your questions here right now, we'll still go over the questions, and some of them may still be answered after the workshop or later in the workshop. Thank you. Hey guys, just testing. You guys can hear me? Excellent. Yeah, we had to briefly switch to a different laptop since mine is doing some kind of update that Windows decided was necessary. And we're just about to start the panel. And in this panel, we're going to be passing around this laptop to get people to ask questions.
Because I just want to check the one sound setting because I'm only hearing it on my
okay. The audio is also still on. Did you want that muted too?
I don't think so. My headphones. Should be.
Okay, should be coming through. So now I'm going to turn the video back on, and then you're gonna we're on this. So we're gonna go on this topic, correct?
That is right. Yes.
Okay. Okay.
And you're going to start us now? Okay.
So I think Ted might be interested in joining.
Yeah. Okay. Testing. Testing for Javon. Testing.
Okay. Yeah, you're good.
Excellent. Okay. Yeah, maybe I'll walk around a bit, but I'll move this closer. Okay. Looks like we have sort of a circle here. Okay.
I think the best would be you can come here.
Yeah.
Since
I'm connected here.
All right. Sorry for all the dizzy-making moves for people who are watching this online. We now have a session that we titled Kernel and Neuralink, but it should better just be titled the for-profit or startup or alternative space for doing work in the kinds of neurotechnology that are necessary for this whole route towards neuroprosthesis and whole brain emulation. And we thought that the best way to do this right now is to just have a panel discussion. And it so happens that we have quite a few people here in the room. I'm just going to pan this around so the camera can pick up on some of the people that are there. Yeah. So what we're going to do is we're just going to start talking about this topic. And of course, everyone is going to have an opinion or a question or comment, and I'll hand around this laptop as basically the video and audio for people who are speaking. And meanwhile, every once in a while, I'll take a look at the question and answer forum to see if there are questions coming in relevant to this conversation. So let's get started at this point, and I'll just begin with a little more introduction, not much though. Yeah, so while most of the work that we discussed this morning has of course been happening in academic labs and has been going on for decades in various labs and universities throughout the country and throughout the world, there has always been an attempt to also bring this into other areas of effort and of funding. And what we've seen, for example, is that sorry, Keith is saying I'm creating motion sickness, so I should stop moving around. I think it's better to be right here for the microphone. I'll just try not to move around as much. I was wiggling a lot. But yeah, so for instance, the place where we are right now, 3Scan, this company, this is a startup, and it started with a machine, the knife edge scanning microscope, that was originally produced at Texas A&M University in its original form.
And the idea there was to use it to slice brains and to image brains. So this place where we are right now is actually a perfect example of a startup that got involved in this problem of connectomics, brain emulation, neurotechnology. And since then, there have been a few more efforts. Of course, a lot of the efforts we see are sort of tangential. There are companies out there that are doing BCI of some kind where an EEG is involved and try to make consumer products out of that. There's nothing wrong with that, but it's, you know, it doesn't get you whole brain emulation. And then, of course, about 2 years ago, this actually goes back a little further than 2 years. I've been working with the people involved in that for a while longer. There was a sudden emergence of interest from a few well-known or high-profile entrepreneurs who got involved in this question. The most well-known probably is Elon Musk, who, thank you, who felt that, you know, he saw artificial intelligence coming up and that this would change society, would change how we interact with each other and with machines and what this is all going to cause. And he felt that perhaps the way to go, the way to ensure a future for humanity that is as desirable as possible, that it would be important to be able to interact with, merge with, or in some way connect with what's going on there on that machine side of things. And this is what got his interest going into the neurotechnology of, say, neural interfaces, neuroprosthesis. While Brian Johnson, the founder of Kernel, was also interested for some relatively similar reasons, although perhaps not with the same emphasis on what's going on in AI. And from that, without getting into too many details, partly because I'm just not allowed to tell, you know, two companies emerged. And they're both still active. They've had about 2 years to do something at this point, which again, I can't talk too much about because products are not things we're supposed to talk about. And we're kind of wondering, of course, how, you know, generally speaking, looking at these as models, looking at 3scan as well, and other endeavors like that, where does this for-profit space fit into the whole? What works? What advantages do they have in some ways over, you know, working in a lab, working in academia, looking for grant funding and that sort of thing? And what kind of hurdles do they also experience? What sort of parts of the problem are really well suited to work in a for-profit environment versus which parts are not? And I'm sure we all have a lot of opinions about that, but I've said enough for the moment. Before we make this a whole, you know, say, audience-wide discussion, I want to just hand this over to Diana Deca for a moment so that she can say a few words, and maybe you can introduce yourself while you're at it.
Yes, hi, thanks very much, Randall. It's a really nice introduction. So, hi, my name is Diana Deca. I'm a postdoc at USC, and I currently work on flies. Yeah, sorry. And I'm trying to find out how a neuron works. In my PhD and my previous postdoc, I've been patching a lot of neurons in the mouse brain and looking at spine and dendrite inputs just to see how all these thousands of inputs into one neuron gather into one output. So this is my focus. But at the same time, I've been very interested in neuroprosthetics in a more philosophical sense because I'm interested in the concept of whole brain emulation. My background is also in philosophy, and I wanted to know whether it's possible for us to one day make perfect copies of ourselves that don't just correspond behaviorally to what our biological cells do, but also are based on a causal understanding of the inner workings of our brains. And I think, you know, there was, until now we discussed all these startups that are getting into neuroscience, but I think one of the questions is, what is the goal of neuroscience? Or are there several? Is the goal to rebuild the human brain? Is it to understand one process and rebuild that, and so on and so forth? It is not entirely clear to me, for example, with Kernel and Neuralink, what their end goals are. I mean, I saw some interesting ideas there. For example, with Neuralink, it seems to be, the wish seems to be to somehow connect the human brain to computers or AIs, but it is not entirely clear to me how that would happen. And if it would happen, what would be the next step? Let's say that if we connect our brains to machines, it just means that there would be exchanges, there would be voltage changes in our brains that somehow would correspond to voltage changes in devices. That's maybe something we want to explore. But yes, I suppose all these have been goals in neuroscience and academic neuroscience in general. They might have been parsed into smaller questions just for the sake of funding. And now I think we have the opportunity to rethink the big questions in neuroscience and whether there is a model that can answer these questions. Anyone?
Yeah. So let's just open the floor to all now. I'm sure somebody has a comment or question related to startups in neuroscience. Anyone want to go next? Okay. Cameraman rolls closer.
Yeah. Okay. So it's just a comment. I mean, it's not obvious to me what the goal of a startup would be in area like neuroscience or partial brain emulation. Certainly, I mean, the most obvious one is most obvious goal would be prosthesis, something that's a medical device. Doesn't have to be a prosthesis, but something that's a medical device that could be used by some part of the medical community for a particular patient population, and the endpoint to make money, to help the patients, of course, but to do that and make money. That's sort of an obvious goal, though it's not, and there are some good examples of that already. But beyond that, it's not clear to me what the goal is, and I think I haven't seen it be defined well. In other words, I can imagine that someone has an idea that by studying the brain in a particular way and by developing brain-computer interfaces, that there can be an avenue, a path for understanding brain computation better. And we all at least have, many of us have the belief that brain computation is better than machine computation, and so that we would be able to improve machine computation beyond its current borders. Beyond that kind of a general statement, I'm not sure how we move from that general statement to exactly how to work that path. And I don't mean to be critical of the companies, of any companies that have gotten involved, because I don't have a good idea either. I think it's very, very difficult to see how studying even human brain computation could be used to improve machine computation. So, I mean, I would enjoy seeing a concept like that become well developed and a path from current status to future status become outlined. I think that would be, even if it were just an example, not something that someone did, but just as an example, what would that look like, and how might we do it? And it'd be incredibly interesting just to see an example be explored. So.
Hi. So yeah, to kind of follow up to that, I guess I'll give some concrete examples. So my name is June Axup. I'm scientific director and partner at IndieBio. We're a startup accelerator program here in San Francisco. We funded 4 neurotech companies, and for the purpose of this discussion, I think 2 of them are somewhat relevant. I think there is, obviously with the goal of uploading everything, it's a very lofty goal, and I like how the first speaker talked about it's going to be a 100-year thing, and in 30 years probably someone's going to die, and that knowledge doesn't get transferred. So I think a lot of the startups really need to hit some more near-term goals. So of course with 3Scan, ultimately their technology can be used for uploading purposes and really understanding the connectome and everything, but they are first looking at medical purposes, drug discovery, interfacing with your big pharmas of the world who just really want to look at the tissue, like cancer tissue, microenvironments. So that's a great way to really start and get in the door and get some revenue going while going after more ambitious projects. The two companies that we have that are relatively relevant, one's called Neuro, N-U-R-O.ca, and it's a brain-computer interface. Basically, they have a headset that allows you to move a cursor on a screen. And they've had quite a bit of computation around calling out these signals and making it more efficient. And of course, you know, for most of us, that kind of just sounds like, oh, it's a new entertainment platform or something. But for them, they're going to go after locked-in patients. So ICU patients, people who have stroke and can't move, talk at all. So that's where it's, again, it's a prosthetic, essentially, an extension to allow people to start communicating. And then you can imagine that kind of technology translating into something maybe more sophisticated for the average user in the future. Another one called Troost Neuroimaging. They take EEG data and they do kind of real-time mapping and display of, like, I don't know, the best way to describe it is like the way energy flows in your brain, and it's like instant. So they have like a video of someone seeing a picture, and they see just like whoosh, this whole, like, all these you know, neurons firing in different places. And that particular technology, again, they were aiming for, you know, Alzheimer's researchers or people who are trying to do research and understand the connectome. But what they found is that there are actually other markets that this can be very useful for. For example, they ended up working with a company that works with flavors and fragrances, and they wanted a way to more quantifiably understand how these flavors were affecting people's sensory systems. Right, so so that was a very interesting way to carve out a technology that seems to be just in the research field into industry and creating value for other industries that might not originally be using this kind of technology. So I think there are a lot of stepping stones all towards building that big goal, and I think we need to really focus on what those stepping stones are, because those technologies that are developed will be able to all come to fruition in the future too.
So my name is Brian. So expanding on that, in terms of some of the near-term goals, you know, you mentioned the was it Troost? Is it like fMRI type?
It's MRI. Oh, sorry, it's EEG.
Oh, EEG signal. So that there's I know there's Open Water.
That's MRI.
Yeah, that they're doing they're working on a wearable fMRI, so more diagnostic approach where it's not just for brain, but it could be for any kind of tissue, right? So more in terms of diagnostic and in the health space. So that's like a near-term solution. But you know, ultimately, I think their aim is also not just for reading neural activity, but also being able to use light, I think, to be able to stimulate as well. So having both read and write. And then, so diagnostics, health is probably the obvious one, but then also communication, which you also hinted at in terms of people are not capable of being able to communicate with the outside world, but also maybe expand upon that as a more general communication device. Options. I think Facebook they were touted to be working on something like that, but I'm not sure where that is, or if anybody else is thinking along those lines. So, you know, to think about it more generically, I guess, you know, there's the health diagnostic aspect, there's the communication aspect, and I don't know if anyone had any other ideas in terms of like what other you know, categories of you know, near-term goals that would be feasible for everyone.
So yeah, I think that's a really good point that there can be near-term goals that can be addressed by smaller startups, which will then gather data, and then this will make the problem a bit smaller overall. So instead of having a 20 or a 50-year timeline, it would be a 15-year or so forth, assuming this can all come together and work. This also brings me to another issue that I would like to fit in this, which is how do we distinguish between techniques and experimental methods of data acquisition? What are the good ones and what are the bad ones? Because, you know, in academic neuroscience, this is very easy to discuss. I'm wondering how to do this in a startup setting. And I can give an example. Hawaii, I think, and other companies try to develop a VR that is more immersive. And one of the issues is that if I would now get up and start walking, I would have some sense of gravity and direction. And that's because of the vestibular system inside my inner ear is telling me I'm walking straight now, or I'm bending, or whatever. And there is a way to stimulate our vestibular system with small electrical shocks that would give us the impression that in a VR, we would, for example, move like this. However, the vestibular system is a bit complex, and stimulating certain parts can have a certain effect, and stimulating the whole thing at the same time can have an effect which is not fully understood. So what is the correct way? What are the right experiments to do if, say, we wanted to have more immersive VR? And at the same time, different companies went for, of course, the non-invasive version of it, which is easier. But then as a neuroscientist, I have to ask the question, is this enough? Is this data conclusive? And is this helping neuroscience as a whole? I do not know the answer. It might be yes, but I just wanted to add that as a thing. In the startup world, how can we distinguish between techniques and methods? And also, I don't want to make this too long, but I was thinking about the philosophical premises of emulation and neuroscience in general. What is it again that we're aiming at, and what Professor Berger mentioned? What is the plan, the philosophical plan here for what is to be achieved in 20 years by connecting the human brain with AI or computers? I was thinking one of them might be, it's just a guess of the background of the people involved in this, but it's the fact that technology appeared rather late in the process of human evolution. And I think, you know, some people believe that the human brain has developed for a very long time to make us very good predators, which is shown in our behavior on this planet. So the issue is, I guess, there might be a need for the human brain to be connected somehow to the technology it creates, just to provide it a quick jump, a quick evolutionary leap, which some people believe in the long run might be necessary. But this is just one possibility. I don't know why the others are, and I would love to hear your opinions about why do you do this? I'm sure you all do very interesting projects. What motivated you to do what you do?
So I'm going to just jump in for a second. That's a great topic, and I'd love to have people answer that. I just want to take a quick peek at the forum here to see what's coming in out there. And I also had a very small comment to make myself. Let's see. Not used to this laptop, so I don't quite know how to make this scroll down properly. Okay. What do we have that is related to conversation right now? Oh, I hate that that scroll bar disappears when you're... I'm not a Mac user. I think that judging by what I see here, we can just continue in the audience right now. Yeah, I from what I've been hearing just now, sort of my summary comment so far is it seems like there's no obvious reason why a startup would be basing what they're doing around helping neuroscience as much as possible. That's one really important thing. So when we ask, how does this help neuroscience, what this company is doing? Well, they have no particular reason to want to help neuroscience as such, even if the original reason why, say, the founder of the company got into that, because in the end, it still has to operate as a startup, can't make that the #1 priority. And that leaves then kind of the selection of what you're working on, because it seems to be the type of thing you're trying to build, the product you're making, whether that is a suitable sort of thing that intersects with neuroscience, but also is something you do in a startup. So, you know, here, 3Scan works because It's building a technology that is a tool that you can apply to brains, but you can also apply to other things, so they were able to find themselves a market. Locked-in patients, you mentioned, as a target group for some kinds of technologies. That's an interesting one because, you know, that comes up a lot. Locked-in patients are patients who love to work with researchers on potential ways to get them a little bit out of that box. But then, you know, you run into problems like how many locked-in patients are there and how big is that market and how much money are you going to get from insurance companies for this sort of thing. So there's so many issues there. Anyhow, that was just my summary. Let's continue with whomever. Yeah.
Hi, my name is Spencer, and I work not in bio, but in artificial intelligence, which is closely related. A lot of the problems that I've had to work on over the last few years have been problems of classification and detection using artificial intelligence. And right now, the best ways that we have to approach those problems are to do kind of a naive analysis where we just have like a deep neural learning network where you feed the data in and we come up with results. But one thing about human consciousness and human observation abilities is that we can detect anomalies and create classification schemes really quickly, and that those can learn and change and develop. And so one of the ways I think of it is kind of coming from the opposite direction of not how can we build the hardware that gets the mind, like the operating system of the mind, ready to be ingested, but rather how can we understand and anticipate the way that consciousness would plug into a machine by developing networks that make decisions and perform classifications closer to the way that humans perform it, rather than just crunching numbers. And I think that there are tremendous applications there in like computer vision and security, like anomaly detection, and that one really productive avenue is to improve artificial intelligence such that we can anticipate and prepare for the way that a consciousness would need to be linked into a network. And I think that that would provide a lot of fertile ground for plugging in. But I also just wanted to add the caveat that I think it's also important to consider the way that we can call on the state as well as industry to fund some of this research, because I think that, especially in an era where there's dwindling funding for a lot of important projects, that we need to also care deeply about getting, you know, the state involved at a fundamental, like pure research level, but also supporting projects that are in private industry that are going to contribute to the benefit of everyone, which I imagine we would all agree is the reason why we're here.
Can I ask a question? And I'm sorry to. I want to pursue this. So I heard what you said. Can you elaborate a bit more on how what you're doing interacts with the neuroscience part? I mean, I can understand you're developing computational systems that deal with anomaly detection and classification, et cetera. But how are those systems based on the brain, or how do you get your systems and neural systems to be closer together, or what? I'm not sure how that works.
Yeah.
If you can answer that.
Sorry, that was unclear. So I'm not a neuroscientist, so the language that I think of it is regrettably kind of pop neuroscience, or like Malcolm Gladwell has the idea of thin slicing, which is that the way that traditionally I work with artificial intelligence to detect patterns is you feed it a set of data, and then you teach it about the different exceptions to the data, and then it recursively runs those experiments over and over again, and that data set has to be confirmed. And the most advanced artificial intelligence networks that we work with today still require, for classification, a lot of human input to say, this is what this item is, this is what this image depicts, etc. So but the beauty of the way that humans perceive the world and that human cognition operates is that from a very limited data set, you can get extremely accurate classification models that rely on not very much information. So there's a lot of inference that human consciousness is able to do because there's so much context, like external context available, that if we can determine the way that you can make smarter inferences from smaller data sets, then it provides a really good opportunity to start figuring out where the base level mechanics of consciousness operate, so you can plug back into them.
Thank you very much. Yeah, thank you.
I'm just going to dive into continuing the conversation. It's interesting, of course, with humans, we don't need very much labeled data, as machine learning people put it, to understand a new phenomenon that we observe in the world. But if you look at children and the way that they get parented, they do get read to from picture storybooks and continually asked to correctly identify all the objects on the page and in the story. And this goes on for years of childhood. Now, it's probably the case that the amount of labeled training data that a baby or a child gets is still way less than we currently need to train convolutional neural networks to do similar vision tasks. So as that field progresses, it's going to get better at learning from data more efficiently. But there's already an analogy there between what's happening in these deep neural networks and humans. So the thing, I don't work in this space directly, and so it's fun to hear people speculating about what commercial projects in neural interfaces might look like. And so just throwing ideas in from the outside, one thing that's really striking is that you need FDA approval. If you want to drop networks of electrodes into people's brains, you need FDA approval for that. And so insofar as companies are moving into this space, they're going to have to figure out a way to be allowed to do this with human brains, and therefore necessarily to do it with some medical application in mind at first. If you could get past that obstacle, if you could demonstrate that such a technology was practical and safe, and I think that's like a long way down the road, you can imagine this being an enormously profitable business, right? It becomes a platform business. You're like, oh, you can install apps in your brain, and those apps can do whatever you want, right? Like depending on which part of the, like if it's your hippocampus or your visual cortex or something else, then suddenly you can have an app that like helps you be more mindful of something, or like helps you notice patterns in your own behavior. It aids your memory, not in a like repair sense, but like we all want better memory. We want our memory between our brain to be cross-indexed with our computer. We have some ability to do that right now through like typing into a search box. But it's obvious that would be a hugely valuable domain to be able to enter commercially. And so there probably are paths for companies that start as startups, find a medical application, and then get investment and value from the speculative like field that they're trying to reach beyond that.
Just really quickly, I think this makes a nice circle of the discussion and kind of brings us back to the title of the discussion, which is related to kernel and Neuralink. And what he just mentioned, that there is a threshold for having an implant that can safely connect with the brain and has passed FDA approval and allows us to do all sorts of great things. I think this is the stepping stone, probably the biggest milestone that many of these big startups are going for. But that usually takes 10 to 15 years in itself. And that's actually the issue. What kind of model will support that kind of project? Because it is very ambitious, but it can also be extremely profitable once it's there. Like the number of applications coming out of this is You want to say?
There's an interesting question on the live chat.
Oh, there is? Yeah, I didn't know where it was. I think Javon just pointed it out as well. Yeah, sure, go ahead. Why don't you take this then and read it out?
Okay, this is from PJ Manny, and the question is, how do the priorities of proprietary IP, ROI, and market share with startup companies contrast with university research, which is by nature collaborative? What is the likelihood as financing moves to VC and traditional investment, which are less likely to share? So yeah, in essence, the combat between academia and industry. Clearly, Kernel and Neuralink are not going to be publishing peer-reviewed science, perhaps, for their initial products. But I mean, perhaps they'll do FDA approval, but they're not going to disclose the meat and potatoes.
Why don't we let Ted talk about this a little bit, because not just for Kernel and Neuralink, but you've had a lot of experience previously working, either trying to spin off companies or working with companies, and how you work with the IP and stuff like that.
Yes, So we've had experience with well, I've had experience with Kernel, and we've also tried to we have spun out technologies that we've worked on that have been in the area of signal identification, and that's been then used for perimeter detection and perimeter security. So these were they started as basic issues in trying to understand how incorporating aspects of biological neural networks into otherwise artificial neural networks could make those artificial neural networks or biologically realistic neural networks better at detecting particular signals. It was starting with speech and then non-speech sounds, etc. And that collection of those things could then be used for perimeter security. And we found that that it was at times difficult to develop the right kind of relationship between the commercial entity and the university, and we definitely had to. I mean, this was work that was started at a university, and it was funded through grants and SBIRs, so to the company and to the university. So we had a lot of things we had to worry about in integrating the company efforts and the university efforts. But we found that as long as we were careful about filing for patents and filing for patents at the appropriate time, and then coordinating the publication with the patent submissions, and as long as we kept up that relationship and the timing of those things, so that they were done in the best interests of the company and in the best interests of the lab, that we could make that work. It just required understanding of the relationship between about the demands of IP and the demands of filing for IP, and then coordinating the timing between the company and the university. So conversation mattered a lot, and coordinating efforts mattered a lot. But otherwise, if you didn't pay attention to those things, there was a lot of friction. And the biggest problem that we had, we've also developed, we spun off a mathematical model of a synapse that was extremely useful for better academic and scientific understanding of glutamatergic synaptic transmission. And that was then spun off to a set of investors who wanted to develop new drugs and use the model to develop new agents that could act at those receptors and channels, which was, as most people know, is very important, because if you try to do that experimentally, the costs are astronomical, and it takes a very, very long time. So if you can short circuit that by having a mathematical model of a synapse, at least get the first few steps done in terms of identifying in terms of identifying a chemical structure that could act at that synapse, then you were at least ahead of the ballgame. That also worked as long as we kept up the timing. But a real problem that remains a problem for both of those companies was just having, you worry about having the funds to support the personnel and the surroundings, the computational systems and the computational oomph to do all the work. It's mostly personnel you have to worry about. But then to come up with the money to pay the licensing fees to the university and to keep up those licensing fees, that was a burden. Without having something that would generate income, then the investors had to provide both the support for the personnel and had to provide the funding to keep the university happy about the relationship, and that was not easy. I mean, it's the same old question about generating money and managing funds, but that was a difficulty.
The university wouldn't accept equity?
In one of those two cases, they had equity in the company as well. So it was I guess you could say it was a little too greedy on the part of the university to want both equity and payments on time. But that fell behind in both of those two issues. Anybody else?
Sure. I mean, one thing that I haven't heard talked about before is in this discussion is getting into the brain, right? So without cracking open people's craniums and drilling and doing whatnot, there's something to be said. The Jason Silva, he talks about the skin bag bias, and we shouldn't consider our cell phones to be not part of us. That they're extensions of our brain and they're already like memory prosthesis. I think that's taking a little bit too much poetic license. What we're really trying to get to is like getting into deep communication with our brains and listening to the symphony of the brain from inside the cranium, not outside the cranium. And so the question I'd like to kind of put on the table is, how do you get inside the brain without in a mass market way, without doing mass surgeries on people and whatnot? And the only one I've heard thus far, that may be viable is going through the nose, because apparently olfactory neurons are not bound. Yeah. So you can infect olfactory neurons, and they seem to be not as protected by the blood-brain barrier. I don't know if this is true or not, but if you guys have any other ideas of, are we going to go through the nervous system? Are we going to do Ray Kurzweilian, nanobots and whatnot? What are your guys' thoughts on that?
Yeah. So we're almost at the end of this panel, actually. So I'm not going to be able to get into that in depth, but it's an incredibly fascinating topic. Maybe we can get into it at the end of the workshop or something. But just to give a quick sort of summary of my thinking about it. The problem today is that, of course, we want at the same time to build products that are appealing to the mass market because that's where the money is. But we also have this problem of breaking the skin, having to get in there if you want to build something that's really a very high bandwidth, high resolution interface, and that can go in both directions, that can read and that can write. And so, yeah, sure, some people are looking for technological solutions that would be able to do that, that would somehow be able to create that connection without having to dig into the brain. But I would say that all of those, without mentioning anyone in particular, are right now still very speculative and sort of serendipitous things. They might work, they might not work. We don't know how good they're going to be, both for reading and writing. And so, but on the other side, the things we use today, electrodes, it's kind of like the steady path, the path where we kind of know what's happening. We know that they're becoming smaller and more flexible so they don't cause inflammation in the brain, and the surgical procedures are getting better. So you could say, sure, this looks risky, but laser eye surgery could also seem risky, and plastic surgery can seem risky. So it's a question of how much risk can you accept? How good is the technology? How good is the procedure? That's the other side of it. So instead of the serendipitous projects, there's also the steady moving forward. How many thousands of sites, recording sites and stimulation sites can you put on one electrode? How many electrodes can you put in at the same time in many different parts of the brain? And what is the surgical procedure like? Both of those paths are worth exploring further. And they get you probably to different results, different types of interfaces you can build, different types of devices. But it's a really complicated topic because, of course, that's where the FDA gets involved right away. And the FDA is now even getting involved with things that we might have called non-invasive, TDCS. But yeah, so that's the other side of the coin. Some things that we think are non-invasive are not necessarily non-harmful just because they're non-invasive. Pump a lot of radiation into the skull is also not a good idea. Okay, so I'm sorry, we can't dig deeper into this right now because we're getting into the next topic. We're going to take a, say, 2 to 5 minute break, even though Sean Mikola is supposed to be on in a moment. Sorry, Sean, I hope this isn't too long for you if you wait for 2 to 5 minutes. I'm going to check my
Yeah, it's fine.
Okay, I'm going to check if my computer's ready to go again, because it was doing a silly update that it should have been doing tomorrow or something. Meantime, let me just put this on mute. Hi, Sean.
Yeah.
Yeah. I just realized the smarter thing to do is for me to just introduce you right here, right now, and then you can get going by sharing your slides and talking, and I'll get my laptop working again while all that's going on. So let's do it this way. So Sean is kind enough to come to us from Japan right now. It's extremely early there. I think it's 7 o'clock in the morning. And yeah, so Sean has just recently joined the National Institute for Physiological Sciences there, and previously worked at the Max Planck Institute. He's been involved in connectomics for a long time and electron microscopy. And so this connects back a bit to what Ken was talking about at the beginning of the day. And here I'll just leave it up to you. If you can share your screen and start, then we're good to go.
So can you see my screen now?
I can. It's probably going to take a second to pop up on the live stream because it's always got a delay of something like 20 to 30 seconds.
Okay.
Yeah, it's up.
Great. So I am excited to be here, and I wanted to thank you, Randall, for putting this together and the other people involved. I mean, it's a great opportunity, I think, and I've learned quite a bit, even though I wasn't able to catch everything, so I had to get some sleep. So I'm going to be talking about high throughput EM and X-ray nano-CT of whole mammalian brains for circuit reconstruction, cellular resolution, connectomics. And at first, the title might seem slightly odd, why I would put together both EM and X-rays in the same talk. But as will become clear shortly, I think there's very good reason to do that. And so we have to begin my talk, I want to go back in time a little bit. If we go back 5 years and we look at the state of the field for volume electron microscopy, there were several landmark papers that were published exploiting all of the spectrum of different methods, EM-based methods for reconstructing neural circuits. So we had serial section EM methods, we had FIBSEM, the focused ion beam scanning electron microscopy, which Ken talked about earlier, and we had serial blackface electron microscopy techniques. And what all of these studies and all these results had in common was that they were using conventional sample preparation for their electron microscopy. And in this type of sample preparation, it always uses this reduced osmium type of stain, which is shown here. So the idea is that you have a mouse brain, you extract a small area of the brain, and you put it into reduced osmium solution. And then there were some variations on this of what you did afterwards. And what I show here is what Ellisman and Deering published as the roto method, or what's commonly known as the roto method, where after reduced osmium, you would then amplify the osmication, if you will, by mixing in TCH, thiocarbohydrazide, and then having a second osmium step, and then some additional staining before embedding in plastic and cutting and imaging. Now, why use reduced osmium? And there's good reason for this. I mean, it's because it gives you very strong membrane contrast, which is essential for if your interest is in reconstructing circuits, which is what these studies were interested in. Now, the problem was when I tried to apply this method to something bigger, like a whole mouse brain, it quickly became evident that it would not work, that the reduced osmium had penetration problems, that it would only penetrate a few hundred microns into a sample. And so you could not use it practically for anything a millimeter in size or bigger. And so this presented a dilemma to me because, of course, I was interested at the time in applying volume EM techniques to mammalian brains, specifically the mouse brain. And so I had to spend a couple of years figuring out how to modify the existing protocol to get it working in larger samples. And I'll sort of just present the result here. This is called BROPA, or this brain-wide reduced osmium staining with pyrogluol enhanced amplification. And the basic idea was to just first examine the reduced osmium step and just explore what I would call an augmented or expanded parameter space. This parameter space included different things that you would add to the solution in order to expect enhanced penetration. So this would include detergents, solvents, various different ions. Different things were looked at in buffers. And after this screen, I found out that formamide was the missing ingredient. And what's interesting is, so formamide is a very small molecule, and if you look at variations on the formamide molecule, like acetamide, which has the addition of just a single carbon, it does not enhance penetration at all. And so there's something very peculiar about formamide. It seems like it solubilizes things that would otherwise precipitate in solution and build up to form this diffusion barrier to further diffusion of the stain. The second thing I did was just modify or replace the TCH that was in the standard protocol, because this was liberating nitrogen gas and causing ultrastructural damage in large samples. And so this was pretty straightforward. I just did a large screen of all small molecular osmophilic molecules that were commercially available. And the best one turned out to be a black and white photographic developer called Pyrogolol, and structure is shown there. And then the rest of it was pretty much the same as the rotostain. And so what was interesting about BROPA is that not only did it work in the mouse brain, but it worked in a variety of small mammalian brains that I looked at. So it worked in the pygmy shrew and also up to the rat. And I didn't explore much beyond the rat. So I can't say how well it scales at this time to larger brains. And on the right-hand side, I'm just showing corresponding EM with a high membrane contrast that is typical of reduced osmium staining. And so, but let's focus a little bit more on the mouse brain and just see whether BROPA is in fact suitable for something like a brain-wide circuit reconstruction. So if we look at the SEM through a full coronal slice through the mouse brain, we would see something like in part A. And we can zoom in on each of the regions, starting from the peripheral, the periphery part of the brain, and just work our way inwards from B to E. And so we see different structures: the cerebral cortex, white matter, the external capsule, striatum, and also some additional areas that aren't shown in part A, such as the cerebellum and reticular formation. And all of these showed high membrane contrast and appeared suitable for reconstructing circuits. So this is just showing a fly through, which I'm sure, based on what I've seen earlier today, will appear choppy to the viewers, but that's okay. It's just showing a fly through the striatum from one of these BROPA brains. And what you can see is a large number of neurites and synapses and quite a few cell bodies, presumably or mostly medium spiny neurons that are the dominant population in the striatum. And so, of course, I wanted to make this a bit more quantitative about the traceability and synapse detection. And so I just went through and randomly selected neurites from different regions of the brain. And so one of these neurites is shown in part A that has been fully segmented along with all of the incident boutons made onto it. And I assigned, starting at seed locations, I assigned this neurite to different people to trace. So this is shown in F actually. So 20 people redundantly traced it. And so based on this redundant tracing, you can estimate error-free path lengths of greater than 1 millimeter in the 2 gray regions examined, and greater than 70 millimeters in the region of white matter that was examined. And part E is just showing how consistent people were able to identify synapses, which is greater than 90% for threefold identification. And so all of this looked good and seemed what we wanted for doing, for then taking the next step of imaging the entire brain. But before I get to that, I want to just talk about looking at larger pieces of how you can actually trace processes through very long stretches of the mouse brain in this case. So this is a corticostriatal stack. It's 1.5 millimeters high and a few hundred microns in width. And so basically you could trace neurites through almost the full extent of the stack. Now, the stack was also taken at coarser resolution, so we couldn't see synapses. It was basically just limited to tracing neurites and identifying cell bodies. So with regard to, now that we have a brain, we want to be able to cut it up and image it using high throughput EM. there are different ways we can go. I mentioned 3 previously. FibSum is ruled out because the limited depth that you can actually mill away before problems occur, which is limited to, I think, 100 microns or less. And so that left the serial block face approach and the serial sectioning approach. Now, with the serial block face approach, I was working for quite some time with others trying to get that to work. Ultimately, or currently, it is not working. However, about 1.5 years ago, I to focus my attention on the serial sectioning approach. It seemed like it might be better suited for cutting through large samples. And so I purchased one of these atom tome machines. So this atom tome, it's this box on the left-hand side. This was based on a design that Ken Hayworth developed some years back in Jeff Lichtman's group at Harvard. And they've been developing this technique extensively. And what it is basically a conveyor belt for picking up sections that are produced automatically from commercial ultramicrotome, which is shown on the right-hand side. Now, when I first attempted this, I explained to the people selling me these machines and other ultramicrotomists that what I was intending to do, what I was intending to use their machines for. And the old school ultramicrotomists were just giving me warnings that something terrible would happen if I tried to actually cut through a centimeter size sample with these ultramicrotomes. And so I sort of, this didn't discourage me at all, but actually got me quite curious to find out what would happen if I seriously attempted this. So of course I went ahead and did this. And so what I show here in the sample that's in the sample holder is one of these whole mouse brain samples. And if you go through and just start cutting, so this is through the olfactory bulbs of a mouse brain sample. Actually, it looks to be just a slab and not the whole brain in this case. So this is not the full size of the mouse brain. This is about maybe 4 millimeters by 2 millimeters area that it's cutting through. But you should be able to see both olfactory bulbs in these sections that are coming off. The sections are 50 nanometers in this case. Now, the much more interesting thing is what happens when we try cutting through an entire brain. And this is shown here. So this is a full mouse brain cut at the mid-sagittal plane. So this is about 15 millimeters high by about 7.5 millimeters wide, using an 8 millimeter diamond knife to do the cutting. And you can see that the cutting is also much slower here. And you can also see the sections on the conveyor belt on the atom. So they have a slightly purplish color. And you can see that they're full sagittal mouse brain sections that are being cut, in this case, at 80 nanometer thickness. So this was the first time that ultra-thin sections had been cut through a full mouse brain. And what I found most remarkable was how easy it was to get this working. And so this made me optimistic that whatever remaining problems there are with regard to section thickness or section quality or number of sections, these are things that would be much easier to solve than trying to get alternative approaches to work. And so I focused my attention almost exclusively on the serial sectioning approach. And let's see. So yeah, what are exactly, why do we want a brain on tape in the first place? And I've tried to explain the main advantages here. So in the images, before I get to the advantages, so the image on the left-hand side is just a custom tape collecting machine that I built using aluminum foil as the tape substrate. And you can see a whole brain used in the sample holder. In the upper middle image is just a collection of these brain sections. I ended up cutting about 6,000 consecutive sections at 80 nanometers, which is about 5% of the mouse brain. And I collected all of those on tape. And on the upper right is just what it's just explaining that a single mouse brain on tape would be about 1 kilometer. It would actually be closer to 2 kilometers if you're cutting at like 40 to 50 nanometer thickness, which is actually the range that I'm aiming for. And I have 500 meters of aluminum foil tape, which is something like 12 microns thick, shown up there. So you would need maybe anywhere from 2 to 4 of those rolls to have the whole mouse brain on it for imaging. Now, why do we want the brain on tape? So I list 3 advantages. Advantages relative to other volume EM techniques, such as FIB and the serial block phase technique. So for instance, it's a non-destructive technique that allows for random access volume EM. So non-destructive, I mean, once the brain is on tape, it's always on tape. You can go back and re-image it at high resolution if need be. If there's some ambiguity you're trying to resolve, it's not a problem. Whereas with FIB and SPEM, once you cut or otherwise vaporize or ablate your sample, it's gone forever. You can never go back to it. If you're going through the data, analyzing it, and you see something that you're unsure about and you want to go back to re-image your stuck, you can't do it. So the fact that the brain is on tape and it's always on tape and allows for repeated imaging is really a big selling point. And the second point is the random access. If you want to go from section 1, for whatever reason, jump to section 1001, there's not a problem accessing that section, whereas in the destructive techniques, you would have to actually cut through all the intervening sections before, and presumably image them before getting to section 1001. The random access means that you don't need to image you don't need to image everything. You can just go to the regions that you're interested in, assuming the sections are properly indexed on the tape, which is pretty straightforward to do. The second advantage is that there are 2 modes of imaging the brain on tape. One is to just do automated bulk imaging of everything on the tape using high throughput EM. You can do that by either using multi-SEM, these multi-beam SEMs, or you can just cut the tape up in a bunch of strips and send each one off to a bunch of single beam or multi-SEMs if you want to increase the throughput. But the other mode is just to do sparse imaging of the sections for just efficient reconstructions of just certain subcircuits that you're interested in, which is compatible with the lower throughput single beam EM. You don't have to image the entire tape to get at brain-wide circuits. And the final thing is this nanoscale molecular interrogation. So I'm thinking specifically of immuno-EM labeling, not so much of mass spec, because this mainly tells you elemental analysis and there are other problems. Now with immuno-EM, there's loss of immunoreactivity, so this is something that would have to be troubleshot. I mean, the preparation for EM causes lots of problems. So this is just showing some of the results for the mouse whole brain serial section EM that I've attained so far. So this is in part A is the micro CT, and with the slice at about the region that I was collecting the sections from. In part B is just showing 3 consecutive sections. You can see section quality needs still considerable work. I mean, there are wrinkles in that first section. There's chatter in the second one. I mean, chatter is one of the big issues that has to be solved, but I have some tricks up my sleeve for that one. And then in C and D is just showing higher resolution EM. And so with this being said, one final thing is that, of course, the brain on tape and all of these brains are compatible with the multi-SEM. So this is actually showing block-face imaging with a 91B multi-SEM. The contrast is actually much higher with thin sections, like as you would have for the brain on tape. Now, I've sort of gone through the EM. I don't have that much time, so I want to just quickly say something about the nano CT. So I promise to explain why there's the relation between the two. If you have a sample that is suitable for EM, meaning it has high metal content in it, it means that X-rays will also interact with those metals, and you can use such a sample for X-rays without any problem. And in fact, EM samples are very good samples for X-ray nano CT. So I'm going to show you some results from my collaboration with Bobby Kastleri, Raphael Vescovi, and others at the Advanced Photon Source at Argonne National Labs. And this is showing pygmy shrew, just a single slice about midway through the brain. I'm not sure what resolution you guys can see on your end, but what I see is a bunch of little dots, which are cell bodies. Now, if you zoom in on that yellow box, you would see, and just do a fly through of about 1,000 sections, you would see this. So the resolution here is submicron. It really is nanoscale. It's closer to half a micron. And so this is showing cortex in the upper part, and then at the very bottom right, you see the external capsule, the white matter coming into view. Now, that's fine, but what's interesting is, so this is showing if you use a lookup table and just color code pixels and do a projection type image. So this is just identifying the cell bodies in blue and the vasculature in red. And again, this is not using any sort of machine learning or there's nothing here except using a lookup table to color code things according to the grayscale intensity level. And so this is promising. This is interesting, but the most interesting part of it is shown here. The fact that if you look carefully at this data in the white matter, if you rotate that stack a little bit and look at it so the external capsule is at you, is facing you, and you adjust your lookup table correctly, you see something like this. And now you see the telltale morphology of blood vessels in the upper part. But if you look at the lower part, you can see bundles of tube-like structures, which I think can only be one thing. And those are large caliber axons, the vast majority of which would be myelinated axons. And so this is what's currently being investigated now. The resolution is certainly there to permit identification of large axons. And if you can see and trace large axons throughout these entire brains, then while you might not have the complete synaptic level circuit map, what you would have are complete projectomes for various mammalian brains. And this is, I think, very exciting. So I would like to conclude by saying that there are two methods that I presented that appear suitable for reconstructing brain-wide circuits: serial section EM and X-ray nano-CT. For the serial section EM, we've been developing the whole brain on tape for non-destructive random access EM, and X-ray nano-CT is a promising alternative, but resolution needs to improve by 10 to 20x before synapse resolution, before synapse level circuit reconstructions are possible. Nonetheless, the X-ray nano-CT has the possibility, if you can get the 10x improvement in resolution, of really, I think, revolutionizing the whole cellular connectomics or cellular resolution connectomics, because it is a very fast method. Like for the whole brain on tape, if you cut it up and you talk about imaging times using a multisem, the optimistic numbers come out to 1.5 years. X-ray nano-CT, the actual scanning itself with the X-rays is a matter of minutes. And then what you have are a series of projections that you have to use some sort of algorithm to back project. And this is something that you run on a cluster that might take a few weeks. And so we're talking about a few weeks for nanoscale whole brain data versus over a year if you go the EM route. And so if nano-CT can push the resolution a little bit, it's just going to make cellular resolution connectomics commonplace. It's going to make it easy. And so I want to just acknowledge several people involved, several groups that I've had the pleasure of working with and collaborating with, located in across Germany, Japan, and the US. I'm not going to go into all the details. Yeah, I mean, I think what excites me is that all the pieces are in place to actually do a connectomic attack on a small mammalian brain. And so with proper resource allocation, I mean, this is something that could be done starting today in 5 years' time, starting with the cutting, the imaging, the annotation, and the complete circuit reconstruction could all be done in 5 years. So thank you very much for your attention, and I'm happy to take any questions. And the complete circuit reconstruction could all be done in 5 years. So thank you very much for your attention, and I'm happy to take any questions.
Yeah, thank you very much, Sean. First of all, it's time for a little applause. Yeah, thanks. This was great. We're going to have time for maybe 2 or 3 questions because we have a rather full schedule coming right up. Sean, you might want to stop sharing your screen because it looks like you went on infinite there. Infinite depth. Okay. Yeah, let me just go to the audience here first, and then I already see something that popped up on the question and answer forum. So audience here, questions? I know this is stuff we've already talked about this morning, so maybe, yeah, Peter?
I have a dumb question. Why is when you're doing whole mouse brains, why is that knife so much slower? And if it is so much slower, why not just take slabs of the brain and do it slab by slab?
Okay, so Peter is asking, when you're doing whole mouse brains, why is the knife so much slower? And if it is so much slower, why not just take whole brain slabs, or what was the, or multiple brain slabs?
So the reason for the slowness is because the cutting force is proportional to the edge of the sample that it's cutting through. So these mouse brain samples have anywhere from 20 to 40 times the cutting force is required for them versus the standard small samples that you can normally cut faster through. And so you have to slow down the cutting to compensate for that. And if you don't, what happens is chatter problems. So it's the slow cutting speed is because the sample is bigger, and the only way, and you want to minimize the onset and the propagation of chatter throughout the sample. Now, regarding why not cut it up into slabs, I think you mean like a hot knife approach or something. But if you do something like hot knife, then then you're sort of tied down to using an ion beam milling approach, and this is something which, yes, it would be possible, but it still has to be demonstrated that it's possible. And with the ion beam milling, it has not yet been demonstrated that you can actually do that through thick slabs through the entire mouse brain. Demonstrated, then yes, that would be interesting to do that, to just cut through slabs through the mouse brain at 10 microns and have each of those ion beam imaged. Not with fib, but with one of these broad ion beams, like Ken was talking about. Not with fib, but with one of these broad ion beams, like Ken was talking about.
Thank you. Are there additional questions from the audience here?
Question is, how do you adhere the sections to the tape? Do they naturally adhere, or?
Okay. Yeah, Daniel is asking, how do you adhere the sections to the tape? Or do they just naturally adhere, or is there some kind of glue?
They naturally adhere. So, there is some relation. There seems to be a preference for hydrophilic substrates for the adherence. So if you have a hydrophilic substrate, it tends to be more wrinkle-free. So with the pure hydrophobic substrates, I don't think I have tested those, but with every tape substrate I've tested, the sections stick without a problem. And these include metal foils, various PET and Kapton, and ones with and without different types of coatings on them. The adherence has never been a problem.
Sorry, I was still on mute. I'm going to give you a question now that came in through the online forum, and it's a little bit different. But I guess, let's address this one once. I guess you're going to be talking for everyone who's doing connectomics of some kind. Leslie Seymour asks, is there any similar work for mapping the second, in brackets, gut brain? A more manageable size, like 100,000 neurons. B, a huge impact on the affective self. C, 90% of DNA in the body is microbiome. D, huge recent interest in commercialization and investments. E, ease of impact, e.g. diet change. So basically, is anything happening with the gut rather than the brain as such?
With EM, not that I'm aware of. With X-rays, yes, there is some work there, but I don't know people doing volume EM that are looking at the gut.
All right. So thank you very much, Sean. You've already stopped sharing your screen, so that's great. We're going to have to move on to the next section now. And in this session, that's topic C, the tech review, I'm actually handing off the moderator responsibility to our research director, Alicia Smallwood. And I think she'll be taking over from here.
Hello, can you hear me? Great. So, like Randall said, my name is Alicia Smallwood, and I'm the research director at Carboncopies. I'm excited to moderate our next session, Meet the Carboncopies Technology Review, where my team and I will share with you our plans for writing the technical review and get your input. So I just wanted to start with a brief introduction before we move to Abul Fazl's talk about what the technical review is and why we are writing it. When we first took on the endeavor to update the 2008 roadmap, one of the first things we did was review criticism of that roadmap, and we tried to address it so we could try to address it in our publication. One of these major criticisms was that it was more of a technical review rather than a roadmap. So we decided as a first step to updating the roadmap, we should do a technical review. So this technical review will cover topics like neurotechnology, artificial intelligence, brain mapping, artificial substrates, computational brain models, and brain theories. We actually could use someone to help us write the artificial intelligence section, so let me know if you're interested. But our goal with this technical review is to present an unbiased overview of these topics and the relationship with whole brain emulation. And this will be distinct from what we intend to do with the roadmap, where we'll want to detail short and long-term goals, key problems and uncertainties, and that would require a bit more opinion. So with these presentations, each of our writers will be presenting an overview of what they will cover in their respective sections. These presentations will be relatively short to allow for plenty of Q&A time. We're hoping to get a lot of your input on what we should be writing about. So without further ado, I'd like to introduce our first speaker, Abul Fazl Al-Abor, who will be leading our effort on the artificial brain section. Abul Fazl received his pharmacy doctorate from Shiraz University of Medical Sciences in Iran and is currently a graduate student in the Newman Memory Lab at Indiana University, where he studies neural coding and information transfer mechanisms in the hippocampus and related structures.
Okay, do you guys have my voice right now?
Yes, I can hear you.
Okay, nice. Okay. I would like to first thank Alicia for the introduction, and thank you guys for joining us.
Yes, we can hear you now.
Okay, nice. Okay. And also, thank you guys for joining us for this part of the technical review presentation. And in this part, I'm going to talk about hardware implementations for brain emulation, and we'll shortly discuss the theoretical considerations for such purpose, and then we'll review some current status of the this technology, and then in a few minutes we talk about the future directions. So let's first start with the question: what is brain emulation, and what's the difference between brain emulation and a brain simulation? So let's imagine that we have a system called System A, and we give the system an input, and we give a system an input, and system processes that input and gives us an output as a result. And then we want to simulate the system. We give the system the simulation the same input, and we expect the simulation to generate a fairly similar output for us with a little bit of tolerance for error. And but in an emulation, we would like the system to process the same input and give us the same output, and map the same input-output relationship, and also considers the internal dynamics of a system. But there's a question in here, and the question is that how much of detail and how much resolution do we need in that internal dynamics? Do we need to recreate the system down to the level of atoms and molecules in that system? Or in case of the brain, do we need to replicate all the neurons and all the dendrites and dendritic spines, or we need to use the circle level analysis, or maybe a systems level replication of the internal dynamics? This is a question that we need to take into account and consider when we want to address this issue. And a second theoretical consideration that I would like to first talk about in here is the enormous computational demands of a whole brain emulation. Let's imagine that we want to emulate the cerebral cortex and not the entire brain, just the cerebral cortex, 17 billion neurons at the neuronal level. As most of you know, if we increase the biological realism of a model, the model becomes computationally expensive. And if we decrease the cost, excuse me, if we decrease the computational cost, we'll have a model that is not biologically realistic. But there's been a solution to this called the simple model proposed by Ishkevich at 2003, excuse me, and in which you can see that equation down. There's a simple equation. There's two dynamical equations coupled together, and it can show a rich repertoire of different dynamics in neurons. And this is the most probably one of the most probable models that we have about neurons. So let's imagine that we want to use this neuron and this model, and this model needs 13,000 operations per second to simulate the neuron at a real time. Let's imagine that we're using this model and we have just 17 billion neurons, which equals almost to the number of neurons that we have in cerebral cortex. And we want to run a simulation. If we want to do so, we need, and if we consider that each neuron has 10,000 synaptic connections, at the end of the day, we will need something like 2.2 exaflops for just simulating the cerebral cortex. And if we consider the neurons that we have in the cerebellum with the same number of connections, we will need 11 exaflops, which means we will need 11 multiplied by 10 to the 18 operations per second. So look at this graph for a better understanding and graphical representation of what we're dealing with right now. So here's the brain. We have something on the order of 11 exaflops and the power consumption of 20 watts. And here's the best supercomputer as of November 2017. So this supercomputer located in China uses 15 megawatts, but it has a computational power of 93 petaflops, which is 3 orders of magnitude smaller than the computational power that we need to simulate the brain. And in terms of power, we'll see there's a significant difference between these two systems. Well, I'm talking about this topic, having this point in mind that it's still debated whether brains are computers or not. So two main points that I wanted to talk about in the theoretical section was that first, there's a difference between emulation and simulation, and depending on how much, depending on our level of abstraction, we would want to design different hardwares. And we need highly power efficient and highly powerful systems to perform the whole brain emulation. These are the two main theoretical considerations that we have to take into account for the whole brain emulation. And let's talk about some partial emulation of neural systems. So previously, we talked about this idea that the brain needs an enormous amount of computation, and it's so much power efficient. And this is not what we have in our general purpose supercomputers. So is there a solution for this? People in the field of neuromorphic engineering and people who had been thinking about this problem came up with this idea that instead of using general purpose CPUs, let's make hardwares that can only perform the neural simulation or neural computation, and then that can probably solve the problem or address the problem in a way. So let's talk about one of the good examples of this effort, the NeuroGrid. NeuroGrid, developed by Kobana Bowen at Stanford University, is a neuromorphic chip that can simulate neurons, almost 1 million neurons with 1 billion synapses in real time, and it's 100,000 times faster than a Blue Gene supercomputer, and it only consumes 5 watts of power. As you can see in the picture, there are 16 neural cores that each of them have 65,000 neurons. And this model had been applied to modeling retina. And another point that is worth considering about this model is that there's a trade-off between the neuronal detail and the number of neurons that we have. For example, for example, if we want to have 1 million neurons, we can have 1 million simple neurons with just one equation and one compartment each, which means you're thinking about neurons as if they're just membrane patches. And or we can have 100,000 neurons, and each neuron can have 10 compartments, and we can consider the morphology and dendrites of a neuron. But it comes with the cost of losing the number of neurons that we are doing the simulation of. So this is a graphical representation of the NeuroGrid, but although it's not complete, but it gives us an idea of where this neuromorphic system plays. So upon our previous discussion about the power consumption, so if we have this supercomputer in here and also the brain in here, NeuroGrid lands somewhere around here. Let's imagine, because NeuroGrid uses 5 watts of power to simulate 1 million neurons. So if we want to simulate 1 billion neurons or 86 billion neurons, it's probably need an order of tens of kilowatts of power to run the entire simulation. While we have seen that we need to use 15 megawatts of power for the simulation on a general purpose CPU and supercomputer. On the other hand, if we compare the number of neurons and number of synapses that we have, so in the brain we have 86 billion neurons, 150 trillion synapses, and in the NeuroGrid, what we have achieved is something around 1 million neurons and 1 billion synapses. But note that the axes are not a scale. It's just a conceptual comparison between two systems. So let's go to the Europe, where people are developing neuromorphic hardware by support of the Human Brain Project. So this project called Brain Skills, mostly people from Heidelberg, Germany are participating in that project, but it's supported by the Human Brain Project. And in 2012, there has been development of a wafer, final steps, and it was ready. And in that wafer that contains 196,000 neurons with 15 million synapses in total had been implemented on that wafer. And the thing that that is true about, and it's good about this wafer, is that it's programmable, and the programmer can change the neural detail as just like the NeuroGrid neuromorphic hardware. And it's also compatible with Pine, which is a simulator-independent language that, regardless of the environment that the programmer wants, it can use it for program different systems. We talk about the Pine more in the upcoming slides. And this system has been mostly used for neural simulations. Here is the Brain Skills machine, which contains 20 wafers. and that's 4 million neurons and 1 billion synapses in this single machine. So for comparison, you can look at the graph at the right side. Again, a comparison between the brain and this system, number of neurons, and also the number of synapses on the vertical axis. It's pretty. It can be considered a smaller system compared to the other neuromorphic systems, such as the Spinnaker. So the Spinnaker system, developed by Steve Ferber, the person who had been heavily involved in development of ARM processors, and you can see a prototype of the chips, the boards that are being used in the system up here. It contains, it's a 48-node system in this picture that it has 868 cores and 1,000 neurons in each core. And upon building of the final Spinnaker machine, which is a collection of these boards, we'll have 1 billion neurons and 1 trillion synapses, which is a major step towards better emulation machines. And this machine had been.
I just want to interrupt you for a second. Yeah. You have 2 to 5 more minutes, and we'll move to the Q&A.
Okay. All right. So this machine had been used for building line follower robots and information representation. And this is a machine with 500,000 cores, and this is visual comparison. And this is another project called True North chips under the Synapse project. Each of the yellow chips that you're seeing here is a True North chip, and this board that had been shown in here contains 16 million neurons and 4 billion synapses. But the limitation on this chip is that the programmer can only use integrated on fire neurons, and while you could in a Spinnaker system you could program your neuron or use HW neurons or integrated on fire neurons. More than any other neuromorphic system that we talked about, this system had been designed for applications, and it had shown good results in power efficient real-time object recognition. So I'll skip talking about the Human Brain Project and also the SPAN. But it's worth talking about the SPAN model because it gets back to the neuromorphic design systems. The SPAN model is the largest behaving model of the human brain, and it contains around 2 million neurons with 800 million synapses, and it can perform 8 different vision motor tasks. So in the future directions, NeurGrid and also the Spinnaker project, they are developing and implementing the SPAN project on the neuromorphic chips. And but something that we couldn't possibly cover in this topic in this short time was the commercial projects and also implementation of neural networks on FPGAs. For example, there's been development in FPGA implementation of neural circuits and neural networks FPGAs using the same program that I've talked about, Pine. And so reaching to the summary and conclusion, we talked about theoretical considerations, the enormous computational demands of whole brain emulation, also the difference between emulation and simulation. We also talked about the current status. We reviewed different neuromorphic hardware project, and also we talked about the SPAN and a little bit about the future directions and the references. And thanks for listening to this talk, and I'm ready for hearing your questions.
Great, thanks, Abulfaz. So looking at the online Q&A, looks like we might have some new ones. Do we have anyone in the live audience that has any questions while I flip through this? Yeah, I don't think we have any new questions on the Q&A link. But Keith is asking if you could comment or speculate on how to reach human brain efficiency.
Well, to reach human brain efficiency, that's a real good question. But the most important step that people have taken so far is to try to build these application-specific hardwares and also type of designs that the circuit usually comes online and works when it's needed, not all the time. The same thing that happens in the brain. So neurons start firing when we need them to fire, not all the time just firing and being in a standby mode, but firing at the same time. They're just silent, and when they're needed, they become active. And this is something that have been used in some neuromorphic designs too. Yeah. But I think Randall might have some input on this. I don't know. Yeah. I think Randall might have some.
Yeah. Yes, there are, I think, some questions here in the house. I certainly have questions myself as well. So let me go and ask that question, and then I'll repeat it to you, and you can answer it.
Okay.
So obviously, some of the motivation behind developing these neuromorphic chips is just like long-term brain emulation and study neuroscience. But I'm curious what maybe some of the nearer-term goals are that these companies have in mind for applications.
Okay. So the question from Brian is, obviously, some of the reasons for creating these neuromorphic chips and these companies is to eventually use them for something like brain emulation and for studying in neuroscience, etc. But what are some of the near-term goals? Why these neuromorphic chips are being created? Could you say something to that?
Well, yeah, that's another good question. So back to the slides. So I wanted to talk a little bit about the commercial neuromorphic chips. And for example, in the short term, we would see some of these chips, and actually they're being used in cell phones for object recognition, fast and efficient and low power object recognition. And I think it might be a driving force for the development of neuromorphic chips in the near future. So Intel had acquired a startup company, and also in UAV industry, they're using neuromorphic chips for different purposes. But what I can say is that efficient and low power object recognition is something that neuromorphic chips are doing really good at, and I think it might be a driving force for the for more development of these type of chips.
I'm unfortunately still not seeing any questions in the online Q&A. Are there any more in-house questions?
Yeah, we still have some in-house questions. And sorry, Alicia and Abul Fazl, that whenever, say, it seems like there is just dead silence and no one is responding to the fact that you stopped talking, it's just because the live stream is between 20 and 30 seconds late, and we're waiting for the live stream to end, and then I get back to you. So, it doesn't mean that everything's over and nobody's talking. Okay, so I'm not sure exactly what Keith Wiley is trying to do there, and maybe that's going to be useful right away. But first, let me ask a question that I had, and then we'll see also if there are some more questions here in-house. Maybe I missed something that you presented, because I was spending a little bit of time talking to Ted Berger in the kitchen area here. But maybe not. When I first saw the beginning part of your presentation, I noted that you were giving some examples of models of neurons and how they work and their implementation and how they could be used to replicate, say, spiking in an emulated brain. I'm just wondering if you could talk to the difference between, say, trying to use a model for a neuron, say a spiking neuron, that you have a model that you could put into a neuromorphic chip, and trying to match that to, say, the structure or circuit structure of a piece of brain tissue, and then trying to make sure that this is producing the same kind of output, versus take, for example, what Ted was presenting earlier, the situation where you are building up functions that are based on what you've observed in terms of spiking, and you're trying to model the probability distribution of spikes in a certain area, and then that you'd use those mathematical functions as the kind of thing that you'd want to implement. So there's this difference between directly implementing what you've been modeling based on how you observe the responses of the system, versus taking an abstract type of neural activity and trying to match that to what you found in a piece of brain tissue. And again, I might have missed a piece there, so I'm sorry if I'm making you repeat something.
No, I didn't talk about this topic much, but I touched upon the topic of level of abstraction, and on what level should we stop? Should we go down to the level of atoms and molecules that constitute the brain, or can we come a little bit higher and work on the neuronal level, or we should go to the circle level, as you said, or we should even go higher and go to system level? And I think that's an open question. We don't know. So it seems that the consensus in neuroscience is that the neurons are the building blocks, and we should build it on at the neuronal level. But I myself, I don't know. And I think it's an open question. It's still an open question. Although, if you just extract the probability distributions and implement them on a hardware, and it works, that we have seen in Ted Berger's publications before in the past couple of years, and it works. But we don't know if it works all the time, or I think there is still so much room for research to find out the exact level of abstraction. I think there is still so much room for research to find out the exact level of abstraction.
Yeah, so quick follow-up there. So in terms of structuring your tech review with these uncertainties in mind, how do you go about constraining, or conversely, leaving open enough the range of different implementation possibilities that you would look into if you're going to say, okay, we're talking about how do you best implement a brain emulation? Like, where do you, how broad do you make it? How narrow do you make it?
I think one reasonable opening would be start from the neuronal level, or at least at the synaptic level, and the neuronal morphology, and then in between we have the neuronal level description, and then we have the circuits. But I don't think that we should go beyond circuits into systems. So we shouldn't try to make abstraction, put the level of abstraction at the level of the systems. It seems plausible to do it at the level of the circuits, neurons, and maybe neuronal morphology. These three scales, I think they sound plausible.
Thank you. Let me just check for in-house questions. That possibly covers it right now. Now, before we switch to the next person, which I'm sure Alicia is happy to introduce the next person coming up, which I believe is me because I'm standing in for Ilya. I just wanted to mention that we have a technical issue coming up, which fortunately we've been warned about in advance. It's that in the using this Hangout approach with the live stream, there is apparently an 8-hour cutoff, which we're approaching. So at that point, we need to start again with a new session. That means that the link is probably going to change. And of course, we'll make that link available on the website, and we'll push it through everywhere we can just to make sure everyone knows about it. But I just wanted to let you all know in advance, once the time is due to make that switch, Our admin here, Javon Feinblatt, is going to let me know, and then I'll remind everyone again. But this is just an advance warning. Okay, back to you, Alicia.
So there is actually one question that came up on the Q&A for Abulfaz, if we can go back to that. He was asking, how much computation do you estimate is happening in each neuron?
Well, it's hard to estimate to how much computation is happening in each neuron. So, and there's another question saying, there's this idea saying neurons do not compute, and the brains are not computers. So, but if we want, let's put this question in this way. If we want to simulate a single neuron, how much computation do we need to perform? That might be a better question to try to address. So let's, as we discussed before, let's go back to this slide. And let's assume that we're using the Ishkevich neuron, and this Ishkevich neuron needs 13,000 operations per second. And then let's imagine that this neuron, the same neuron that we're going to simulate, receives and sends 10,000 synapses. So it's simplified, it's stupidly simplified, but let's just make the simplification. And let's imagine that each synapses per each millisecond needs just one operation. So if we do a simple math, it will be 13 million computations per second. This is an approximate number that we can think of right now. But still, I don't know if Randall have any opinion on this, or you want to add something, comments on this answer, or this question, actually.
No, I don't really have a particular comment to make because, of course, you know, the kind of computation that's being done, it depends on the abstraction level at which you're looking at the problem, or, you know, what you're going to include and what not and why. So it's too early, I think, to talk about that. We all throw out some, you know, some big estimates. That's where the exaflops come in, but I think it's actually quite early. So I'll give, once again, giving it back to Alicia.
We have one really popular question on the Q&A asking about how we could speed up the timeline that Ken Hayworth outlined for us earlier. For 2100 is the year for mind uploading. I'm wondering if we can apply that to this discussion here. Abulfaz, do you think that there's anything in neuromorphic technology that is part of why the timeline is so much longer, is so long?
So you're asking if there is anything in the neuromorphic hardware technology that makes it to be a longer time after 2100?
Right. So is there anything in this field that we could do to help speed things up?
Well, yes, we have already seen this in projects like Synapse project, that they have achieved marvelous numbers in terms of the neurons that they have simulated, or the skill that they're performing their simulations. One thing is that if the commercial companies and also the scientific community become more and more involved in this process, I think it might be it will definitely help so that we can have a possible emulation platform sooner than that time. But any forecast and anything, I don't think that it's, as Randall said, it's probably really soon to talk about this. But I think any process, any advancement in the neuromorphic technology will definitely affect that. And but on the other hand, I don't think that the neuromorphic technology would be the bottleneck for this, for the mind uploading process. And so what seems to me is the bottleneck is the way that people want to transfer the information into the emulation software and destination platform. So, but this destination platform is probably not the bottleneck process, the slowest process. That's my standing on this point. The bottleneck process is slowest.
Yeah, I actually have a comment on that. It's really interesting to see how these projections for what year is it going to be that you get a whole brain emulation, how they come about. And also, I found it interesting that Ken said at some point that his projection looked a lot longer than mine. Meanwhile, he had put somewhere in the middle of the 21st century, he had put the first upload, the first emulation of a human brain, which was kind of the same that I put on my slide. But you know, I think there's this effect, and it's kind of related to the Dunning-Kruger sort of thing, where you know, the less you know about something, the more likely it is you can be very optimistic, and as you know more, you become more pessimistic because you know the real problems that are there. So, for example, that's why we'll say things like, okay, it looks right now like the big problem is data acquisition. Once we have data acquisition, the next step's going to be really fast, and you know, emulation hardware is no big deal at all. That's something we'll just, you know, be able to do in a year or two, because why would that be a problem? We haven't really even had to tackle that because we don't know what we're going to try to emulate on a hardware platform. As we get closer to that, then we'll see just how hard that really is. So that's just a comment about how, you know, we make estimates about some things. We'll say like, oh, it's going to take forever to collect all the necessary data for an emulation, because that's the problem we're dealing with right now. Right, okay, so having said that, because throwing out these estimates without taking that into account, and without taking into account that it's not just what can you do and how fast could it be done, but also, is there going to be a project to do that, and is the money going to be available? Is there the will? Those sorts of things are not even scientific or technical. They're just economics and politics and whatever else. I tend to just give other types of answers. Like I'll say something like, I'd be super surprised if this got done in 20 years or less. But then I'll say something like, I'd also be super surprised, or I'd be sad for what must have happened to the Earth in the meantime if we didn't get this done in 100 years. Sort of saying, you know, it looks like the sort of problem that should be doable by this civilization in some time span like that. But then within that, where you peg it, it's going to depend on so many precise factors that it's just not even worth speculating. That's my comment on that anyway. I don't know if there are any more, if anyone wants to say something about that before we move on. No? Okay. Back to you, Alicia.
All right, so we're ready for our next presentation. Ilya is in charge of the neurotechnology section, but unfortunately he's very ill and is unable to join us today. So our very own Randall Kuna is going to be presenting this section for us.
Okay, thanks for handing me the mic again. So as you already mentioned, I'm doing this in Ilya's stead at the moment. And at one point, Ilya thought that he was going to prepare a recorded presentation for us, and that was what we were going to play. So I don't even have that. So instead, I'm just going to take his topic area with a small outline that was there and to try to formulate what it is that he might want to try to capture in doing a tech review of his area, which is basically neurotechnology relevant to brain emulation. So neural prosthesis, neural interfaces, brain-computer interfaces, that area. And I guess, you know, if I were Ilya, one of the first things I'd try to do is I would try to define those terms. So if we're going to be building a neural prosthesis, what is, for example, a satisfactory neural prosthesis? For what kind of goal? We're going all the way up to brain emulation, but there are, of course, a lot of steps below that. Say a patient with a dysfunction is going to be satisfied with something a lot simpler than somebody who is a healthy person and who is thinking, well, should I get my, you know, my hippocampus replaced, or my visual cortex, or my motor cortex, or something like that. And depending on those criteria, you might have to look for very different technology. Also, if I were Ilya, I would try to make very clear the distinction between creating neural interfaces with the brain, so a way to talk to neurons in the brain, a way to just get at data to be able to observe, access, monitor, and even write back, versus the process of creating a replacement. So neural prosthetics, neural prosthesis, the building of devices that can take over the function of an area of the brain as something rather different than a simple neural interface. And when I say simple, I don't mean it's easy, just rather different. So I would definitely focus on that. And then, then there's the question of what sort of data is most important for both for the access. So what is it you want to see? Do you want to see spikes? Do you want to see membrane potentials? Do you want to be able to probe the neurochemicals that are diffusing through some part of the brain at that point? Do you need to be able to register the level of LTP or LTD that's present somewhere? Does that matter at all? So there are questions about what type of data you're trying to get at with this neurotechnology, what you're trying to see with a neural interface or brain-computer interface, and also what you're trying to reconstruct. What are you trying to build? And then there's this issue of combining different types of data. So the one that we talk about a lot is the problem of combining functional data, so the responses, the activity you see in the brain, with structure, trying to use both of them as sources of information that will allow you to more feasibly create either a good interface or a prosthesis. And by that I mean, either way, whether you're trying to build a prosthesis based on functional recording, or if you're trying to build it based on structural recordings, like connectomics, you're faced with some serious challenges. If you're trying to do this using structural data, you're starting with something that is basically a three-dimensional image or a network of nodes with connections between them. But to get from that to something that's functional, how do you do that? How do you infer function from structure? For that, you need to have a really good understanding of how certain brain areas work, how various components of the brain work, the sorts of probability distributions there are for exactly what they may be expressing at this time, and how you have to sample from those probability distributions to build up your functional models. And all of that is information that you can gain by doing functional recording and by looking at what's going on in the system as it's operating. So the structural approach, which is the one that, for example, the 2008 Oxford workshop talked about most when we produced that roadmap back then, that one is totally undoable at the moment unless you bring in all this extra that wasn't talked about at the time. Conversely, if you're trying to build a neural prosthesis based entirely on functional data, then you have the problem that, of course, first of all, you're usually only looking at a subset of the cells that are really involved. How many do you have to look at? Do you need to see 20%? Do you need to see 80%? Is it enough to get by with 1%? To really capture what the function is of that system, of this little piece, this little black box that you're investigating. That's one question, but there's also the question, how long do you have to observe that black box to capture what's going on? Because, naively suspect that the only things you can know about the system are what you've observed. Now, if you're doing good machine learning, you can often extrapolate beyond that. You can build functions that generalize, that predict beyond what you've seen. But how do you know that those predictions are valid, that they're correct? So validating that system is trouble. So just because you can't observe that piece of the brain from birth until the moment where you're trying to build this prosthesis, that's a problem. And even if you could observe it throughout its entire lifespan, you don't know what things you haven't seen that just haven't manifested, but that were there in the blueprint from the beginning. So that's where trying to constrain your functional problem in a variety of ways and making that whole thing, making it computable, making it possible to derive parameters for your models, making it so that you better predict what it should be doing or may be doing. For that, having a lot of information about the underlying circuit structure, getting that structural data is very important. So the two things, these two data sources are very important, and it's important how to combine them. And that's where I would focus on trying to look at not just what are the methods out there that people use to either do functional recording or to do structure mapping, but to think very carefully about, well, how would you use this to combine the information you're getting into something that constrains your problem as much as possible and makes it as validatable as possible. And if you can use those two criteria as a guide for this investigation, as a way to, say, decide what is a really promising approach, what's a less promising approach, or what might need to be added, that's where it gets really interesting, if you can do a tech review with that sort of detail. Now, of course, that's a tall order, and I'm sure this is not going to be done in the first iteration, but that's sort of how I would approach the problem. And that's what I'll leave my version of saying what Ilya might have said at this point, and I'm open for questions.
So unfortunately, it's very quiet still on the online Q&A. Do we have any live questions?
Yeah, we've got a question here in the house. I'll repeat it as soon as I hear it. Daniel asked a fantastic question. He asked, so about the difference between trying to create a prosthetic that actually replicates the underlying neural code of a piece of a system of a neural tissue or neural circuit that you're trying to build a prosthesis for, versus one that merely accomplishes, say, the same or an equivalent functional role, but where the brain has to do some learning, where the plasticity of the brain is being used to actually be able to use the prosthesis. This is a very, very significant question because, of course, most of the work that has happened so far in neural interfaces as well as prosthesis here, the example Daniel asked about was cochlear implants, which of course don't operate at all like the underlying system. And also in interfaces where, say, people say, oh, you just have to stick an electrode anywhere in the brain and the brain can learn how to use it and move an arm, right? Move a robot arm or something. This is true, but I think there's a huge distinction there between the goals, what you're trying to achieve. In some cases, oh, by the way, I'm getting some feedback. Someone else is not muted. I think it might be Javon or someone isn't muted.
I'm sorry. Well,
yeah, I'm getting sounds from you over there. The
mic is off.
Oh, okay. Well, I'll just keep on going. So I think the big difference is the utility of the neural prosthesis that you've got. So for example, in the case of a cochlear implant, you've got a patient who couldn't hear at all or can't hear much. And they're very happy if they can learn to use any tool, be it a crutch if you're trying to walk or a cochlear implant, to just get the function they're trying to get. They don't care a lot about whether it feels the same as it used to, if they were hearing the same as they did before they lost their hearing, if that's the case. Similarly with people who get a retinal implant and all that. So that's a little different than, for example, the case of a healthy patient who has a very well-working piece of brain tissue that does something, and who, first of all, wants the function to be just as good, who may not want to spend a year learning how to use it. But then also in some parts of the brain, if you're trying to build a replica of what's there, it's very important to capture exactly what's there. Because let's say there's a portion of your brain that remembers what your parents look like. You can't just put an arbitrary system in there that can remember faces, because that no longer remembers what your parents look like. Now, of course, you could go and learn what your parents look like again. But the idea is if you were going to use this as a process for eventually doing a whole brain emulation to, say, copy what's going on inside of your brain, what you've remembered, your experiences, you can't just toss them all away and put in, similar functional pieces. Now, of course, if you're trying to use what you learn about a piece of the brain to then build an artificial brain that doesn't need to be anyone in particular, that's a whole different ballgame. Then you could just build functional components like that, and it should work. And I'm sure that there are gray zones and areas where this is all, you need a little bit more of the underlying detail or a little bit less. And it probably depends on the part of the brain you're working with as well, because maybe for most people who need who would like to get a brain emulation at some point, even if it's for a whole mind uploading project, they might be totally happy with a less precise emulation of their cerebellum as long as it still helps them carry out their motor functions and all that and the connection with the body. So this is really, in part, a question about what are the success criteria for mind uploading? What are the success criteria for whole brain emulation? What are the success criteria for partial brain emulation? What are the success criteria for neural prosthesis? And that's something that we have to iteratively figure out what those are and then aim for them depending on the project we're working in. So that would be my very long, unnecessarily long answer to that question.
There, we often think about developing emulations of parts of the brain or neuroplasticity in the context of creating the function of that system. If we're thinking in terms ultimately of using that model in the context of prosthesis, we're thinking about putting that model of that system into another system that's malfunctioning.
Mm-hmm.
There's a difference between part of the brain being missing and a part of the brain being present but generating the wrong signal.
Yeah.
So if we're going to interface a prosthesis or a neural model with the existing brain, we have to think often about how to modify what's in the existing brain to make it correct.
Mm.
And that's very different from the way we usually think about developing the prosthesis.
Yeah. Nothing. Apparently I was on mute trying to say something that you couldn't hear before either. You couldn't hear what Ted Berger was saying, which was a very good comment on why you have to care about capturing some of the underlying coding even when you're building a neural prosthesis for a functional goal like helping someone who has a dysfunction, because in many cases, like for example in the case of the hippocampal prosthesis, you have to put this device, this prosthetic device, back into a malfunctioning or partially functioning piece of the brain. It's not that that piece is necessarily entirely missing. First of all, that new device needs to be able to communicate and operate successfully with the remaining part of that piece of the brain and all of its surrounding neighboring parts that communicate with it. Now, yeah, so in that case, it's important that that these neural codes you're producing, that they're intelligible, that it makes sense in the context where you're trying to strengthen or trying to improve, trying to heal, trying to correct the coding that's going on. That is not always correct in the case of the patient's situation. I do remember that we've had several debates in the past about whether you could, despite that, force the patient's brain or the surrounding regions, the receiving area, to learn to use whatever codes, whatever strong codes you can create. So I think, again, even though that's true, there's probably some gray area and some debate about what's really usable. But that's a really good comment. Thank you. Okay, so any more in the house questions at this moment? Okay, I don't see any. Alicia, do you have any more there?
Yeah, someone asked if we could clarify OpenWater's claim to have achieved 1 billion times higher resolution than MRI. Can you speak to that?
Okay, so you're asking, can I speak to the claim by OpenWater to have achieved something like 1 million or 1 billion times better resolution than MRI? Yeah. Mm-hmm. Oh, okay. So I think if you, because MRI is like 1 second IR spectroscopy, which is. Okay, so Daniel here had the answer for that. He's suggesting that where they get the number from is not just the spatial resolution, but it's a combination of the spatial resolution and then multiplied with the temporal improvement in the temporal resolution. If you're going down to or going up to a much higher temporal resolution than the MRI could achieve, that's where you get that big number from. Now I know how weird it is to wait for that live stream to catch up. Okay. Okay, I am not hearing anything from Alicia, so I'm assuming that there are no further questions from the
crap,
board.
I was muted.
Oh,
Sorry.
you were talking. Okay. Yeah,
I
this is
was
still tough.
talking.
All right.
Okay, so Keith posted a question. It's kind of a three-part question, so I'll do one at a time. Ted said he successfully determined firing patterns associated with certain concepts. Did those patterns persist between subjects?
Oh, okay. So this is actually a question for Ted from Keith, who was saying, if I don't know if I'm getting it correctly because I don't see the question itself right here. Actually, I would like to see it, so I make sure I get it right. Oh yeah, here. Yeah, that's okay. I've got it. So Keith says Ted said he had successfully, or 50%, determined firing patterns associated with certain concepts, animals versus non-animals. Did those patterns persist between subjects? Was there a discovered universal representation of the animal category, or did it only categorize in the context of one subject? And gosh, I wish that Ted could just talk straight into the mic. Oh.
The answer is we haven't tested enough.
Okay.
We don't have enough subjects.
All right, so Ted is saying the answer is we don't know yet because there haven't been enough subjects to figure that out.
Another month.
One more month, he says. Okay, Alicia, are you muted again, or?
Yes, sorry. We ready for the second part of the question, or did you get to all of it?
I think we got to all of it. And yeah, so maybe we either move on, or if there's time, we could take a break in between if there's still time left.
I think we're ready to
doing?
move on. If everybody
Yeah?
wants to take a break, though, we can pause for 2 minutes.
No, we can move on for now. Then we can take a longer break a little later. Okay.
Okay. So the next section we want to discuss is the brain mapping section, and this is led by one of our team members who would like to remain anonymous. We're calling him John Doe. John, whenever you're ready. John, are you there? We're ready for you.
Connect for a moment, but hold on, let me put that right quick. Okay. Can you hear me?
Yes, I can hear you.
Cool. And screen?
It is breaking up a little bit, though.
But I'll get through what I can get through here. So, first of all, I did want to say thanks to the speakers and thanks to Randall for orchestrating the workshop. Been incredible, high quality talks with such a serious scientific treatment of emulation. Now, as part of our tech review project, and wanted to cover a few things here today. I'll be talking about what I mean by the brain mapping problem, touching briefly on its major challenges, talking a little bit about brain mapping, current initiatives, a little bit about economic drivers, commercial opportunities, and then just to finish on a selfish note, I was not expecting the quality of the questions that we've had. I was a little starstruck by our speakers, so I'm looking forward to having a discussion to wrap up all of you. So, David famously described and whether the organization of realized in silicon chips in the population of China or in beer cans and ping pong balls does not matter, as long as the functional organization is determined. And
Jesse, we're having a hard time hearing you. Are you trying to share your screen?
5 billion neurons. But
John? We're having trouble with the audio. Maybe we could take a pause and come back.
Sure.
Okay, let's go ahead and switch to Bill Krugen, who is in charge of our computational brain model section. Sorry to jump right to you, Bill. Can we maneuver things around there at the live stream or the live audience? So I'll do a brief introduction of Bill while he's getting ready. Bill is a Louisiana native who studied computer science at Carnegie Mellon University. And he is currently working toward his PhD at UC Berkeley for advanced studies. You're on, Bill.
Hey, can you hear me?
I can hear you, yeah.
Okay, great. Cool. The background, maybe if I move. All right. I'm going to try to avoid some feedback real quick. Okay, let's try this. So we're talking about the section of the technical review about computational brain models. And basically the way that this fits into the bigger picture is you've heard a lot about neuromorphic hardware on which we might implement some of these things. You've heard a lot about how we scan a brain and try to get a good 3D reconstruction of the brain. But even if we had a perfect three-dimensional reconstruction and the world's greatest supercomputer, we would still need to know the equations and how to describe how neural activity changes going forward in order to actually run some type of emulation of a brain. So this section will just kind of get at how we derive these mathematical models and talk about some of them that are being researched today. So there's three main sections that we'll talk about in this section. The first and the main focus of this will be on single neuron models. So how do we just model the activity of one neuron if we know all the inputs and try to generate the outputs? And theoretically, if you can get a really good single neuron model, then you can just combine a bunch of them together for to model an entire circuit of neurons. And then I'll also talk a little bit about system models. So if we try to abstract away from the activity of individual neurons, can we still model how that system behaves over time? And then finally, I'll briefly touch on some of the current large-scale simulations that are in progress and what they contribute to the field. So on the left here, we have maybe the most simple single neuron model that many of y'all might be familiar with. This is what you'd see in artificial neural network. And it's inspired by a neuron because it has inputs which come from other neurons. It does some transformation on them and it has an output. the reason this is a very successful neural model, and things like deep learning nowadays is using those artificial neural networks. And it's so successful because it's extremely computationally efficient. So we can implement this on a GPU and run hundreds of thousands of these, millions of these in parallel, and it works really, really fast. But it exists on the spectrum of trying to balance computational efficiency with biological realism. And obviously for something like whole brain emulation, we need both computational efficiency and biological realism. And this model that I've introduced on the left doesn't really capture a lot of the biological realism of a single neuron. And just to illustrate what it's missing, here on the right are two example neurons from two different parts of the brain. And you can see the top one has very different morphology than the bottom one. And presumably these two neurons have evolved these morphologies because they perform different computations in the brain. The shape of these is presumably very important to the computations they perform, and you couldn't just take the top neuron and put it in place of the bottom one and expect that circuit to work the same. So all of that variation between different neurons is kind of lost at the cost of computational efficiency in these artificial neural networks. So we're talking about, in this section, going into where current research is on trying to balance these two things and get the best of both worlds in terms of modeling biological realism, but also retaining that efficiency. And I'll briefly just touch on a couple of examples from recent years. So on the left, if you were on this live stream earlier, you saw Ted Burgess talk about his hippocampal prosthesis. And this is from one of those related papers talking about how to model individual neurons' output in terms of their inputs. And here, he's abstracted away from every little compartment of the dendrites of different neurons by just considering each neuron in the input as one point with a certain spike train, so a certain pattern of inputs, and then modeling just a very simple output spike train of that neuron. And so this can be done very efficiently just by implementing these functions directly on a CPU and not worrying about maybe modeling the entire complexity of the shape of different neurons, but the math is such that you can still capture a lot of the computations that are happening within that single neuron. And so that strikes a good balance between biological realism, computational efficiency, by doing that abstraction of one layer. And on the right is just another example, getting at the same question a little bit differently. So this lab is trying to basically model what different types of computations happen in these dendrites. And here, this is from their paper where they looked at different types of inputs on different dendrites and whether those inputs were spread across the cell and how correlated they were in time, and basically different variations of inputs that you would see in a brain. And they tried to extract some computational rule of how the neuron was exactly integrating these inputs and constructing an output. And so this again gets at, we abstract away from neurons and come up with something that retains as much behavior as possible, but while allowing us to efficiently implement it. So we can balance those two competing drives as much as we can with single neuron models. But there's another possible answer to this dichotomy, which is what if we can abstract even away from individual neurons to try to describe the behavior of a whole section of the brain, of a whole neural circuit at once. And so on top is kind of one example of this type of thinking. So this is a deep convolutional neural network. And these were originally developed for, or I should say, gained a lot of success originally by doing visual input processing. So you show it a picture, and the picture input is on the left here, and that picture is processed more and more by these different layers of artificial neurons as you go from left to right. And the first layer might respond to something like very low-level features like a line in the photo. The second layer will respond to slightly more complex features, maybe like a circle, and the third layer will respond to more complex features, until eventually you go down this entire list of layers and you get to the end, you have maybe one unit that responds to if the entire picture is of a dog, and another one that responds to if the entire picture is of a cat. And so this is a very successful architecture, and it's based on, originally was inspired by trying to model the visual system. So on the bottom is a very oversimplified version of the human visual system, but you have the same overall architecture where you have the input is an image that goes into the retina, and then as it goes through the thalamus and then into the cortex with these different areas, V1, V2, V4, you see a very similar architecture at first glance where V1 neurons tend to pick out individual low-level features of images, and then V2 neurons will pick out slightly more complex features, V4 neurons slightly more complex features. And so the advantage of these system models is you can imagine if this was all the visual system was doing was ultimately going from visual input to very complex features and categorization, then we could implement that entire system much, much more efficiently with a system model like the deep convolutional neural network that we designed from the ground up to be computationally efficient, and it would recreate the same behavior. Obviously, in this particular example, the visual system is a little bit more complicated in this. It does a lot more things than just pick out whether there are pictures of a dog or a cat. And so that's why these deep convolutional neural networks don't describe the entire behavior of the visual system. But this just serves to illustrate why these system models are important to develop, and down the road as they're being refined, how they might come into play in some type of emulation environment. And finally, I'll just briefly touch on this. So most of the section will be about the different models that people are developing, and then I'll also touch on the models that are currently being implemented in things like the Blue Brain Project and Allen Institute for Brain Science Human Brain Project. So these are kind of the closest things we have so far to validating these the behavior of these models as they're applied to very large neural networks on very big supercomputers over a long period of time. And so I'll touch on kind of the results that each of these organizations has contributed to our understanding of computational brain models. For instance, the Allen Institute for Brain Science implemented 2 different patches of cortex that they were modeling, one with a very simple point neuron model, which is equivalent basically to the on the first slide, that very simple artificial neuron that I presented. And then the other model, they did a what they called a biophysical model, which is much more complex and takes a lot longer to simulate, but reproduces the activity of individual neurons much more with much higher fidelity. And so then they were able to compare and contrast the different behaviors that came out of these 2 models and talk about where some models were appropriate for some questions, but maybe failed at answering other questions, and maybe didn't reproduce the full behavior of what they were expecting. So that's that'll be the final thing we'll talk about it in this review section. And with that, I'm more than happy to answer any questions that you might have about anything I've discussed, or I'd love to hear what else you think would be interesting to talk about in this section.
Thanks, Bill. The online Q&A is quiet again, but Keith did post a question. He says, is Bill just describing a standard perceptron or some modern variant?
Yeah, basically just a standard perceptron. So I guess what he's referring to is this artificial neuron from the beginning. And yeah, I was just putting this as an example of what would be one quote neuron in one of these larger neural networks as a very oversimplified version of a biological neuron. But yeah, I think in most of these cases it is implemented as a perceptron. Right, but it totally doesn't capture kind of the grander complexity of these individual neurons.
Other than that, it's still pretty quiet online. Do we have any in-house questions? Other than that, it's still pretty quiet online. Do we have any in-house questions?
So, when you're not talking to yourself,
Hi, sorry, Alicia, did you ask about in-house questions?
Yeah.
Yeah. So I don't know if there are in-house questions by others here, but I certainly have a few questions of my own.
Alicia, did you ask about in-house questions?
Okay, keep that from doing this twice. So I'll just start and ask a question. You went through a number of different models. Thank you, Bill, by the way. Thank you very much. And it's terrible that we're not doing the clapping for the people who are doing this tech review. We probably should. So Bill, you mentioned these different levels at which you can do a model, different types of models. And I was just wondering, because single neuron model seems like an easy thing. Like just do a single neuron model, and then if you can put the individual neurons everywhere, great, the whole system should work. But is that actually true? I mean, has anyone ever made a good single neuron model that really predicts the activity of a real neuron?
Well, I guess a lot of that answer depends on what you qualify as a good prediction. So it depends how you want to validate that model, right? If you, for instance, you can do something like Ted Berger's hippocampal prosthesis, where your model is validated by being able to recreate in an actual brain with your model of a neuron some activity of a real brain. So that's one level of validation where, yeah, you can make a single neuron model that does kind of function in some way. But the question of whether it functions perfectly like a real biological neuron, I think, is very difficult to get at, because there's so many levels of complexity, right? I don't think we've even totally captured as a field how dendrites fully contribute to how a neuron operates. For instance, there's some, for a long time, the field kind of considered dendrites to be these passive input receiving areas of the neuron, and then everything gets sent to the body of the cell, and then computation happens. But now there's a lot of research coming out about active dendrites, where they're actually actively performing computations in the dendrites, and I touched on that a little bit. And of course, there's even subcellular things like the exact organization of receptors on the cell and different transcription regulators that occur over the course of neural activity that is crazily complex to model, and I don't think we even have a good idea of how to validate one of those models yet. So the answer, I guess, would be on some level, yeah, we've implemented these single neuron models, but definitely not to the level of what you might consider like perfect biological realism.
Thank you. Sorry, Diana, did you have a question? Is it? Oh, I think we still have time. Yeah. So you want to come ask it? You can ask it straight into the microphone. Yeah.
Hi, can you hear me? Thanks very much for your talk. Okay. So I had a question about, it's sort of a follow-up question on the previous one. I was just wondering, in terms of system identification for single neurons, what do you think is the correct way to start? You mentioned all these details that probably play a role, like ion channel distribution, neurotransmitters, active dendrites, and so on. So let's say, I mean, there must be, in principle, there should be an equation that can describe the behavior of a single neuron. And how would you go about adding parameters? Is there a certain order to it, like in terms of importance? What contributes to that? And especially when we add functionality and behavior to that. Yeah. Thanks.
I think a lot of it is probably limited by what we actually have experimental access to. So we're only very recently being able to probe things like the exact arrangement of receptors at a synapse with very fine detail, fine detail resolution, and actually being able to see that in real time and correlate kind of the structure to the function, as people discussed earlier. That's something that we're still definitely working on. So in terms of what you should model, I think currently the cop-out answer is it's what you can model, right? If we have enough data to basically validate whatever your predictions are, then yeah, you should include that in your model if you're looking for perfect biological realism. In terms of kind of mapping out what would be the most kind of important features of a neuron to model, I think that's not totally clear right now because there's, as I mentioned, there's a lot of different interacting things and even things like the how DNA is regulated in response to neural activity. I think is a hugely potentially complex interaction that hasn't been explored in nearly as much detail as many other aspects of neurons. So I think it might be too early to tell exactly where we should go, but luckily we don't directly have to answer that question necessarily because it's kind of just whatever we can access. We need to develop tools to get better temporal resolution, better spatial resolution on these things. As soon as we can get the experimental methods down to investigate quantitatively how these neurons act, then yeah, we should include those observations in our model as well.
I've got another question from Keith on the online Q&A. If richer bio-inspired or morphology representative models still produce the same spike train behavior, then doesn't that just suggest we don't need those richer models? Or are there further theories of neural behavior that would require such models even if they fire the same way?
I suppose if it perfectly recreated the relevant output, then yeah, you're absolutely right. You wouldn't need the morphological details. I guess it depends on a couple things. What do you actually consider your relevant output? So maybe the exact spike trains for a short amount of time that you validate on are correct, but you still need to capture things like plasticity and how the spike trains will develop over time, and maybe that's not captured in one of the simpler models. It also depends, right, how you're going to apply these models. So for things like the hippocampal prosthesis, again, that we heard about earlier, it seems that just recreating the estimated spike trains does recover a lot of the function. So if that's kind of your metric, then yeah, that's as deep as you need to go. But if you have other metrics, such as trying to maybe simulate things for drug discovery or something on a lower level, or things like the neuromuscular junctions, so where a nerve attaches to a muscle, if there's some kind of organization there based on the exact orientation of the receptors and things like that, that would obviously require maybe a lower level explanation. So I guess it's difficult to answer exactly where we should stop in terms of complexity of modeling. That's kind of one of the big questions of mind uploading and whole brain emulation. But yeah, totally depending on what your application is, if you consider your relevant output to be, say, spike trains, and you have a model that gives you good enough spike trains to whatever measure you want, then yeah, that's the simpler the better, and then the more computationally efficient the better. So that's totally application dependent.
Great. So things are still pretty quiet online. Do we have any more in-house questions for Bill? Randall, if you're talking, you might be muted.
Okay, so I was muted apparently. I was just saying I had a similar question, and I think Brian already did a good job of answering most of that anyway, because I was going to ask something along the lines of how can you evaluate the promise of a given model, how to test its possible use in brain emulation. And I think this kind of comes down to the same thing. You know, if you can only record from a certain proportion of neurons, then, for example, it's really important that you can try to model to some degree what you don't know, that you can model the rest as some kind of noise or some sort of other contribution that you can't actively record. And if your output is producing still a spike train that is plausible, that follows the same kind of probability distribution, then you're fine. Or maybe not. And again, this depends a lot on what we learn as we go along. So I think Brian already answered most of that by just saying, you know, we have to dig in and find out why some things are more useful or less useful in a given case. Do we have any further questions on this in the house? Anyone there? Nope. Okay, so we have no more in-house questions on this topic, Alicia.
All right, we have no more online either, so maybe now would be a good time to switch to the new live stream link. All right, where can I tell people to go for this new link? We'll go to John Doe's talk after the switch. Everyone will want to go to carboncopies.com/live, carboncopies.org/livestream for the new link. Randall, if you're talking, you're muted.
Thank you. People should head over to that link to carboncopies.com/livestream. That's sorry, carboncopies.org/livestream. I'm just reading it straight off the chat and making the same mistake Alicia made. So go back to the original page on the live stream page. You find a new URL there. That's the one that we're going to be using from here on out because this one is expiring. And I guess we as speakers here are going to receive a new URL for the Hangouts as well, I assume. Okay, right, we're making the switch now.